finally, on the dais, Dr. Milind Shiralkar, our Dean, R.D.GARDI Medical College. Knowledge grows when shared, innovation flourishes when minds connect, and breakthrough emerges when disciplines unite. On behalf of Rukshmani Deepchand Gardi Medical College and associated institutions, I extend my heartfelt welcome to every participant present over here today. We are particularly honored to host internationally acclaimed experts whose pioneering contributions have transformed the field of genomics, proteomics, system biology, transformational medicine, regenerative therapeutics, biomarker discovery, etc. Your presence enriches this symposium and inspires the next generation of future doctors, scientists, and academicians, as well as clinicians. In the Indian tradition, every auspicious beginning is marked by invoking knowledge and wisdom. The ceremonial lighting of lamp symbolizes the triumph of wisdom and knowledge over ignorance, discovery over uncertainty, and hope over limitations. May I request all the dignitaries on the dais to proceed for the lamp lighting ceremony. We now seek the blessings of Goddess Saraswati through Saraswati Vandana.
As a token of our respect and gratitude, we shall now felicitate our distinguished guests. May I invite Dr. Gavarikar sir to felicitate Dr. Joshua Lebar sir. Our Dean Sir, Dr. Milind Chiralkar Sir, to felicitate Dr. Harish Vasudevan Sir, please. Dr. Kalpana Mahadik Ma'am, to felicitate Dr. Yoken Martin Sir. Dr. S.P. Dhaneria Sir, to felicitate Dr. Sanjeeva Srivastav Sir. Dr. Ashish Sharma Sir, to felicitate Dr. Prashant Agrawal Sir.
फ्लैश लगाओ ना यार, फ्लैश लगा ना।
Dr. Shailen Sharma Sir, please felicitate Dr. Gavarikar Sir. Dr. H.M. Mangal Sir, to felicitate Dr. Mahadik Sir. And Dr. Ashish Pathak Sir, to felicitate our Dean, Dr. Shiralkar Sir, please. Let us all express our appreciation to our distinguished guests with a warm round of applause. So with this introductory felicitations, I thank all the distinguished guests to be present here. Now we will start with our series of lectures, and also I will ask everyone to please take their seats in the auditorium. To welcome all, I now have the honor of inviting V.K. Mahadik sir, our Medical Director, R.D.GARDI Medical College, to deliver the welcome address.
Good morning, everybody. Now, as the people had the knowledge of this symposium, Multi-Omics, so at least 200 people asked me, what is multi-omics or what is omics? And I said, to know that, you will have to come to the symposium. So that is the present state of knowledge about the omics in general population and even in doctors. I am really grateful to Sanjeev sir, Professor and head of the department, which his lab has wonderful machines doing various multi-omics. I have been there, and he will be showing you the actual how do they do it in the afternoon session. Now we have such a distinguished faculty that to fetch them from Sweden or America, it's difficult. They are tremendously busy, clinicians as well as researchers, and have their credit hundreds of papers. And I am sure that a day is not far when the omics will take over the major diagnostic criteria and markers. They will develop markers, and you will know the presence of cancer or susceptibility of cancer at least 10 years ahead, or maybe from the birth. So that is the magic of omics. I don't think that I am competent enough to say more than that. So welcome you all again. Thank you so much.
Thank you, sir. Your guidance and your inspiration is always with us, and we all, the entire institute thrives because of you. Thank you very much. After this, I would like to invite the President of UCTH, Dr. S.B. Gavarikar sir, to say a few words.
Good morning, everybody. On behalf of the Charitable Trust, R.D.GARDI Medical College, I welcome you all here. The symposium on multi-omics, and apart from many other, I was also the one person who went and asked about what this is all about. Anyway, gathering information, we had been reading about genes, proteins, markers and these things. But this field is now really coming into forefront, and how we practice medicine is going to change in a couple of years or 10 years time. There is going to be precision medicine. The experts will be speaking on it, and there is proteomics, and there is genomics, and changing the genes, transforming the genes, producing mRNAs and whatnot. The field it is like a Isaac Asimov travel through the space. Something is like that happening in the field of medicine. I am not a competent person to speak about it. I welcome you all here, and thank you very much.
Now we move on to our next session, that is the session for lectures. First of all is the plenary lecture. The future of precision medicine rests upon our ability to understand biological systems in their entirety. Multi-omics technologies are making that vision a reality. It's now my privilege to introduce our plenary speaker, Dr. Joshua Labelle from Arizona State University, United States of America. Dr. Labelle is internationally recognized for his Dr. Labelle is internationally recognized for his pioneering contribution in biomarker discovery, proteomics, and precision health. As the founding director of the Virginia G. Piper Center for Personalized Diagnostics at Arizona State University and former director of the Harvard Institute of Proteomics, Dr. Labelle has transformed the landscape of biomarker discovery. His innovative NAPPA protein array technology and groundbreaking work on autoantibody biomarker have led to novel approaches for the early detection of cancer, including the development of the Vidisha breast cancer diagnostic test. Today, he will share his insight on the exciting field of cancer proteomics. Dear all, please join me in welcoming Dr. Labelle.
All right, thank you so much, and I just want to give my heartfelt thanks to the invitation to come to Ujjain. I've never been here before. It is a beautiful city, and this is an amazing auditorium. What a beautiful campus you have. All right, so I'm going to ask to have my slides put up. Oh, I thought they... Were we supposed to do? Yeah. The COVID pandemic, which was understanding who was going to respond well to vaccine and who is not going to respond well to vaccine. But in order to get you to understand how we looked into identifying people who would struggle responding to vaccine, I need to tell you a little bit about the technologies underlying how we did that study. So if I can have the next slide. That's one too fast. Yeah, back. Okay, is there one before that? Go back. One more. No, back one. That. Okay. Yeah. So years ago, in fact, with Dr. Srivastava's help, we actually developed a technology called nucleic acid programmable protein array, or NAPPA. And basically, I'm not going to go into a lot of details here, but conceptually, what we do is we print genes for proteins on microscopic arrays, And then at the time we want to do an experiment, we synthesize the proteins in situ on the chip, make full-length proteins, and then we can study those proteins, for example, for their ability to be detected by antibodies or for interactions with particular components. Next slide. The key of this technology is that it doesn't require us to purify proteins, we just have to purify genes. It makes very consistent protein levels. And because we're making it with human ribosomes and with human chaperone proteins, the proteins are well folded, they take their natural shape, which allows us to study how proteins behave naturally. We can easily design these arrays to be custom, and they're quite stable because they're effectively DNA on a chip until we turn them into protein. If we go to the next slide, the challenge of the protein arrays was that although they are outstanding at screening many, many, many proteins, currently we can screen 20,000 full-length human proteins. The challenge is it's not inexpensive to test that many proteins. And most experiments don't require looking at the entire human proteome. Once you've identified a subset of proteins that are of interest to you, you really don't need to keep looking at everything. Moreover, we really, to identify a biomarker that you can predict will be persistent and useful in clinical populations, you need to test them in large populations. Typically, you want to look at thousands of people. And the protein arrays are just simply too expensive to look at for thousands of people. So we really wanted to develop a population scale protein array, and that's what I'm going to describe today. If I can get to the next slide. The goal for this study, this technology, was we wanted it to have a throughput of thousands of samples. We wanted to be able to test many proteins, so at least hundreds of different proteins. And we wanted it to have a large dynamic range so that we could look clinically at a large scale of signal strength. We wanted in-solution binding, we wanted natural folding, and it had to be compatible with working in the clinic. So in the next slide, what we ended up developing was a solution-based protein array. And the idea behind this array is that we have a mixture of proteins, and each protein is uniquely tagged with a barcode, a DNA barcode. And we mix all those proteins together, and in the next, if you can just hit forward. And once again, you look to see which proteins bind to your serum, and then, next slide, you just read that result using a barcode reader. That's the concept. Let me show you how it's done in the next slide. Conceptually, what we do is we covalently attach DNA barcodes to proteins. We mix the proteins together so that they have roughly similar amounts of protein for every protein in the mix. Incubate that with whatever serum we're testing. Separate out the bound proteins, and then do a brief PCR amplification of the barcode. And during that PCR amplification, we can add another barcode, this time for the sample. So now we have a sample barcode next to the protein barcode. That allows us to mix many samples together, and we can look at literally thousands of samples in a single experiment. And so in the end result, we have the throughput of roughly 2,000 samples per run. We can look at anywhere between 200 and 1,000 proteins per run. We're still doing solution binding. Because we're doing DNA sequencing, we get 3 to 6 logs of data, and it's compatible with the clinic. If I can go to the next slide. So now I want to show you a little bit about the technical performance. Next slide. This just shows you that when we use our technology compared to commercial FDA-cleared assays, we get similar results. So if you can hit forward for me. Yeah, if you look in that box there, you can see that we have roughly 3 logarithms of signal from the top to the bottom. And once again, now you can see if we look at samples that have been defined by a clinical grade assay for being positive or negative for COVID response, that we get virtually the same answer that they do. So the agreement is well over 95%. Next slide. One of the surprising findings with this approach was that there was very consistent levels of antibodies in the blood over long periods of time. In one of our assays, we looked at a population of individuals, about 137 people, who had had blood tests for 150 different proteins, so antibodies against 150 different possible antigens. And that was taken in the fall of 2021, and then again 6 months later in the spring of 2022. And we compared their antibody levels for each of those 150 proteins over that 6-month window. And what you're looking at is a scatter plot of signals for each of those proteins. And from, if you can just click forward once, what you can see here, and click one more time, looking at this one, for example, this is an individual whose blood was measured twice. Indeed, when we talked to that individual, it turned out that they did have COVID during that 6-month window. So we can effectively use this test to measure the presence or absence of clinical events. Next slide. So now what I want to tell you about is a large population study that really is the core to this question about who responds well to vaccination and how we can identify them. If we can get the next slide. We did a large study as part of a program called SeroNet. This was a National Cancer Institute sponsored study in the United States during COVID-19, looking at a large population of people before and after their first time ever vaccination for COVID-19. So COVID-19, as bad as it was, did provide us with the opportunity to ask the question, how do people respond to a brand new virus? How do they respond to something that their body has never seen before or that the population has never seen before? So you can see in the red box the schedule that we used to detect the serum responses, starting with pre-vaccine, and then several tests post-vaccine, and then pre-booster, and then post-booster. If you click one more time, you get an idea of the number of samples we looked at. We had over 4,000 participants in this study, and most of them had multiple blood draws. So we had approximately 9,000 samples in experimental samples, plus another 3,000 control samples that we added to ensure that we were getting consistent results. If you click one more time, in addition to looking at the vaccine response to COVID-19, we also looked at antibody responses to over 160 different human pathogens. So we looked at something like 40 different bacterial species and almost 100 different virus species. Things like adenovirus and rhinovirus and herpes virus. We looked at antibody responses to all these different organisms to see what was happening in our population over that time period. If I get the next slide, this is what those data look like. So what you're looking at here are antibody profiles to about 184 different antigens in roughly 9,000 samples. It's kind of hard to look at all this data, but there are a couple of things I just want you to pay attention to. If you look in the red box, you can see some element of streaking that kind of looks down that column. That particular population was a multiple myeloma population, and those patients are immunocompromised, presumably both because of their cancers and also because of their treatment of their cancers. And you can see that they tend to have lower antibody responses overall. Not surprising, since their cancer is a B-cell related cancer and because the treatments kill B cells. I neglected to mention that in this study, about half of the participants were healthy, and the other half of the participants were people with various forms of immunocompromised conditions. That is to say, they had either autoimmune disease, cancer, HIV, organ transplants, anything that might lead to them having a reduced ability to mount an immune response. If I can get the next slide. Oh, yeah, I go back one. Yeah, there, stay there. you can see in this little red box, this sort of bright yellow signal strength, those are antibodies to the HIV virus. Not surprisingly, you see a lot of signal among the HIV patient population, just showing that the technology does what you expect it to do. But if you advance one more slide, if you look carefully in that little red box on the far right, you can see that there are some healthy patients, that is to say, self-identified as healthy, who also had antibodies to HIV and probably had HIV infection but didn't know it. And that's what a tool like this can detect. Okay, we'll go to the next slide. So looking at immune responses to see who had trouble developing antibodies, you can look at these different cohorts of patients. On the top left, you can see the healthy cohort. They had an immediate and strong response to vaccine and an even stronger response to booster. HIV, for the most part, also had a pretty good response, not as good as the healthy. But keep in mind that these HIV patients were well managed and typically had good CD4 counts. Now when you get to things like multiple myeloma and solid organ malignancies, you can see that there was a much weaker antibody response. And then inflammatory bowel disease, autoimmune disease, and solid organ transplants also had much weaker vaccine responses. And so clearly, the immune-suppressed population has weaker vaccine responses than the healthy population. But a couple of things worth mentioning. First of all, not everybody in the immunosuppressed populations had weak antibody responses. In fact, many of them had just as good a response as the healthy patients did. And so just being a member of a particular cohort doesn't predict whether you'll have a good vaccine response. The second point is that even among the healthy population, around 5% of the healthy people had weak vaccine responses, which is to say that even among the healthy, there may be people who don't have strong vaccine responses. Okay, so we'll go to the next slide. This is just a plot of where there were particularly big drops in vaccine response, and you can see, as I mentioned before, multiple myeloma, autoimmune disease, solid organ transplants were among the ones that had the weakest response. We'll go to the next slide. As I mentioned, in addition to looking at the SARS-CoV-2 proteins, we also looked at a lot of other microbial antigens. One more slide. These included all of the seasonal coronaviruses. We looked at other respiratory viruses, RSV, HSV, HPIV. We also looked at viruses of interest like herpes viruses, adenoviruses, papillomaviruses, various gut viruses, and a number of different bacteria to determine if any of them had any response to the COVID pandemic. Next slide. What we found is overwhelmingly most of those antibody responses did not change over the pandemic. The ones that you see on the right-hand side of the slide are the only ones that really had an appreciable variation during the course of the 1.5-year study we did. If you can go forward one, you can see in the box there, by far the one antigen that had the most variation was, not surprisingly, the receptor binding domain or the spike protein of COVID-19. And that's because all of these people got vaccinated for COVID-19. If you go one more advanced, you can see that the second most common response was to the nucleocapsid protein in COVID-19, and that is the response that indicates a person has been infected with the virus. And you can see that we had a particularly strong response right around the time that the Omicron virus came out, when there was sort of a resurgence of the pandemic, if you remember that. Other than that, there were a couple of seasonal viruses that also showed responses, which you can see here, but for the most part, most antigens didn't change. Okay, now we can go forward, and here you can see that a number of these other antimicrobial antibodies were especially prevalent. What you see in dark red are responses that were present in nearly everyone. So there are some viruses that all of us have been exposed to in our lifetimes, and that nearly 100% of us produce antibodies to. And then in blue, you can see there are some viruses that are very uncommon and that most of us don't have responses to. And we wanted to know if any of these common responses had any bearing on whether someone had a good or a weak response to vaccine. If you can go forward one. And surprisingly, when we did this study, we found quite a few of them in fact did correlate with vaccine response. That is to say, antibodies to these microbial proteins in the blood before vaccination can predict whether or not you will have a good response to vaccination. So in a sense, it's a measurement of immune readiness. How ready is your body to respond to vaccine? We did this by comparing the top 25% responders to the bottom 25% responders and looking to see which antibodies correlated with that. And what you can see here in red on this, the boxes colored in red are ones that correlated strongly with vaccine response, and anything with an asterisk was statistically significant. So quite a few of these were both predictive and statistically significant. And we're calling these antibodies sentinel antibodies because they effectively predict an individual's ability to respond to vaccine. Next slide. This just gives you one example of one of those sentinel antibodies. This happens to be the ISDB protein from Staph aureus. And you can see if you look in both the vaccine of the healthy or in the booster of the healthy, that there's a strong difference in the antibody response to this protein, depending on whether or not you had a good vaccine response or not. And that was true both in the healthy population and in the HIV population. Okay, so we'll go to the next slide. So we thought maybe using these kinds of data, we could use AI as a tool to develop a model that would help us predict who's going to do well if they get vaccinated. Keep in mind that this study had over 2 million data points. We separated those data into a training set and a testing set. We didn't touch the testing set until after we developed the model and the training set. We did the model, each model with 5-fold cross-validation. And then after doing that, we repeated making the model 100 times with random seeds. So we were really confident that at the end we had a pretty good assessment of how well these models performed. In the end, it was a multi-layer perceptron model that gave us the best fit, although many other models did well. The MLP model did the best in terms of working with all the different clinical cohorts that we studied. Next slide. So this is just showing you that the universally trained model did well in multiple cohorts. You can see it predicted well for the healthy population, for the HIV population, certainly the solid organ malignancy population, and so on. Overall, these AOCs were better than 70%, and that's in the testing group. And so we feel like this is a pretty good model for predicting immune response. Keep in mind that diseases like autoimmune disease and solid organ transplant did not do so well, but those two diseases in particular are ones that are treated constantly with immunosuppressive medication as part of their regimen, so it's possible that that's playing a role. Next slide. So this is just to summarize, we've done a comprehensive immunoprofiling at what we feel is a massive scale. We've identified immunocohorts and their ability to respond to vaccines. Some of them are significantly suppressed, but that response was heterogeneous. Not everybody in an immunosuppressed cohort has a weak response, and some people in the healthy population also have a weak response. Pre-existing sentinel antibodies, however, seem to predict who's going to respond well, and we believe that the future will be to develop AI models that will help predict those responses. And the last slide is just to thank the people that were here. You can just click it multiple times. A lot of these folks are the scientists that in particular played a role. Yeah, Jicho, Velmurugan, Lusheng Song, and Jay Park in particular were ones who really helped develop this program. And with that, I will stop and thank you.
Thank you, Dr. Leber. It was really a very inspiring and thought-provoking lecture. May I now invite Professor S.N. Dwivedi sir to present a memento to Dr. Leber.
Medical Innovations, University of California, San Francisco. Dr. Harish Vasudevan Sir, Assistant Professor at UCSF and a renowned physician scientist in neuro-oncology and radiation oncology. His research focuses on growth factor signaling, neurofibromatosis and nervous system tumors, while his clinical work is dedicated to the treatment of pediatric and adult brain cancers. Today, he shall share his expertise at the forefront of cancer biology and precision medicine. Please join me in warmly welcoming Dr. Harish Vasudevan Sir.
Perfect. All right. Thank you everyone for organizing, for the invitation. We are very excited to be here. Beautiful campus, lots of great young minds. The introduction, I think, summarized short and sweet, which I loved, so I will just jump into the project. The theme today that I've been asked to speak about is multi-omics. I think you've heard a lot in the last talk from Josh, Dr. LeBert, some of the beautiful work they've done. I can share a little bit of the translational perspective. I think multi-omics is not only coming, it's already here. And I've spoken to so many of the pathologists and other physicians here who have told me it's coming here too. So what I would like to share today is a little bit of our work leveraging multi-omics specifically for clinical translation, new diagnostic tests and new therapies. Almost all of the research in my lab is focused on developing better tools to take care of our patients. What I'm showing here is a comic from The New Yorker. It's a famous magazine in New York City that I read every day for 10 years when I was in training, like many of you. And this comic really stood out to me. Multi-omics just provides data. It doesn't give you information, it doesn't solve the problem. And so the quote here is one question: if this is the information age, how come nobody knows anything? I would really encourage all of you to think of multi-omics as one more tool in your toolbox. The same way you have a history, the same way you have a physical exam, the same way you have pathology, the same way you have imaging. The fundamentals of medicine don't change. This is just one more test for you to then take as a clinician and integrate into your multidisciplinary practice. Okay, so I think all of my fonts changed, so this will be fun. It'll be hard to read. I wanted to share a little bit about who we are and what we do. My lab really takes a bedside to bench approach. What does that mean? Bedside, I see patients now 1 day a week, full day, 14 hours, get the full experience. And really most of our questions are motivated by problems that our patients face. So we take those questions from the bedside and we go to the laboratory bench, where now we can do scientific research to develop better options for our patients. And most of the way that we do our research is we start with patient samples. We live in a really amazing time where we can measure so much from human patient specimens. So we do a lot of molecular biomarker work. We do multi-omics, single-cell omics, bulk omics. These are all just fancy terms of other ways to measure biology. Again, think of it like a history and physical. Think of it like a pathology test. Think of it like MRI or CT scan. One more data point. Then we take that from the bedside and we go to the bench. I've been very lucky to have been trained as a geneticist, so I think a lot in terms of how genes are important. They're not everything, the environment matters as well. But we do a lot of work in mice. I'll show some of that today. Many of you may have heard of CRISPR technology. It's a gene editing technology. Nobel Prize was awarded for it. It has really changed what we can do. And the last thing that's a little bit more old school is biochemistry. So this is proteomics, thinking about how the gene becomes DNA, RNA, protein. For me, proteins are ultimately the main players in biology, so how can we use that knowledge? My lab is in San Francisco, as you heard. It's usually very foggy, so we work in that fog. Often we cannot see our hands in front of our face, so our lab is somewhere under that tower. And I get to work with a lot of amazing people. They're pictured here at our lab retreat. One thing I really enjoy about our group is that we have both scientists, clinicians, and clinician scientists. So we have a nice mix of people to bring different perspectives. So I want to start with some background, and I'll kind of dive right into the clinical part that motivates our question. What I'm showing here is a graph of the most common cancer predisposition syndromes that we are aware of. What is a cancer predisposition syndrome? This is a class or a family of diseases where people are born with a genetic mutation that puts them at a significantly increased risk of cancer. And what I'm showing here is this graph that I actually put together for a class that I teach for our medical students. So this is, I hope, very right level, pertinent. The x-axis here is the prevalence in the general, no pointer. The x-axis here is the prevalence in the population. So how common do we see the mutation? The y-axis is the lifetime risk of tumors. And so you can imagine that there's two parts to this question. One is how many people have the mutation, which we call the prevalence. And the second is how many people with the mutation actually develop cancer. We call that the penetrance. And so what you can see here is the two things in the very top right. So the most common things that have the most cancer risk are NF1 and NF2. We call these as a family neurofibromatosis. People that work with brain tumors are probably very familiar with these. These are the most common neurogenetic cancer syndrome. They occur up to 1 in 1,286 live births. So common. This is just to say this is how we diagnose NF1, and so this is a clinical diagnostic criteria, but I'll just say even in our clinical guidelines, we updated them just last year as part of a large group, and essentially we added this point that you can actually do sequencing of the germline, and if you see a mutation at 50% frequency, that is sufficient to make the diagnosis. So this is new, but I'm just saying even in clinical practice, the guidelines all of us need to memorize and learn. Actually, I'll be honest, I don't memorize it. I print it and put it in my office, so it's there every day for me to remember. It's part of it. And so how does that clinical context translate to the lab? And so this is a slide I like because it gives one way to think about it. I always tell everyone in the lab the facts often change as we learn more advanced science, but the way to think is the one thing that never goes out of style. And so this is how we think about the problem. As I mentioned, people with NF1 on the far left in black, they have one mutation they're born with. And what happens when you have that mutation? These people actually have a lot of phenotypes or physical manifestations that are not related to cancer. Things like cognitive problems, things like cardiovascular problems. And so we think a lot of those phenotypes have to do with not having one copy. One of my mentors used to always say, when there's chaos, there's an opportunity. And so this is the opportunity here. We can try to detect early in these patients. We and others are working on gene therapy and other ways to restore NF1 activity. This is something that we've done quite a bit of work on. And you may have seen in the news with CRISPR for many non-cancer syndromes. We actually have gene therapies now. Hemophilia is a great example. And in cancer prevention, I think cancer prevention is really the future, especially as population grows worldwide, in India, in the United States. It'll be very hard to treat everyone with malignant tumors, so our goal should be to decrease the risk as much as possible of malignant tumors. When you lose that second copy of NF1, what happens is you actually start to develop what we call benign tumors. It's a little bit difficult sometimes with new students to describe what is a benign tumor. The way I describe it is a tumor that grows locally, but does not spread or metastasize, and typically does not cause death. It can definitely be a problem if it pushes on a nerve. This is something we see a lot in the clinic. If it pushes on a blood vessel, these can be problems. The common benign tumors in NF are nerve sheath neurofibromas. They arise from the coverings of your nerves, and low-grade gliomas in the brain, which we take care of in the clinic. And so we've done, this is more just for people who are interested to read more. We've done some work trying to understand this process. I won't talk today, but we have an idea to target RAS signaling in NF1. We have some ideas about how when you lose the second copy of NF1, are there therapeutic opportunities? One famous example people may be familiar with is the BRCA genes. PARP inhibitors, discovered by the director of our cancer center, are synthetic lethality. This idea that when you lose NF1, there may be specific things you can treat with. And then, of course, biomarkers, which I think need no introduction. And then kind of the way, and maybe a point for some of the students and trainees in the audience, is the career trajectory can be very funny and unpredictable. I actually started looking at malignant tumors, and over time, the last 5 years, I've realized I really want to move toward detection. So you can see all the papers we've published are in the malignant tumors, and I just told you that's where we should spend less of our time. But it takes time to learn everything, and we're always all learning. These malignant tumors are malignant peripheral nerve sheath tumors and glioblastoma, the most common malignant brain tumor. Median survival is still 18 months, so a big problem. So we've looked at tumor heterogeneity, the idea that these tumors are not all the same, all the cells within one tumor are not the same. We've looked at combination therapies. We believe that one treatment alone is never enough for malignant tumors. You have to combine surgery, I'll tell you about a radiation story, and multiple therapies. And then we really think we're in this era where we get to build next generation therapies. Cell therapy is a great example, which I know people here are working on in terms of building new cell therapies. And then one last bit of background, I'm still a biologist, so I like to think about basic biologic principles. One of the challenges I give to everyone in the lab, that I'll give to all the trainees in the room, is really think about how you can do work that makes it to a textbook. You should be doing work where if it's successful, you will learn something so important that you can't write the textbook without including that information. And so this is kind of the textbook of NF1. NF1 is what we call a tumor suppressor gene. What that means is when you lose the gene, you get cancer. The way NF1 works is it blocks another protein called RAS. So when you lose NF1, you get too much RAS, and too much RAS activates many other things. If you Google or ChatGPT or Gemini or your favorite AI strategy, RAS, you will see whole textbooks. So this is a very simple version of what RAS activation does. Whenever you have a break with NF1, you also have a gas pedal in your car, and so the way the gas is added to RAS are these receptor tyrosine kinases and these GAFs. Why am I telling you about these? Half of human cancer has a mutation in one of these proteins. So these are very common. So for people that have a clinical part of their work, 1 out of every 2 cancer patients you see has a mutation in one of these genes. So there's a very clear clinical opportunity. And so it's not surprising that when you have this many mutations, we have built a lot of drugs over the last 25 years that target this pathway. Tyrosine kinase inhibitors, RAS inhibitors, just very recently for pancreatic cancer. This is now in the US FDA approved, and I think it'll change cancer care worldwide. MEK inhibitors. It's a very important pathway and a great example of how omics, before we had multi-omics, transformed clinical care. And then the last thing I'll say is I pitched this originally as a problem of kids and predisposition, but I believe in our labs, one of our main hypotheses is cancer and development are the same thing. The way we grow from one cell to baby to adult and the way that process is corrupted to cause cancer are the same thing. And so this gene NF1 is actually mutated in 16% of all cancer patients. So I asked my friend Claude on the way over to India to tell me how many new patients with NF1 mutant cancers would be diagnosed each year. In the whole world, 3.2 million. Just in India, 230,000 people each year will get diagnosed with NF1 mutant cancer. So I often say this is the most important cancer gene no one ever studies, because I think this is absolutely a big problem. And this is just a graph of the different kinds of tumors I told you about. Nerve sheath tumor, glioma, melanoma. Okay, and then the last slide before I show some data is trying to provide a framework for what we think are the big questions in cancer biology as we enter this multi-omic era. I don't think AI is solving all our problems. In many ways, it's creating more problems than it's solving. There's still a lot of important questions to answer. The first question we think a lot about, and I'm sure everyone in this room that has any clinical training has seen, is the gene mutation does not predict alone what happens in the clinic. Again, really this point that multi-omics is another tool. It is not your destiny. Multi-omics can complement history, physical, pathology, imaging. We want to understand what that process is. So that's number one. Number two is how do we target tumor suppressor loss? This is one of those questions, every time I read it, I think more and more about what exactly it means. It's in some ways easy when you have a mutation in a protein that turns it on, because then you can just target that mutated protein. If you think about it, when a tumor suppressor is gone, that protein is not there. How do you target something that's not there? That's a fundamental question that we have not answered well. And working with people like Sanjeeva and Josh and Joachim, we've really tried to think about how we can use proteomics to study that question. And then last but not least, this idea of multi-scale or multi-omic data. How do we predict which people will actually develop malignant tumors? I think this is really important because it would change clinical practice. Another thing I often tell the translational trainees in my lab is if you're going to have a translational project, if the project is successful, it should change clinical practice. It's not enough just to describe something. You need to actually go all the way and see how it changes clinical practice. And I'll hopefully show you a couple of examples today. So I want to tell two stories. The first story is using something called spatial genomics to understand tumor evolution. This is a cartoon ChatGPT helped me make that describes many of the scales at which we talk about tumor heterogeneity. In the smallest scale, we have the multi-omics, genomic, epigenomic, transcriptomic, proteomic. That's one scale. The next scale up, we have cellular heterogeneity. For cell therapies, this is absolutely critical. We now have many examples where we've developed cell therapies against a target, and either the cells don't get there, or once they get there, they get exhausted, they get tired, they can't kill the cells anymore. So this cellular heterogeneity matters. I'm going to tell you a story here in a moment about spatial heterogeneity. I feel like I've spoken to only pathologists, which is great. So for all the pathologists, this is you've known this for 250 years. When you look at a tissue under the section, every part looks different, and part of the training is finding where to look, what to look for. As a radiation oncologist, I look at a lot of imaging, and so for anyone that's a radiologist, surgeon, radiation oncologist, really all clinical doctors, radiographic heterogeneity, we know the tumor doesn't look the same in every part of the lesion. I'm not going to talk about that work, but we've done quite a bit of work understanding how the different areas are different. And then, of course, the most important thing, clinical heterogeneity. How do we go from that smallest scale to actually change patient outcomes? Okay, one thing that I should say is we all think of all cancers as undergoing a stereotypic evolution from normal cell to benign tumor to malignant tumor. For neurofibromas, this usually happens with NF1 loss. You can also have NF2 loss, which causes a similar type of tumor called a schwannoma. And if you lose both, you actually end up with these malignant peripheral nerve sheath tumors. How this happens is critically important to develop better tests and treatments. If we can identify which people with benign tumors will later get malignant tumors, that changes the game. Things like colorectal cancer, breast cancer, lung cancer, we now have ways to screen and do this. But for brain tumors, unfortunately, we don't have that. So a big research goal is to build that. This is just one I always have to show some MRIs as a radiation oncologist, so I thought it was worth putting it in the talk. Many of you that have clinical training, these should look very familiar. These are schwannomas on the left in green, the bilateral, ice cream cone shaped, homogeneously contrast enhancing. No multi-omics needed, likely NF2. The middle is a neurofibroma. You can see that it's well encapsulated. It's what we call plexiform, so it has many different parts that are cupped together. And that same patient, 2 years later, had an MPNST, and we could not predict it. This is one of the first patients I saw in training many years ago. And so this case stuck with me over time. The other thing I'll say is we know that not all benign tumors are created equal. If you have a schwannoma in green, you never develop a malignant tumor. If you have a neurofibroma, it takes you about 20 years. But if you have an atypical neurofibroma, it only takes 4 years. So we've already started to build these types of stratifiers that I hope make it to the clinic. If you have a patient with one of these tumor types, hopefully you'll be able to give them some sense, 5 years, 20 years, never, of when this will become a malignant problem. So what did we do? One of the new technologies that I've been really excited about is something called spatial transcriptomics. The idea behind this technology is you can take a H&E section and you can do sequencing of every part of that lesion and then read out how the molecular information connects to the histopathology. So again, all the pathologists in the room, you've known for many years, not every part looks the same. So my pathology colleagues, this was led by Dr. Kanish Mirsha, who was a fellow in my lab, now is faculty at UCSF. He wanted to take these neurofibromas and see if what he saw with his eyes as a pathologist correlated to the molecular program. So we took 15 of these tumors, we looked at all the clinical data. Our neuropathologists, I feel, are world class, so they looked at all the histology, we did an immunohistochemical analysis, DNA sequencing, DNA methylation, the multi-omics as we call it, and then did spatial transcriptomics. This is the entire cohort. I'm not showing this to go through all the details, but just to say again for the trainees, it really takes a lot of work. This was a 5-year project, and there are many setbacks along the way. I always tell people in the lab about 20% of what you do should work. If more than 20% is working, we're not trying to go big enough. The cases I want to focus on are these 2 cases that I've outlined in red. They have what we call a transition zone. For the pathologists in the room, the way I would describe this is you can tell if it's a benign tumor, you can tell if it's a malignant tumor. This is everything else. So this is what we think is the evolution happening in real time. And so we looked at these transition zone samples and we wanted to ask how is the molecular profile evolving over time? So every time I show this, and I know there's pathologists, I feel bad I don't show the zoomed in versions, so you'll just have to believe me, that we looked at these sections very carefully. The top right, as you can see in this teal mustard color, is low grade. The bottom is high grade. The areas with N are just necrotic areas. You'll see that those have their own signal. And then that middle dashed area is, I think, where the really cool biology is happening. This is the transition zone. So what we did was we took this section and we did spatial transcriptomics. So then every single spot I'm showing in that spatial cluster slide, we have the RNA sequencing data. So we can see all the genes that are on or off. And if we take that analysis and ask, how do these spots look similar to each other, we can develop what we call clusters of spots. So these are spots that are similar. And the really cool thing is we develop those clusters, we're blind to where the clusters are on the section, but then if we project them back onto the section, they're all together. Again, my pathologists are never surprised by this. They say, obviously, the spots that are close to each other look the same. But as a physician scientist, I thought this was a really cool result. Okay, so now that we've kind of convinced ourselves that these spots do look similar, how do we integrate this on a multi-omic level? The very first thing we did was we used those genes that are on or off to infer or build programs of cell cycles. So we know what genes go on in each phase of the cell cycle. All cells cycle from being non-dividing to dividing. Cancer cells, as I think many of you know, are more likely to be dividing in G2M or S phase. So we can actually predict those spots. And what you can see here is in black, the G1 phase are the non-dividing cells. In the transition zone in red and blue, you start to get cell division, and that really takes off in the malignant region. So this is just saying something that everyone that's a pathologist has known for 100 years. Tumors that are malignant cycle more quickly. You can also play the same game with cell differentiation, and so you can see loss of cell differentiation as you become malignant. So we really think there's two things happening. Cells are growing more quickly, they're also de-differentiating. So a transcriptomic cell cycle signature, and then an epigenomic, another multi-omic component, differentiation signature. What can we do next? Cell therapy is big, but to do cell therapy, you have to know what cell types are there. So you can actually use this data to predict the different cell types. And we saw something cool, which are in that transition zone in the middle. If you look at that second plot with the CD68 positive macrophages, this macrophage population only goes up in the transition zone. So what this means is that as you go from a benign to a malignant tumor, there is a short-term or transient increase in immune cells coming in. Those are shut off in the malignant tumors. This might be what some of you are familiar with, a immune cold tumor, right? This is what's happening. What is neat is we can see this is not static, this is dynamic over evolutionary time. And then of course I can't forget my favorite part of the multi-omic, the genetics or the genomics. What we can also do with this data is predict which chromosomes are deleted. Many of you are probably familiar with certain chromosome deletions in certain types of cancers and diagnoses. Many of you are also familiar that not every part of the tumor has all of those mutations. What we can see here is that the top right of the part of the tumor has 9p chromosome intact in gray. This is lost in the bottom part of the tumor. And in that cluster 2 in green, you see it's subclonal. What do I mean by that? Not every spot is lost. So again, I think that's where the opportunity is. The opportunity is to identify patients with those early events and then intervene early. Now you might say this all looks great. This was about a $250,000 US project, so certainly not something we can do for every patient. How do we make this something everybody can use? Well, we have an idea. It turns out what you can actually do is use this data. You heard a lot about foundation models and a few different perceptron networks from Dr. LeBaire. I won't get into those details other than to say you can take the H&E slide. There are many convolutional neural network approaches to actually predict outcomes. We can take those 51,000 ROIs from our 15 samples, build a deep learning model to predict what chromosomes and what transcriptional programs we have. Similar to what you heard earlier, 70% training, 30% test that we kept out. This is just some ROCs to show that it indeed works reasonably well. And this is those ROCs projected, which I think is really, really valuable. This is that same case. You'll remember the top right is benign, the bottom left is malignant. The left column here are the observed data. The right column is what our AI algorithm predicts. And you can see it looks pretty good. So I'm cautiously optimistic this is something that will validate in larger cohorts, and that's what we're doing right now. So to summarize this first 15 minutes, if there's nothing else you remember, please remember that these genes are common in cancer. They're important. It's important for your patients to be tested for them, especially if it's a young person. We typically say under 40 years old with a brain tumor. Multi-omics encompasses genetic, epigenetic, transcriptomic, cellular. You'll hear about proteomic, and that's really important. And we think this deep learning approach might be one way to potentially democratize, to make widely available some of this molecular classification. This has already been done for other brain tumors. So if you look for primary brain tumors in the brain, There are now 3 or 4 approaches where you take the H&E section, you run it through the algorithm, and it'll predict what the molecular classification is. There's one famous one from the German group. If you look it up, the Heidelberg classifier. We have one from the United States now. We call it the NCI classifier, National Cancer Institute. So if you have a patient with an H&E, you can actually anonymize the data and upload it, and it'll tell you what the methylation class is. So it's very powerful. All right, last 5 minutes. I just want to give one example of something that's a little more cellular in origin, given the theme of cell therapy, which is using single-cell and functional genomics to improve radiation, which is my day job. So one of the things we learned a few years ago was that MPNSTs are bad. These malignant peripheral nerve sheath tumors, median survival of 5 years, as you can see on this Kaplan-Meier curve. Unsurprising again to my pathology colleagues, cells with high Ki67, high proliferation, do worse in red than ones that are black. This was maybe the most obvious finding in my entire scientific training. What was unexpected was if we looked back at the immunohistochemistry, this was in the pre-AI, pre-single cell era. We just had IHC, this is an old paper now. You get 2 clusters, and only that cluster 2 in red do the patients respond to radiation. You can look at this paper if you want the details. It's now 6.5 years old, so I won't talk about it today. But we wanted to ask, what is the mechanism? I tell everyone in the lab, if you don't have mechanism, you just have phenomenology. You just have an observation, you have a belief. You have to have a mechanism of how this works. So I had a really talented MD PhD student in the lab, Dr. Iowa Zhu. He's now over at Harvard doing radiation oncology residency. And he did something called a genome-wide CRISPR screen, so a new technology I'm introducing. This is another multi-omic technology. The idea here is you can take your system, you can knock out each gene individually in one big experiment, and then see how that affects response to therapy. So this is a way to discover on the whole genome scale what modifies response. Very powerful tool. One of the many ways CRISPR has revolutionized how we do it. When I was a PhD student, this technology didn't exist, and I tried to do it with old technologies. It never worked for 5 years. So I think I always did it in a year and a half. So he's very good, but also the technology got better. So what we did was a simple experiment where we took patient-derived tumor cells, we treated them with radiation, or left them untreated, and we saw which genes mediated the response to radiation. The idea being this might give us a way to improve radiation response. This work actually came out earlier this year. I didn't know it would be published by the time I spoke, but you can actually look it up. It's now online. When we do this kind of analysis, we do many, many different kinds of analysis. This is one place AI has really made a difference. We've now automated our analysis pipeline, so we give it the raw data, and it does a bunch of quality control and spits out an initial set of plots. Anything in red on this plot, if you knock it out genetically, you improve resistance to radiation. So these are things that you might have to block to improve radiation response. Anything in blue sensitizes. Many of you know that radiation works in large part by damaging the DNA. So it's not surprising that DNA repair genes, ATM, RAD51, are sensitizing. Another well-known effect of radiation is oxidative stress by causing oxygen-free radicals. So again, another what I call positive control. Very important in science. How do you know your experiment worked? The new thing we found is that interferon signaling actually turned out to be critical. Many of you may be familiar with interferon signaling in the immune system. These are secreted chemokines that bind interferon receptors and allow the immune system to work both in normal physiology in response to infection. I spoke to a couple people working on infectious disease as a key pathway, but also in cancer. We all know cancer immunotherapy is here to stay. So we had that result. That's just an observation. What happens if we actually do it in an animal model? And so we took the set of mouse tumors, we implanted them in mice, and then we radiated them in an immunocompetent host. What we saw is radiation works initially, but then the tumors do grow back, so we were curious, how does this happen? So we did something called single-cell RNA sequencing, yet another multi-omic technology. I'm just throwing out these terms so that those that are interested can go read about them, learn about them. There is a lot to learn and a lot of ways to use them. What this lets us do is measure all the cells in the tumor all at once and all the genes in each individual cell. And then we'll often draw something called a UMAP. So each dot on this graph is one cell from the tumor. And the dots are organized on the graph based on how similar their gene expression is. So dots that are close have similar gene expression. Dots that are far have very different gene expression. And we typically conclude from this what are the cell types, because we think gene expression is different between cell types. So we name each cluster. This is a whole other AI-aided process nowadays. We used to do it manually. Now AI can help us find consensus gene sets. And what we can see is we find both cancer cells and non-cancer cells. The interesting thing is what we saw is that radiation actually promotes T cells getting into the tumor. And so if anyone that thinks about cell therapy, you're familiar that CAR T cells are, they're already here, current big thing. So we thought this was a really nice observation to start building cell therapies. And this is one of my scientific heroes, a famous physicist, Richard Feynman, who used to teach where I went to college. And one of his famous quotes is, I feel like when you're in training, you're always trying to be right, you're always trying to get 100% in every test. But actually, as I do this longer and longer, it's much more fun when I'm wrong. Because that means that there's an opportunity to learn something new. So this is one of my favorite quotes that I try to keep with all of our students. So getting to the end here, whenever we do science, we want to show the same thing multiple ways to make sure that it isn't just something that's technical. So we also do something called flow cytometry, radiation-treated tumors on the right, non-radiated tumors on the left. You can see that radiation increases these T cells, both CD4 positive and CD3 positive. We can do gene expression analysis, and I'll just say that essentially when you radiate the T cells, not only are there more T cells, they're also more active. I talked about how the T cells need to be active in the tumor. So we think this is a great strategy to maybe prime the tumor for cellular therapies. So anyone that does radiation, hopefully you're taking notes because I think this is going to be the future. And so we then asked, is this really true? Is this required? And so this is probably my favorite experiment in the whole paper. We initially did these experiments in healthy, immunocompetent mice. We did the same experiment in mice that were athymic, they're nude mice. What does athymic mean? They don't have a thymus. As many of you know, the thymus is where T cells are born. So these mice don't have T cells. So the hypothesis would be if T cells were really required, radiation should not work in this model. And that's exactly what we see. The pink and the gray you can see are just right on top of each other in both our models. So T cells are required for radiation response in this system. This is really my favorite experiment. Sorry, I went too soon. So this experiment, Ios is a very thoughtful, methodical, mechanistic physician scientist. He's going to be great. He wanted to ask, well, we know interferon signaling is really important in the immune system, but I think it's actually important to the tumor cells as well. So he took those MPNST cells, used CRISPR to knock out the interferon alpha receptor 1, IFNAR1. This is the key receptor that mediates, we think, all interferon signaling in the tumor. And he asked, does radiation still work? So this is 4 conditions. The top 2, control cells with radiation in pink, control cells without radiation in gray. You see there's a nice difference. The pink cells or tumors are smaller. But if you knock out that interferon alpha receptor 1, radiation, again, doesn't have an effect. So not only do you need T cells in the microenvironment from the thymus, you also need the tumor cells. So it's really a 2-part system. I think people forget this with cell therapy sometimes. It's not just the cell therapy, it's not just the antigen, it's also the tumor. And this is where multi-omics helps, right? You cannot do this with multi-omics. I don't think I could have done this when I was a student because these technologies didn't exist. Last but not least, always coming back to the patient. We took our cohort of malignant peripheral nerve sheath tumor patients and we scored how many T cells were in each tumor using another multi-omic technology, methylation arrays, which I won't get into. All I'll say is this Kaplan-Meier curve, x-axis is time, y-axis is progression-free survival after radiation. Black are the patients who had CD8 T cells. You can see radiation works quite reasonably well, 70%. The gray is no CD8 T cells. So even in patients, these T cells are required. The other thing we noticed is that I told you about that chromosome 9p loss in the initial story. It turns out that's also predictive. So in red, if you lose 9p, you have less of an effect than in blue. And I think this is just really cool because often we have these observations, but we can't tie them back to genetic mechanism. It turns out chromosome 9p, that area of your genome, has this cluster of genes that I'm boxing here in red. This is the interferon gene cluster. So we actually think the mechanism here is chromosome 9p deletion deletes the interferons from the tumor. Interferons then cannot be activated because there's no interferons. And that's what's leading to this different radiation response. So we kind of come full circle from cell therapy to genetics to multi-omics. And so this is the last slide. In an MPNST model, we think radiation requires interferon signaling. This is kind of the most simple cartoon form that I hope my 8-year-old daughter can understand. That's always my goal for the summary figure. So we have tumor cells, we have macrophages. Tumor cells and macrophages both make interferons. You need interferon signaling in both compartments that increases T cell recruitment and activation. And then it's interesting because there's been a long history in radiation of using interferon therapy to improve the radiation response. One concept I didn't get to touch on today is therapeutic window, which is the idea that your treatment has to work better on the disease cells compared to the normal cells. If it hits too many normal cells, people can't tolerate it. That's why interferons didn't work. Fortunately, in 2026, we have multiple new ways, and the trial we're running now is using something called TVAC, an oncolytic virus that we inject into the tumor, drives an immune response, and hopefully gets better outcomes. I'll skip this last thing just to say we use foundation models. And thank you everyone for your attention. Take questions maybe at the end. Thank you.
Thanks a lot for that excellent presentation. Sir, 2 minutes please. So it had all the components of omics and started with basics until all the excellent study you presented. May I invite the head and neck oncosurgeon of our institute, Dr. Ashok Shinoy sir, to present the memento to sir.
Great talk, Harish. Predicting chemosensitivity, radiation sensitivity, and trying to improve the local regional control and disease-free survival. Really, it is something. Yeah, we are doing it, but the point is we don't have the money to do the basic research, and hopefully we should be able to get lines open with your center because we have a lot of disease here. So, and we have our ethics committee, and we can certainly look at phase 1 trials, if not anything else. May I have the pleasure of acknowledging your wonderful presentation.
Our next distinguished speaker is Dr. Joakim Martin, Professor of Translational Proteomics at KTH Royal Institute of Technology in Sweden, and is the leading scientist at Science Life Lab. His pioneering research focuses on plasma proteomics, biomarker discovery, exploring how proteins circulate in human blood and can provide insight into health and disease. A global leader in proteomics, he has played key roles in the Human Protein Atlas and the Human Proteome Organization. Please join me in warmly welcoming Dr. Joakim Martin.
Okay, good morning everyone. It's great to see you here and your vivid interest in multi-omics research. As Harish said, we have some reformatting here, so bear with some non-stylish visualizations here. But yeah, I think I'm here to talk a little bit about the work we've been doing out of Stockholm, Sweden, and how we think the future of multi-omics can be accelerated by using microsampling. I also want to spend a few moments to talk a little bit about a project that I've been working with for the last 20 years. It's a project headed by Professor Mattias Ullén, and in Stockholm, Sweden. Since 2005, we have provided an open access website for the community called the Human Protein Atlas, where you can search and browse for protein expression across the body. We have a lot of tissues and immunohistochemistry images, subcellular localization of proteins, network analysis, single cell analysis. So really trying to deconvolute where in the human body and in which cells and which subcellular compartment proteins are being expressed. And more recently, we have also integrated how we can measure proteins in blood as potential indicators of health and disease. And all of this information is freely available for you and the community. So in case you have a favorite protein, like here I have one of the proteins I often talk about, GDF15. You can see which disease you have this protein being increased, You could also see how longitudinal analysis of this protein describes, for instance, the increase in the expression during puberty. But it also shows that you have some phenotypes, for instance, as pregnancy. I don't know if you can actually see that here. You have an increased level of this protein. So the interesting part of GDF15 is it's also a protein often talked about in relation to aging, but it's also a protein that is highly being affected by the use of medication. So I guess when we then think about what happens when you grow older, in most Western countries, then the number of medicines you are using is increasing. So, we have additional things that influence our molecular health, which is actually our life and the use of medication. I also want to come back briefly to the example of pregnancy, because GDF15 has been sort of linked to nausea. So some women, experience sickness during pregnancy, and actually this is related to this protein, which causes, depending on which variant of the protein you express, you feel sick. So again, we see biology, one protein has many different facets, and I think that's what we really are fascinated about, and that we want to understand a little bit more detail. But I think you also know, and I guess you as clinicians, taking a blood sample from a person requires many things to happen. And most importantly is that the patient shows up. And then, of course, you need to have equipment, you need to have experience of taking a blood sample. So what you can see here is my arm a couple of years ago when I got a regular healthcare test. And I had to book a time to come to the hospital, and I had to schedule my meetings to different times of the week just to be able to be making it to the hospital to take a blood sample. And I mean, the purpose was to understand, some of my health measurements, and to see, whether proteins that are in the circulation can report something about my cardiovascular or diabetes risk. And when we study proteins in the circulation, we can of course look at where they're coming from, because that will ultimately tell us what the disease is that we are facing here. So, I mean, we can access molecular health in regular blood samples, but we have also realized during the pandemic that that's not the most efficient and meaningful way to do and to use modern technology in the most effective manner. So as Chair nicely introduced, I'm also been chairing a project called the Human Plasma Protein Project with many people around the world, where we looked from a technology perspective into what is the community doing and how are they analyzing blood plasma primarily, in terms of which technologies that they are developing, which protocols have been optimized and presented. And every 2 years we write a review for presenting the status of the technology in the community. So if you're interested in the subject, these are very good starting packages for you to learn a little bit more about that work. So the circulating proteome, as I like to call it, is really a treasure trove to understand human health and disease. And I think with technology, come back to that in a slide, enabling us to go deep, we can also use this information that we have generated in relation to other types of data. So there we can touch upon the multi-omics opportunities here, and it's integration in a sense that you now can couple proteomic information with genetics to understand what is the regulatory aspect of your genetic predisposition or variant in relation to your molecular health, because some of these technologies can actually read your single point mutations and they may have an effect on the readout. The second part is standardization. Now we have multiple technologies that allow us to measure many thousands of proteins, but as you can see in the image in the center, these are isolated islands because each technology produces its own list of proteins, its own sort of characteristics. So it's very hard to actually connect these modern technologies so they become meaningful together. Even though people are talking that they are complementary, they are independent data sets. And the third step is then, of course, why are we doing all of this? Well, ideally, we want to translate proteomics into the clinic. And I think we have realized that our hopes, wishes, promises, and dreams are not aligned with the reality. And in this recent review, we have sort of touched upon some of the bottlenecks that we see in in going from discovery into the clinic. And I think one of the key messages here is that the way we do discovery is really research-centric. But what we want to do is clinical implementation, and that's probably as furthest away as you can think of doing clinical research, right? So the discovery approaches are not aligned with clinical reality. So you have to implement several steps to get from the left to the right, and often these steps cause that this initial excitement and idea breaks apart because of regulatory aspects, because of alignment of protocols and many things. So it's a quite complicated path, right? But we still think that there are possibilities at what we call the translational inflection point to bring proteomics closer to the clinic. So I mentioned there are several ways of measuring proteins in blood. So the most common one in our proteomics community is mass spectrometry, where you chop apart a protein using an enzyme, and then you have these mass spectrometry measurements to look at the mass to charge ratio, and then you use a database to identify which protein this peptide actually belongs to. Fantastic technologies, but almost impossible to bring into a clinical activity because it's so fine-tuned, it's so sensitive to contamination and other things that people have tried and invested a lot of money, but it's very, very hard. Then there are other methodologies that are either using an antibody or an aptamer. So an aptamer is a DNA molecule that does the same thing as an antibody. And then you can measure thousands of protein in just one single drop of blood. But the interesting thing here is that because these are initially based on protein binding or aptamer binding, later on what you actually use as a readout is sequencing. So next generation sequencing technology that have been, of course, now has been spread around the world, and now maybe you will see even in India and in other places around the world where there are sequencing centers that you can actually do proteomics, because it's just a different way of preparing your sample. But at the end of the day, what we're actually doing here, measuring proteins in the circulation using either of these technologies, is something I call molecular phenotypes. So because we're measuring different proteins, because we're using different technologies, because we're using maybe different sample types, we might measure and reveal different lists of proteins that describe your health. So your urine proteome may very much be different from your saliva proteome, even though it's the same person, and even though you might be using the same technology. But if you measure your blood proteome with these different technologies, it's the same case, because not always you have proteins that are overlapping between these. What we want to do is, of course, for instance, we want to measure early disease onset, for instance here in the pancreas, and we want to, of course, be sure that the protein we're looking at is coming from the pancreas and not from other parts of the organ, because then we can actually intervene, for instance, try a different medication or change of lifestyle, and then again we do the measurement again and see has something changed because of the intervention that we have applied. And that's, of course, something we want to track more closely, and we want to see exactly that it's affecting the organ of interest. But what we actually are measuring today is more of a systems biology view of human health, because the circulating proteome contains proteins from all organs of the body, and each of these organs communicates with each other to sort of establish hemostasis. So what we are seeing actually is a list of different comorbidities or different flavors of molecular processes across the human body. And of course that would then likely help us to do more stratification, for instance patient classification. And I think that's where proteomics has been really, really powerful. But of course at the end of the day it's not a population of tens of thousands of people, it's a single person we want to have a measurement for to classify. So what I think is really exciting about the proteome, for instance in comparison to the genome, is that you subscribe to your proteome. While your genome, for instance this image looking at all these buildings, is constant throughout your life, the way you experience the architecture, your genome is changing as you age. This is an early morning here in Copenhagen a few years ago. So you're looking at the same image, your same genome, something changes, and that's of course the proteome. So we need to do measurements in the proteome continuously to learn more about our health. One way of course, understanding where do proteins come from in terms of disease is to compare the different diseases. And so this is a recent study we published in Science. Assumed as a viral infection. So of course one may also think about virus as the cause of cancer, and one can sort of start to spin certain ideas around these observations. But again, the fact is that we do these sampling and these individuals at a specific point in time. We also need to consider, as I showed you before, that the proteome is changing continuously. So if you would just slice this graph in a vertical manner, you would get a sort of different level for these different colored lines, indicating different sort of molecular phenotypes depending on what time of this investigation you're taking a blood sample and measuring the person's biomarkers. So here as an example, in terms of COVID or an infection, you have an early response that is very strong, like the red line, but then you have this purple line which might be completely unaffected by the infection. So still you might sort of have an information value in relating these proteins with each other. So it's really important when we take the sample, and not only that we take the sample. So here are some examples of why I think this is important. On the left you see something I call a confetti plot. These are 100 people that gave blood every third month for about 1.5 years. And based on 700 proteins we used here, you can see that each person has a unique fingerprint in relation to the population we are investigating, which of course is very nice to see, because that's what one expect from its genetic profile. But it also means that we are very different in a sense that the proteins present a different sort of profile of our health. And if we then think about standardization and sort of using one single biomarker for putting these people on a single trajectory, the heterogeneity that we observe here bears a huge issue. What you see in the center is basically the same data, but transposed in the longitudinal manner. So these spaghetti plots illustrate several proteins in relation to their longitudinal changes, and the colors basically are your genotypes. And as you can probably see, the height in which certain colors appear is depending on the protein, and it's depending on which genotype you have. So meaning, not knowing the genotype of a person, we might misinterpret the observations we are making, because the change of protein level or the observed level of protein might simply be dependent on the genotype. And the last slide, where it's sort of this radial plot or a spiderweb plot, is what you can see is a single person losing weight during the course of 1.5 years. And without talking to the person or measuring the person's body weight, you might also assume that there might be some health issues occurring, but actually this person is just getting healthier, right? So what we need to do is to do repeated measurements. We need to have more data, like genetic information, and most importantly, we need to talk to the people that we're investigating, so we can hear about their human health status from a personal perspective. So during the pandemic, as many of us, research has changed a bit. So what we have really started to do, instead of taking a regular blood sample as I showed you before, we've been using this micro-sampling cards. So it's simply a drop of blood that you take from your fingertip, you put it on these devices that I've shown you here, and the magic about these devices is that it measures exactly 10 microliters of blood for your analytical purposes. And why is this volume control so important? Well, it's important because if we want to compare many people with each other, we need to know how much material we actually put into our instruments that do all these amazing measurements. And the beauty about this system is also that we can simply send these sampling cards to the person's home. and they can do the sampling at home. So don't have to come to the clinic, schedule a meeting with the doctor or the phlebotomist, and they can do basically everything independently or in a timely manner. So we can, for instance, do a measurement in the morning, after lunch, or after they have started to take medication. So I think this really uncouples our possibilities in precision medicine to bring all of these activities closer to the patient or the donor, as we call it. So we've tested this idea now in COVID in multiple studies, but we've also tested it in mice to illustrate that we can actually sort of rewrite biology, or we can basically confirm the biological textbooks, if you wish. So here we used a mouse model that is sensitive to viral infection, and after a week of infection, these mice developed diabetes. So what we did, we sampled the mice every day through the course of about 2 weeks. And what we of course could see that the infected mice in yellow, they have responded quite strongly in the sense of their immune response to the viral infection. And after about a week, you get symptoms and molecular measurements that are related to diabetes, which of course is the proof that you can actually follow the molecular measurements to the observations that you do. Let's say in the case here with the mice, the criteria is losing weight. And then later on we do histology of the pancreas to really show that the beta cells have been destructed, which was also the case here. But as I mentioned, we prefer to do these things in humans. So we have now several studies published and under review, but the workflow is roughly the same. So we have this home sampling device that I just showed you. We have some sample preparation that we do in order to do different types of molecular measurements. And for instance, we can measure antibodies for infectious diseases like Josh presented. And then we can use this to do molecular classification, so simply using antibody levels as a way to categorize individuals. And then we can do all sorts of other types of measurements like affinity-based proteomics, here using antibodies. And here you see a picture of the 3 PhD students who've been working very hard and successfully. So when we have these new frontiers, meaning that we do these measurements in unknown samples with unknown or with high sensitive measurements, we of course want to be sure that what we measure is actually true. And the only way that we can currently do that is to take another technology to measure the same things. And what you can see here in the bar chart is that depending on how sensitive each method is, the reliability increases with sensitivity. So if you have a technology that gives you high sensitivity data and you use another technology that is equally high sensitive, you will probably get a very nice correlation between the data. But if one sensitivity is lower, then there's a disconnect. So what we actually did, we measured 450 people from the general population using these technologies, measuring 384 inflammatory proteins. And what we could see is that about a quarter of all the individuals in this population had some sort of deviating phenotype, which meant either they were smoking, either they were taking medication, or they had an immune-compromised molecular phenotype. So the good thing here is that we can actually extract this information that gives us some confidence about the proportion of heterogeneity. But it also was a surprising thing because we had no information about these individuals that were randomly chosen out of the population. So we didn't enrich for people that were healthy or sick. Simply this is the average population in the 2 major cities in Sweden, meaning that when you do some kind of population-based health studies, the expectation should be that about a quarter of those have some sort of molecular phenotype that separates them apart from the remaining population. So we've then also continued to do this longitudinally, and here we worked with a cohort that started about 30 years ago, and these 800 people here sampled themselves 3 times during the pandemic. And I think also, as Josh showed, very nicely we can measure all the immune response against the vaccine and the infections, but we can also measure autoantibodies that pre-existed before the pandemic, and we can see which individuals were actually affected by the pandemic, for instance, in their increase in autoantibody levels. We will also see that the environment, meaning the temperature and the pollution around where these individuals live, have an impact on their proteome. This is something that normally nobody ever takes into account. So what does it mean? So it means the outside temperature days before the sampling actually change your proteome. So in Sweden, of course, we have summer and winter that sometimes can differ with 50 degrees centigrade. So in the winter we might have -20, and in the summer we might have plus 30 degrees, which of course is an extreme span. But it could actually be that this temperature change has an impact on how people sort of present on their molecular level. There are a lot of things we can talk about this, because of course temperature in our climate also means behavior. So in the winter you wear warm clothes, in the summer you wear light clothes. In the winter you likely are more indoors, and in the summer you're likely more outside, exposed to the sun. You lie on the beach or you meet friends in the park. So it's a lot that one can sort of implement into such investigations. But again, it clearly shows that your proteome is changing. It's depending on your genetics, it's depending on the exposure, and it's depending on your lifestyle and behavior. Press the wrong button. So for us, of course, it's also important taking this new sample type. Can we reference it against other types of data? So what we did here is a genome-wide association study. So we looked at how does genetic variant in this individual influence the protein levels, and this is a very popular sort of topic in the field at the moment, and we have large studies that trying to understand how genes influence proteins. And nicely for us, it showed that the data is of such high quality that we can see the same things as people have done using clinical sampling. We also look at how the blood cell counts influence the protein measurements, because here we still have all the blood cells present in our analysis, and we could nicely see that the white blood cells influence many of the inflammatory markers, which is expected, but we could also see that there's a low proportion of red blood cells influencing our measurement. On your right-hand side, you see two proteins, and the most interesting one is actually the protein called LAB3. And on this dashed line that is vertical to the center, you can actually, we compared individuals before and after PCR testing. And what you can nicely see that there's really a slope on how this protein level decreases post-PCR confirmed infection, which means that there's a molecular phenotype which changes continuously depending on how far away you take the blood sample from the actual infection. So again, it really demonstrates the importance of when you take the blood sample will influence what you get out from these observations. So longitudinal measurements are really key, because then you can sort of build all these single snapshots and create a molecular movie out of your analysis. So we also think that we should open this up, and I mentioned that we're working as part of the Human Protein Atlas project, so we now want to make the dried blood proteome a little bit more accessible to the community. So hopefully later this year you can read more and browse more how can you actually measure human proteins in dried blood compared to regular blood samples. But I think the simple message I can leave you with, that actually taking these small samples and measuring whole blood will give you much more biology. So we're really, really excited about the possibilities, also because it is a way to cut down on costs. I think we usually say a regular blood sample taken in the clinic costs in our environment around $150, but using these devices it's a tenth. These devices are not cheap, but everything around it will be the aspect in which you actually gain in investing in these. So it's a 10-time reduction in cost, which I think is it's amazing. And we know all these measurements that I presented to you are very expensive still nowadays, unfortunately. And I think it's only when we manage to democratize multi-omics that it would actually become meaningful for us in the global community. So with that, I can also say that you can even go smaller. So from just 1 microliter of interstitial fluid, you can do similar types of molecular measurements. We're working hard to further improve this, because that again could open up many more applications, could even be more cheaper and faster way to do molecular measurement in a longitudinal fashion. And with this, I'm at the end of my presentation. I want to thank all of you for your time, interest, and if you have questions, please shout more. Thank you.
Thank you, Dr. Jochen. It was really extensive work, and I really thankful for you to share the work, especially on the development of the human proteome atlas, which is really extensive work. I now request Dr. Professor Mamta Dhaneria Madam to present a memento to Dr. Yokin. Thank you, ma'am. Thank you, Dr. Yukin. Knowledge advances not only through lectures, but also through conversation. So let's have a break for networking and tea, so that we'll be happy and we'll reassemble after 20 minutes. I suppose 12:10. So we reassemble at 12:10 after tea break.
R.D.GARDI sir, who is a professor and associate head research at the University of Manitoba, Canada, and director of the Canada-Italy Tissue Engineering Program. An internationally recognized leader in regenerative medicine, his research focuses on cardiac regeneration, stem cell therapy, tissue engineering, and innovative biomaterials. Through his pioneering works and global collaborations, Dr. R.D.GARDI has made significant contributions to cardiovascular regenerative medicine. Please join me in warmly welcoming Dr. Sanjeev R.D.GARDI sir.
To participate in this exciting event. Oh, this is amazing city. This morning I got opportunity to visit Mahakal temple, and amazing. I'm impressed. So, as mentioned, my research program focuses on cardiac stem cell therapy and tissue engineering for the last many years. And we use different types of stem cells and nanomaterials to repair the injured heart after a damage due to heart attack. And in that process, we end up using different omics techniques to understand the mechanisms and to come up with newer strategies. So nothing to declare other than these patents on a nanomaterial that we filed in the last 2 to 3 years. These are all approved now. Heart disease, I think I don't need to spend too much time on this slide, is now turning out to be one of the leading causes of death all over the world. But if you look at the scenario in the last 25 years or so, mortality after a heart attack has come down thanks to the currently available medical and surgical interventions which are available for patients. But all these interventions have been able to, they have been able to improve the quality of life and prolong lifespan of patients, but they have not been able to provide permanent solution to the problem. And the reason is cardiomyocytes, the cell type which is responsible for beating of heart muscle. These cells, unlike many other cells in the body, they don't multiply themselves. They are terminally differentiated. So any loss in terms of death of these cells after a heart attack leads to permanent damage to the heart muscle. So how can we fix the problem in a sort of permanent way? By replacing dead cells with new ones. That's where stem cell therapy comes into picture, because stem cells have the potential to turn into pretty much any cell in our body, including heart cells, and replace dead cells after a heart attack. And some of the stem cell types are also known to secrete several growth factors, anti-inflammatory factors, and angiogenic, pro-angiogenic factors. So that's how stem cells help in repair process after transplantation in the heart. When it comes to heart repair, different types of stem cells are being tested at different levels. Some of these cells are in preclinical stage, in animal models but some of the cell types have taken lead in terms of making it to clinical trials. But they are not there yet in terms of making it to clinical practice. And there have been several challenges reported in the field. And we'll talk about some of those challenges and mitigation strategies that we are developing in our research program. We'll mainly focus on these two cell types, bone marrow derived MSCs and iPSCs or induced pluripotent stem cells or iPSC derived cardiomyocytes, because these two cell types are in clinical trials. And again, there are challenges with these cells as well. So we'll talk about those challenges. Just a bit of background for those who are not in the field. These days there are different techniques available. You can reprogram pretty much any somatic cell in our body. What I'm trying to say is you can take any cell. I can take cells from a small piece of chunk of your skin. I can take cells from urine, even one hair I can take from anybody and isolate millions of cells, culture them, reprogram them to bring them back to stem cell stage. These are called induced pluripotent stem cells. So once we have induced pluripotent stem cells or iPSCs, we call them in hand, then we have better control on them. We can turn them or differentiate them into any cell in the body, heart cells, brain cells, kidney, liver, you name any cell type, there are protocols available in literature to differentiate iPSCs towards that phenotype. And these iPSC derived different types of cells can be used for personalized manner in a patient specific manner to understand the pathophysiology of some genetic disorders or any disease, you can say. You can also use these cells as a source of cell therapy for patients. In our lab, what we do is we would take blood from patients and healthy individuals, isolate peripheral mononuclear cells and reprogram them to iPSCs, and then we differentiate iPSCs towards neurons, cardiomyocytes, fibroblasts, and many other cell types, depending upon the phenotype of patient, if we are modeling any patient through these. This is just an example. This list includes some of the disorders that we have already modeled in our research program through iPSC technology. This is all FDA approved, by the way. This is another example of iPSC-derived beating cardiac cell sheet from the lab. Allogenic stem cells or donor-derived stem cells, whether they are MSCs or iPSC-derived cardiomyocytes, you transplant these cells to infarcted heart in animal models. They start the repair process and promote angiogenesis in the infarct areas. And the outcome of clinical trials was very encouraging, no side effects after transplantation. And there was improvement in heart function as well, but it was not consistent. So we wanted to know what happens to these cells after you transplant them in the injured heart. We figured out that host immune response kicks them out of the system, and that's why they don't survive in the heart after transplant. So in our ongoing studies, what we are doing is we are trying to fix the cells and host immune system to prolong survival of transplanted cells in the heart. These are some of the publications on why cells get rejected, and this is the team that is working on. Some of these students have already graduated and some are here in the lab. So I wouldn't get into that detail, but I want to talk about some of the biomaterial-based strategies that we are developing in our lab to prolong survival of transplanted stem cells in the heart. And one of the materials that we use in our lab is mexine. Mexine is a carbon-based material. we synthesize mixing quantum dots and nanosheets. We start from scratch. And this is the procedure that we use to prepare mixing quantum dots. The application of mixing as an anti-cancer agent, antibacterial or antiviral agent, and as a source of for energy storage is already being tested at different levels by different investigators. In my research program, we are mainly interested in application of mixing nanomaterials in cardioimmunology. Dr. Ali Reza Rafired, who's sitting at the top, he is leading these projects, and these three students, Keshav, he recently graduated, now he's a postdoc in the lab. So they are trying to use mixing quantum dots for different biomedical applications in the lab. So we have now optimized the protocols to make mixing nanodots immunomodulatory in nature intrinsically. One of the reasons that attracted us towards this material was its safety profile. When we tested the biocompatibility of mixing with different types of cells, we found that mixing is highly safe with mesenchymal stem cells, iPSCs and iPSC-derived different cell types, neurons, cardiomyocytes, fibroblasts, and several other cells that we tested in the lab. In our ongoing studies, what we are doing is we would take mixing quantum dots and embed them in chitosan-based hydrogel to make immunomodulatory hydrogel or platform, and we use it to deliver stem cells to the injured heart. What we do is, in our models, we take cells from Wistar rats and induce myocardial infarction in Lewis rats by ligating coronary artery. That this model mimics heart attack or myocardial infarction. And we inject hydrogel loaded with cells right in the infarct area, and then study host immune response and survival of cells in the heart. So what we have found so far is that mixing quantum dots are able to cut down host immune response and improve the survival of implanted cells in the heart. Since we know that mixing is able to cut host immune response, we are also testing its whether it is able to prevent rejection of whole organ as such. I'm talking about stem cells here, but I think heart transplant is still the gold standard for patients with end-stage heart failure. But immune rejection of transplanted organ, any organ, heart, liver, kidney, is one of the major issues when you transplant donor-derived organs. Allograft vasculopathy in this case plays a major role. Activation of T cells plays a very important role in the pathogenesis of the allograft itself. So we developed this model in the lab. We transplanted a piece of aorta from one species of rat to the other with or without mixing quantum dots to see if mixing is able to prevent allograft vasculopathy and prolong the survival of the transplanted aorta. So we studied infiltration of immune cells within the allograft. What we found was that the infiltration of cytotoxic CD8 positive T cells decreased significantly, and there was an increase in the infiltration of regulatory T cells. Regulatory T cells are able to suppress the host immune system. So this was a good finding for us. We also wanted to know whether mixing is able to prevent damage of the allograft itself. So for this, what we did was we stained the tissue sections with alpha SMA, which is a very good marker if you want to study damage within the allograft. So what we found was that in the control or vehicle group where there was no mixing, there was a decrease in the expression of alpha SMA, but treatment with mixing dots mitigated this decrease in alpha SMA expression. So I think I can comfortably say that mixing quantum dots have the potential to prevent allograft vasculopathy and prolong survival of transplanted organs. How mixing does it? We performed global RNA-seq analysis, and we found that the level of several genes related to cell survival in immune cells went down, and there was a decrease in the level of genes related to inflammation or interferon gamma pathway. So this is how mixing prevents prolonged survival of allograft and cuts down immune response. When we got into depth of it, we found that treatment with mixing quantum dots was able to decrease the level of CD4 positive cytotoxic T cells and increase the production of regulatory T cells in our immune cell population. And our metabolomic data suggests that mixing quantum dots are able to reprogram T cells towards Treg phenotype. So that's how mixing promotes immune suppression. We also have data that tells us that mixing quantum dots, the size of these dots it varies from 3 to 10 nanometers. They are very small. We can actually control it. So they get into the antigen presenting cells through active endocytosis. Once inside the cell, they are able to cut down the expression of CD86 and upregulate PD-L1 with the net result of suppression of T cells. So this is how mixing promotes immune suppression. This is all reported. The other application that we are using mixing quantum dots is for maturation of iPSC-derived cardiomyocytes. For this particular application, we use tantalum carbide, which is another form of mixing that is there in the lab. So tantalum carbide mixing is highly conductive in nature, so that's why we thought it could help us in promoting the maturation of iPSC-derived cardiomyocytes. So when we differentiate induced pluripotent stem cells or iPSCs to cardiomyocytes, we don't get fully mature cardiomyocytes. This is how the cell in the picture with green fluorescence, this is how iPSC-derived cardiomyocyte looks like. But an adult cell would look like is a rod-shaped, binucleated. So that's where we want to reach. In the past, people have used different conductive nanomaterials, including graphene, to promote the maturation of iPSC-derived myocytes. So that led us to hypothesize that mixing, being conductive in nature, would help in the maturation of induced pluripotent stem cell-derived myocytes. So what we did was how we grow. So normally what we do is we would grow iPSC-derived cardiomyocytes on a matrix gel. So we embedded mixing quantum dots in the gel to make it conductive in nature, and then we were growing cardiomyocytes on top. And then we studied the level of maturation in these experiments. One of the key parameters to understand the maturation level of iPSC myocytes is calcium signaling, which is a very good marker for maturation. So in our case, what we found was that when we treated our myocytes with mixing quantum dots, there was increase in calcium kinetics, and calcium signaling was better. We also studied the electrophysiology or field potential of these myocytes that were grown on top of mixing-based hydrogel using microelectrode arrays. Cells were grown on top of tiny electrodes. So the conduction velocity among different cardiomyocytes was much better when cells were grown on top of mixing-based platforms. So that also led us to think that mixing helps in maturation of iPSC myocytes. How mixing does it? We performed multi-omics analysis, and our transcriptomic analysis suggests that several pathways that are related to maturation got enriched. Calcium signaling, gap junction pathway, focal adhesion, there was enrichment in these pathways. And our proteomics data suggested that the top enriched pathways in our proteomics included translation and ribosome related process, amino acid metabolism, and long chain fatty acid transport, and cellular responses to growth factor stimulation. These were all indicating that cells are gaining a mature phenotype. Metabolomics data also let us think that when you treat cells with vaccine quantum dots, they are able to gain a close to mature phenotype. So when we integrated this Tri-omics data using OmicsNet, we found that one of the pathways that was consistently being regulated was focal adhesion kinase, which is very important key parameter in the maturation of iPSC-derived cardiomyocytes. So this tells us that mixing quantum dots help in the maturation of iPSC myocytes. So we wanted to see if it all works in the in vivo setting as well. So what we did was we created mouse iPS cell lines and optimized differentiation protocols towards cardiomyocytes, and then we transplanted iPSC-derived cardiomyocytes with or without quantum dots in a MI model in mice, and studied maturation and heart function. Maturation level increased significantly. This was done by using immunostaining. Well, the factor that we studied was MLC2B, which is a very good indicator of maturation. Heart function in terms of ejection fraction and fractional shortening, there was a significant improvement in this as well when we transplant cells along with vaccine quantum dots. So vaccine helps in maturation of iPSC myocytes in vivo as well. So I'll stop here. I just want to mention that biomaterials have a lot of potential, but we need to know the right combination of cells and nanomaterials. We also need to understand the depth of mechanisms that help in repair process. Of course, there is a need for extensive collaborations among clinicians, scientists, and engineers. That will play a very important role. I'll stop here. I want to thank people in the lab who actually produce data that I present at different podiums, and my collaborators and funding agencies, CIHR, NSER, and European Union. Thank you very much. I'll take any questions if people have.
Thank you, sir. Thank you for that explanation. Just 2 minutes. May I invite Dr. M.K. Rathore, sir, our ex-dean and Professor Emeritus in Ophthalmology, to present the token of appreciation and memento to Dhingra, sir. Please.
Thank you, Rathore sir. Thank you, Dhingra sir. It's now my privilege to welcome one of the most respected scientists and innovators in proteomics and system biology, Dr. Sanjeeva Srivastava from IIT Mumbai has been instrumental in advancing proteomics research and capacity building across India and globally. His pioneering work in biomarker discovery for cancer and infectious diseases, along with his contribution to the Human Proteome Project, has significantly advanced precision medicine. An acclaimed researcher, educator and innovator, Dr. Sanjeeva Srivastava, to bridge cutting-edge science with real-world healthcare solution. Please join me warmly welcoming Dr. Sanjeeva Srivastava.
Good afternoon, everyone, and thank you very much, Dr. Manju, for a very kind introduction. Dr. Mardik for this, decision that we should do education for the medical doctors and then try to bring some of the new cutting-edge concepts of multi-omics here. And that's how we thought, along with Dr. Prashant Kumar, to co-convene and do this meeting here. I can do this as a point for slide change. Alright, so I come from IIT Bombay. Some of you who have not visited, please do visit the campus, beautiful, and have a lot of technologies to watch. Recently we conducted one international conference, Advances in Proteomics Technology, where we considered Dr. Mardik for his wonderful lecture, and we had a huge gathering of different outreach programs, and that also shows the interest among the students and the general public to learn about these technologies. Of course, some of these technologies are still very new, as some of you have mentioned in the morning, that knowing about what multi-omics are is not very straightforward. But again, we are trying to advocate that, we are trying to outreach that, and again we have the give award to Dr. Mardik for his visionary view of looking at how to take this next step of connecting the clinicians with the researchers and scientists and technologists and bring the best for the patient care. I think we cannot work in silo, we have to work together to really bring the best of technology and the clinical aspects of the patient care. With this, let me just come back a little bit basic. Some of you are probably need a new and basic introduction of why we are doing this symposium, right? So the very basic fundamental of life is that your genome is defined once for all. And you think about a very simple example of butterfly, all the stages genome remains same. But what changes dynamically is the proteins, and that gives the different flavor, different type of physiology, different changes, the colors, all of that given with the proteome. Likewise, another big example is the frog, amphibian. When you look at both the tadpole larva and also the adult frog, two different type of lifestyle, only it can survive because of the dynamic protein which changes. Again, our human body, right? Like when I went from Delhi, several years back, of course, to Canada to do my PhD, from plus 48 degrees to when I went to Edmonton, minus 40 some degree, our body was able to adjust, that 88 degrees, So what governs that dynamic process, not the gene, but what makes from genes essentially looking at the different shifts of protein, different dynamic protein interactions, which provides that buffer and helps to take that forward. So proteomics, actually not just looking at some protein, we are looking at thousands of protein in totality. We are trying to look at their multiple properties, how the proteins interact with other protein and govern biological function, where they localize, how they modify, and all of that helps only possible if we can look at some of the cutting edge technology. Then we look at the mass spectrometer, which we established at IIT Bombay as a national facility for doing proteomics research. Some of these we are going to cover you in the afternoon session. And we will also try to run some sample live on the mass spectrometer, which is one of the latest 4D omics technology which we established at Timstorf, and we'll show you some live run from here that you can do the things very straightforward and simple way. Our idea is to convey you, don't get away that these are very difficult technical thing. I'm sure while sitting in your clinics here, still from your patient samples a lot can be obtained in the future. So as a part of the understanding the human proteome, molecule called the human protein, first time the draft of the human proteome mapper published in 2015, but then much more refined draft and a stringent draft published in 2020. Now we have idea for 91% proteome, which is keep increasing, almost 94% proteome, but still we have not reached to knowing all the protein of human body. A big initiative happening in China named PIHub, which where I am also representing India, is much ambitious project to look at the human physiology and diseases over the next 30 years to really understand the things in a much more comprehensive manner, and those kind of efforts are required in India as well. So my lab does work in various aspects of even looking at the technology, looking at the database and software development. Being an IIT, we have a lot of those innovative students from different fields and other kind of engineering discipline, plus a lot of biologists as well joining, medical students joining, to also investigate problems like looking at the disease like cancer. So I thought just to give you, in my today's presentation, two brief aspects of the things. One, some recent work what we have done on the brain tumors, and a little bit more work about some of the databases and software which we are developing from our lab. So brain tumors are very deadly. I don't think I need to emphasize you more. We have been looking at different type of brain tumors from glioblastoma, meningiomas, pituitary, medulloblastoma, etc., and trying to look at what kind of biomarkers which can make the detection early or make it much more precise, make it, you can make prediction for the disease severity form, and what could be best therapeutic targets could be identified. I just thought to show you a more recent story which we are doing along with Dr. Prashant Kumar and my PhD student now, Dr. Ayushi, has already graduated. We have first time looked at Indian patients, glioma and glioblastoma samples, high-grade glioma patient, and thought to utilize both the cutting-edge technologies, genomics and proteomics together, and see can we get something more useful and much more informative. And what the results came are pretty promising. First time we see the kind of mutation what we have not been reported in the cancer genome atlas, which is one of the largest forum for looking at the cancer mutations in brain tumor and other kind of cancer from US data set from NIH and other programs. Now we see some very unique mutation which are defining the high-grade gliomas in Indian patient, which was earlier not seen. Then we thought, can we now take this data forward and also look at the entire proteome data and all the kinases and modifications, and then overlay the two layers of the gene changes and the protein changes and the modification, and that led us to find out three different clusters emerging, which is defining from the IDH wild type mutant and the mixed population. So think about the brain tumor and the glioblastoma, which one would consider as just one subtype, but within that there are actually multiple subtypes, and they will require attention for further treatment, and those kind of various pathways which we have identified will be utilized, taken together, how well one could take those cues and take those towards the better treatment for these subtype of the patients. So again, we are getting a lot of information, a lot of interesting things to look at the immune responses, looking at which of the key pathways are being perturbed in those subtypes, which is eventually going to help out the clinicians to take better decision for the patient therapy. Additionally, on the another brain tumor, which is meningioma, more prevalent brain tumors, not at this day as glioblastoma, but even there was no signature available for defining low grade versus high grade of meningioma. So we did some discovery work, and then we did a lot of machine learning and bioinformatics to come up with a panel which we defined with a machine learning model with Professor Graham Ball, our collaborator from UK, and that led us to come up with the cluster and the panel which we also patented, and then we further validated and tried to come up with a panel which could utilize to differentiate low versus high grade of the glioma, and this work is done with Tata Memorial Hospital in Mumbai, and we will be taking more samples to then take it forward for the clinical testing as well. So this is something, again, the trajectory of looking at a clinical problem, defining the targets, validating the using mass spec technologies, and then planning to take it to the next level. So while we are doing all this kind of research on different brain tumor, what was actually challenging, that even the fundamental brain itself is very challenging and very complex organ to understand. There is no defined proteome of brain itself. So we look at the brain proteome using the Human Brain Proteome Project, collaborators from Dr. Enrique and others from Spain, and then we try to develop first time different type of brain map and publish that along with the database, which my student Dipru Biswas was leading, and I'll just show you some visuals for that. So if you could just play this video. So idea was which region of the brain are having what kind of basal protein expression level. Then only you can compare them when you are looking at the gliomas or meningiomas, because you need arachnoid sometime, dura sometime, glial cell sometime, and you need to know the protein expression across all of these lines. So we looked at each region and sub-region of the brain from both Indian patient population as well as from the Spanish population, and we then try to define the what is the protein expression level and the phosphoproteome expression level across different spheres, and then we develop this database so that anybody can go back and look at the expression of these protein profile. Additionally, the editor encouraged us to make a database and also then utilize other data sets available currently, which we then integrated. This was the COVID time which we started this work, and almost 56 human brain diseases for both neurodegenerative disorders as well as for the different type of brain tumor. Everything we integrated with the unified pipeline of the data analysis, and we then developed different type of domains which we thought to provide the data and information to the community. So what you get from this portal, if you go to brainprod.org, you can look at different hemisphere, you can look at protein expression profile. From the Human Brain Atlas, you can obtain all the information from that brain disease. You can then look at the marker curators, which we have done machine learning with a lot of IIT Bombay undergrads, to find out which is the best classifier panel for that disease. Like you are hearing from Dr. Harish about the NF2 gene, and we could see that coming in the meningioma as one of the top candidates. But when we look at the neurodegenerative disorders, Alzheimer, Parkinson, the new set of molecules and markers are emerging, which is very satisfying that you can see the very relevant clinically tested markers as our top leads. So I'd encourage you to go through this portal, and whatever brain disease you are looking at, neurodegenerative disorders, try to look at, for example, APP and ApoE, we found as the best markers for Alzheimer's disease, which is actually clinically already being in use. So while we're doing all the work, we realized that the brain research is very complicated, and what kind of drugs are being given, even that we do not have clear ideas. So if I can go to the next slide and the next video. So we developed a new portal, which is, next slide. Correct. We developed a new software known the drug-prod AI, which is utilizing the, again, the power of the artificial intelligence, but how you will see the process. You need to develop the good models from the actual data sets. So we utilized different features of the protein, almost 183 features of the protein properties, from various different ways, including from the looking at the PubMed, looking at the 3D viewer, what is available, druggability index, drug information, all of that, assembled that together, and then a lot of machine learning specialized in 7 models were tested, and then we were looking at which models are more accurately predicting the druggability of these proteins based on these properties. So now, when we compared across the 2 models like XGBoost and Random Forest, they turned out to be most efficient to classify. We are using the US FDA dataset, United States Food and Drug Administration dataset, to build a model, what drugs have been approved, what have been disapproved, so there's authentic data to build with. And then we use that to predict the unknowns, and that's where we were quite happy that we could reach almost 85 to 90% of accuracy for unknown clinical charges happening. And that's where I think AI can reduce the wastage in our time and guide us better. Please go next slide. Next. Yeah. All right. So additionally, in collaboration with Dr. Arun Vitra from UCSF, I was on my sabbatical a few years back, and another problem of protein analysis, proteome level, is which protein conformation and protein functionality can be changed in cancer in response to the disease, which is maybe surface protein and membrane protein. So to look at that, a cross-linking mass spec is being used. Then we developed the software Alpha Cross Excel, which is using again the AI tool, the DeepMind AlphaFold in the back end, so that now we can take any structure of the protein, and then we use some simple Python program to come up with this software, which can now make predictions and look at the cross-linking the protein, also the structure elucidation where these crossings are available, and this entire thing can be done in the next 2 minutes time, which was done in the day's time. So this is another software which we recently published in MCP and is available publicly for being used. So what I want to just, maybe another video to play, I want to emphasize that we have to leverage on various ongoing activity happening around you. You are looking at a patient and they should be benefited. Around you, a lot of technologies are there. We have been looking at genomics. It started coming to the different ways in the path labs and the clinics. But now we have to go much beyond that and add more functional information. What kind of protein changes happen? What kind of metabolite changes happen? Because they are more dynamic. They will be changing in response to disease, in response to stress, in response to your drug treatment. You can monitor those ongoing manner. So can we look at the intricacies, the dance of these protein molecules inside our body and develop those networks which can help define the systems type of analysis of looking at individual and much more totality. Of course, it's not so easy. You need all of this, amalgamation, all of these technologies and the clinicians biologists, bioinformaticians together. But the future of medicine will be really powerful if we can do the things in a longitudinal manner. We can take the patient samples at different time interval, generate these data sets, and develop those to build a model which is like digital twin, and then utilize that to then make prediction in the future. Like if a patient comes, you have their digital twin and 3D model, and then you can look at which drug can be resistant, which drug will be more useful, and make those kind of educated decisions based on that patient data, not just from the available literature data. And then we can go from cellular level, moving to the various molecular level. So a lot of these interesting things will be happening very soon, and sooner you will be, understanding them and embracing them, more you'll be beneficial, more you'll be in the forefront. Therefore, I think it's important that we should start thinking about how to utilize the power of artificial intelligence and multi-omic technologies together to build this personalized omics map and use these digital twins to really take forward for our various type of medical predictions. But ongoing, you should not blindly just rely on the AI tools. It's not as simple as ChatGPT, which can develop your the grammar-based models. You have to very carefully look at patient data. It has to be much more patient-centric, much more population-centric, so that you have much more defined and tight information. You can't just use US population data and use that model and make prediction on Indian patient. So what we are looking at a partnership of clinicians, technologists, along with them support from artificial intelligence, and then use our intellect to do the better patient treatment. Thank you very much.
Thank you, Dr. Srivastava, and we are here to work together. So you have to be please over here, Dr. Srivastava. Now I invite Dr. Gigi Potesel, Professor Biochemistry, to present a memento to Dr. Srivastava.
Thank you, Pote sir, and thank you, Srivastava sir. Well, we have one last scientific lecture, and to conclude this series, I invite Dr. Prashant Agrawal sir, who is a distinguished cancer biologist and translational researcher with expertise in precision oncology, liquid biopsy, cancer genomics, and molecular diagnostics. Currently serving as Director of Research and Development at R.D.GARDI Medical College, Ujjain, and Chief Scientific Officer at Datar Cancer Genetics. He has played a pivotal role in advancing innovative approaches for cancer diagnosis and personalized treatment. Kindly join me in warmly welcoming Dr. Prashant Kumar.
Hi everyone. Thank you, Dr. Gautam, for the kind explanation of the work what I have been doing, and I must thank and extend a very warm welcome to Dr. Sanjeeva Srivastava. It is his vision that he decided to come to R.D.GARDI Medical College, and the idea is to collaborations and the cohesion. I must thank and also extend warm welcome to all the experts coming from abroad. Thank you very much for your time, and also the 5 students who came from IIT Bombay for the workshop session. So kudos to all of you. Can we clap for them, all of you? So I have been told quickly to speak about the adoptive therapy, what we are trying to establish in R.D.GARDI Medical College. And I must thank sir and madam sitting here that the vision of them, which they want to establish a center of high throughput and cutting edge technology. And why this is important, I'll try to summarize that why in cancer we are moving towards the adoptive therapy. The title of my talk is Harnessing Adoptive Cell and Gene Therapies. So when we talk about any such therapy, and especially adoptive therapy, when you go back in 1970s, the cancers were getting treated by chemotherapy. And that is what we knew. And still in this era, 70% of cancer we treat with the chemotherapy. But over the time we advanced. And we understood the technology came and developed. We understood that NGS and next generation sequencing, which took 13 years to just sequence one human genome, and with $1 billion of expenses. Now, probably maybe in 2 days, you can sequence a human, whole genome sequencing, with a cost of 100 USD. So the technology improved, and from genomics, we moved to transcriptomics, and you have heard all these experts speaking about the multi-omics. And multi-omics, omics is coming from the word ohm. And ohm is the large amount of data. Doesn't matter what data you talk, but whenever you talk of omics, ohm, you just imagine that we are dealing with the large amount of data. But the bigger challenge is not only the data, and I love the videos what Dr. Sanjeeva was showing, that all the AI, because the artificial intelligence, why we need artificial intelligence? Because you want to integrate the data. And believe me, the 40% of the world data coming from the healthcare. Imagine, so 60%, we talk any other facets or any other domain, but the healthcare is actually contributing a lot. And this is where the artificial intelligence is going to help us, because we could be very quicker, we could be very robust. Now, if you see this slide, when we talk about the therapy, I talk about the chemotherapy, and then we learned surgery, then we understood the combination that we have to combine chemotherapy with the surgery or radiation, and then targeted therapy. And we talk about the targeted therapy, it is coming because we identify a single mutation, SNV, single nucleotide variants. And against that variant, we have developed the drug. And beautiful example, many such examples are there. So single nucleotide variants, you can target with the targeted therapy. Then we came in the era of immunotherapy, where the immune system could be activated. And PD-L1 and CTL4, these are the two amazing, I would say, the warrior, where we know that, okay, how can you target them? But when we talk about the adoptive therapy, or these are the advanced immunotherapy, in a very simple layman language, these are the advanced immunotherapy. The term adoptive means adopt. So you want to adopt your cell, cancer cells, so you are going to give the immunotherapy, but the cells are getting expanded ex vivo. And those cells are given to the patient because they can attack with the tumor cells and they can kill. So all this technology which is going to happen ex vivo, we are putting them in the patient, infuse them. And this is where the whole idea of establishing this center in R.D.GARDI Medical College, where we are focusing on adoptive therapy, CAR T cells, tumor infiltrating lymphocyte, and gene therapy. Now if you see the therapies, the landscape of advanced therapy, first one is gene therapy. So when we talk about the gene therapy, it is all about the gene. So you can manipulate the genes, and there will be viral gene transfer, you could see non-viral. Both ways you can do this. Then we are coming with the cell therapy. Cellular therapy, the term it comes, we are not manipulating anything. We are taking the tumor cells from the patients, we are expanding them, ex vivo culture them, and then we are re-infuse. Only thing we are doing is we are enriching those cells with the high T cell populations. And these T cells are going back to the same patient and they are attacking the tumor. And it could be autologous and allogenic. Autologous means you are taking the patient's own tumor. Allogenic, there will be a donor. RNA therapy also, so DNA, RNA, and RNA therapy where you could use the mRNA, and mRNA vaccine is one such beautiful example that we are using. So gene therapy where it could be viral, non-viral, and viral could be lentivirus, adenovirus, AVV, so multiple viruses depending on what is the size of the DNA you want to insert. And cellular therapy I mentioned you. So the bigger challenge with the tumor is the tumor microenvironment, which we understood later that we can leverage. And tumor microenvironment, when we speak, it has all the different cells. Initially, we only targeting tumor, but we understood that we need to target the tumor microenvironment also. There are multiple peptides are made now, especially in pancreatic cancer. And those are targeting the tumor microenvironment. So it has hypoxic, it has necrotic, it has acidic. So all such kind of environment is there. And of course, you have all different cells, macrophages and fibroblast cells, blood cells, CTL. So these are the tumor microenvironment. And when we talk about especially the CAR-T, we become, I think, we champion in case of liquid cancer because we identify a target, CD19. So I'll tell you very simple, what is this concept of CAR-T cells is that we need to first identify a marker which are overexpressed on the tumor cells. The goal is that you are targeting only the tumor cells, not the healthy cells. So any such therapy, if they are killing your healthy cells, then it will be toxic. And that's one of the bigger challenges with the chemotherapy. So the advanced technology, because we wanted to identify those targets, and liquid cancer we are very successful because we identified those targets which are prevalently, they are overexpressed on the tumor cells. But the solid tumors still a big challenge. And the challenge is, I was mentioning about the tumor microenvironment, very thick tumor microenvironment. So for invading those tumor microenvironment, reaching out there is a bigger challenge. PD-L1 overexpression, so this is another challenge. And there are multiple such challenges are there. The ECM, extracellular matrix remodeling. But every challenge brings you an opportunity. And the opportunity is that you can target those with the drug in hypoxic condition, anti-cancer drugs, targeting VEGF, enhancing T-cell infiltration and adoptive cell, which could be vaccination or T-cell activation. So, as I mentioned you that all these processes looks that you just collect the cells and then you activate all the cell infusion and lymphodepletion, cell expansion and isolation. So the different type of adoptive cell therapy, and I think by the time you are moving from this room, you will remember this term adoptive. Why adoptive? And what are the different adoptive cell therapy? And you can use CAR-T or TILs or TCR-T. However, if you see the overview, each of these methods, when I am talking about CAR-T or TILs, there is a manufacturing workflow, very simple workflow. Antigen recognition is the challenge, and then clinical evidence. So I'll very quickly touch base some of the very exciting trials which worldwide done, and that excited the clinical community that why we need to enter in the adoptive cell area. So I would just skip some of the slides and I'll show you the findings. So this is called lifileucel, a tumor infiltrating lymphocyte which got recently FDA approval, and beautiful data in melanoma. So I'll just show you this data where 2% or 2 patients, so 3% of the patients showed complete response. So what you see here in the red, in the green, sorry. So they got the complete response, and then 33% of the patient got partial response. And most of the patient, when we talk about the adoptive cell therapy given, these are the very advanced stage patients. So all those therapy has been given. The patients are exhausted with all chemo and radio and immunotherapy. So imagine that the clinician finally landed that what could be the treatment modalities. And this is where we enter in the area where we talk about the adoptive cell therapy. And you could see the patient, 44% of the patient showing a stable disease. And that's also again a miracle that you can keep the life of the patient could survive. So these data were overwhelming because you could see that in case of at least some of the cancer, we got beautiful results. Now again lifileucel, you can see the therapy response, 1 year DOR 69% and patient with overall survival more than 1 year. Again, tumor infiltrating, so LN-145 for the treatment of recurrent metastatic cervical cancer, and you could see the 78% of the patient had a reduction in the tumor burden, 78%. And median follow-up 7.4 months, and mean number of TIL cell infused, so I'm talking about the billions of cells. When we talk, then we expand the cells, and then we grow it to the billions, and then we infuse. And FDA grants LN-15 again breakthrough designation for cervical cancer. Another phase 2 multi-center study, which is LN-145 cell therapy, which is on the non-small cell lung cancer. And I'm just giving you such examples. And I truly believe that this is the time, this is the data-driven science. There was a time 20 years back or maybe 10 years back, we were much more dependent on hypothesis. But now the time changed. We show the data, there's no need to convince because the data tells everything. And these are the trials which shows us that how these adoptive therapy, either you are using CAR-T or TILs, making a sense, giving a far better survival, and this is another good example. So I'll just skip, it's all talking about the survival, response, the benefits, all these trials have shown, and multiple such trials are in the pipelines. So adoptive cell transfer therapy, ex vivo, and again lymphocyte depletion, I wanted to extend longer discussion, but I think we have to back things. So I'll just skip some of the things, but the idea is that for you to understand these three adoptive therapy, the TILs, CAR-T and gene therapy. So I'm just switching the gear and I'm talking about the CAR-T cell therapy. And recently you might have heard that the China approved the CAR-T cell for Claudin 18.2 for gastric. So these are the breakthroughs, these are the findings which are actually getting adopted in the clinic. And CAR-T cells, when we talk, it is not where you are not manipulating. TILs, we are taking the tumor infusing without manipulating. But CAR-T cells, when we talk, its name itself called chimeric. So we are going to do the manipulation, engineering, and this engineering you will do at the gene level, and those genes has to be inserted. And this is what you could see, that how it is administered, modification of T lymphocytes, then the CAR-T has been produced, and then it has to be infused. Now I was mentioning about Claudin 18.2, targeted CAR-T cell, and this is again new therapeutic option for HER2 negative advanced gastric cancer. And again, this is amazing findings. It's helping for those patients, trust me, for the gastric cancer patients where there is no therapy working. And again, the tumor response, you could see among 90 patients with the targeted lesion at the baseline, 70 patients showed various degree of tumor shrinkage. And you will see 51 patients had target lesion, an ORR 54.4%, DCR 96.1%. So this shows, and again you could see the response, and the graph shows beautifully that how many patients responded, complete response, partial response, and stable disease. So I will show another FDA. So FDA approval, first two CAR-T, you are aware, and these are two CAR-T which was approved 2017. And again, this is on the diffuse large B-cell lymphoma. Again, you could see the survival. It impacted the survival. The best overall response rate was 52%. And you could see a total 93% and 12 months of this relapse-free survival, which was 65%. Another CAR-T which was approved by FDA, and again it impact, and you could see that the B-cell lymphoma that was refractory and hard relapse, no more 12 months, first line chemotherapy, 180 patient was taken in the trial, and the randomly assigned, these are the random trials, and you could see the trial response. So all these examples just shows that why we are heading towards the adoptive therapy, especially CAR-T and TILs. This is another example for B-cell lymphoma, and this was 5-year follow-up, ZUMA-1, and this again you could see the trial response. On ongoing response, 29, relapse 51 patient, and the median, if you see, 65%, 35% relapse, and 12% in non-responders. Long follow-up, this is CD19 CAR-T cells. I was talking about the leukemia, and this worked beautifully. And there are multiple companies, now you may be aware that Indian-centric, there are companies who has developed the construct, and now they are making it in India. The cost has been reduced from crores to lakhs. Now people can avail probably what percentage of patient, but it could be given to 25 to 35 lakhs, those CAR-Ts. So again, I'm just giving you many such examples. This is phase 3 trials, again very supporting data, and this also open the randomized trials which give an amazing findings. So the bottleneck, the factors limiting the treatment efficacy, and I was talking about the tumor heterogeneity, antigen escape, T-cell infiltrating immunosuppressive microenvironment, and we talk about how we would go for the solid cancer. Another beautiful example, anti-tumor activity, EGFR, specific CAR-T cells in non-small cell lung cancer. So I'll skip because I have been told, I think Dr. Sanjeeva is showing that my 10 minutes is over. So I wish I would have speak more. Claudin 18.2, I mentioned CAR-T demonstrated therapeutic potential in advanced gastrointestinal cancer. Phase 1 trial given a beautiful results. So CD19, I would just touch base quickly. The
last gene therapy I was mentioning you. So when we talk gene therapy, it means we are going to use a delivery system, and it should be a viral based delivery systems. And again, it's a mutation compensation, suicide, and different kind of approach, but the goal is to how to kill the cells. And I think this is one of beautiful example, first and only FDA gene therapy treating in adults, and this is in the high risk BCG, non-muscle invasive bladder cancer. So I would move forward, I would talk about this particular product which is called GenDC. This is a China based company. These are the only company who has developed the gene therapy product, and this is where they treat head and neck cancer. So in cancer, when we talk about the genes, these are two genes which we focus. One is oncogene, because oncogene causes the tumorigenesis. The other gene is tumor suppressor genes, because tumor suppressors which actually suppress, and we want more tumor suppressors. And I always give example, that anecdote, that elephant will never get the cancer because elephant has 50 copies of P53 genes. We have only 1 copies. So the idea is that where there is a P53 mutation, what is required, that can we restore the function of tumor suppressor gene P53, and that's the idea of gene therapy which we are working in R.D.GARDI. We are making first Indianized product where we are also working on those P53 gene with the different kind of micro RNA cassettes. So hopefully very soon we are going to start the phase 1 clinical trial. So these are all the data which shows that how this is very impressive. And this is such beautiful example with Advaccine. You could see the patient has been given gene therapy, and you could see post gene therapy, the tumor lesions reduced drastically. Another beautiful example, Advaccine has been given, and again it gives a beautiful example. You could see the arrow where the tumor is completely disappeared or gone. So I would move forward. Key challenge in gene therapy, we are talking about all these limitations. But the conclusion and the future is very bright, where we could use the gene therapy based therapeutics approach and design much more advanced method, selective about the genes, and that is where I think the guys who are sitting here, the proteomics, where we can identify more of the proteins which are super expressed, overexpressed on those tumor cells. And I would see beautiful example, I'll just quote that I was in ASCO this year. And RAS is one such inhibitor. Historically, 40 years the scientists were struggling to find a drug, because the motive was very shallow, very difficult for any drug to fit. But now with the findings, the inhibitor has been developed, and this is coming with a very complex, the glue. So it's coming and it's binding, and it's doubling the time of survival, what have been known and what have been shown in pancreatic cancer especially. So such findings are very important, and I really thank you for your attention. This is an initiative at R.D.GARDI Medical College. I must thank Dr. Mardik sir for his vision, that when I approached him that I am very keen to establish a center where we could work, and especially first in MP, Madhya Pradesh, and you see sooner that we are going to develop things from this state. And this is the team who really contributed. So Dr. Manju Purohit and Dr. Gautam extensively, very handful for me coming here and asking and troubling them with all my questions. They were very helpful. Dr. Sevanti Lime, again, she is a stalwart. I call her precision goddess of the country. Dr. Kumar Prabhas, you have seen that he was the one who came and in the opening ceremony as the chief guest for this auditorium, and he is the director of Tata Medical Hospital in Muzaffarpur, one of the very refined medonc, and Dr. Amol Patel from the Army Medical College. So these are the very motivated clinicians, and these are very naive team I have, but they are doing and learning. So thank you very much for your attention, and looking forward to interact with you all. Thank you. So I'll just take this discussion forward, because we have a panel discussion planned. And I am very excited when we talk about the panel discussion, because when we talk, it is one directional talk when we speak or any of these speakers speak and talk. But when we talk about the panel, we bring such questions and ask their expertise. You're changing the slides, eh? So Dr. Sanjeeva I will tell guys, follow him. He's impressive. He was showing the slide and I was talking to Dr. Sanjeev Dingra. We are talking that from where he create all those extraordinary imaging and 3D, and he is AI person, being a proteomics he used, and this is the video he asked to play here. This was played in APT meeting in IIT Bombay, and just think his imagination that he brought us in bigger picture. Yeah, please.
Thank you, Prashant Kumar. He just told that the first cancer gene therapy by Chinese, the government has already permitted. And for last 1 month, I am discussing this with Professor who has developed this. And you all will be pleased to know that only 3 days back he has agreed to transfer the technology and develop the CAR T cell therapy against stomach cancer here in R.D.GARDI. The only condition he has put that this is permitted only for India. Outside India, they will deal directly. Well, all the talks, because you see how much lower we can make the CAR T cell cost or the genomics cost is a great question, because majority, say 95% of the cancer patients coming to us are through what you call Ayushman, and there the Ayushman or the government doesn't allow any of payment for any of these. They were allowing the PET scan about 6 months back, and now they have stopped that also. So our aim is to bring down the cost by producing in India, and I hope that we will succeed in short time. Thank you so much.
Yeah. So by the time they will fix it, may I invite the panelists. As I mentioned that we have a panel discussion, and I'll make it very interactive so that you find involved in the panel discussion. So may I just call the panelists. Dr. Josh, please come to the dais. Dr. Joshen, please. Dr. Harish, Dr. Sanjeev, and Dr. Sanjeeva. Please. So what Mardik sir told, I think it conveyed very strongly the message that what is needed, and yes, Doctor, yeah, Devashish, please come here, Dr. Das. That affordability and accessibility.
Good afternoon, everyone. Over the past 2 decades, multi-omics research has unveiled an extraordinary universe, hidden deep within our molecular fabric. From the code of the genome to the dynamic architecture of the proteome, we have mapped the invisible blueprints of health and disease with staggering precision. Yet a profound chasm remains. Much of this brilliant innovation remains locked within the quiet walls of the laboratory. The journey from a breakthrough at the bench to a cure at the bedside passes through a vast, untamed silence.
The scientist and the physician see this path through fundamentally different lenses. In the lab, the pursuit is depth, a relentless search for biological truth, mechanics, and mechanistic understanding. But in the clinic, standing face to face with the patient, the pursuit is utility. A desperate need for rapid, actionable answers that can guide a treatment plan or counsel a frightened family. True translation happens when these two distinct worlds finally begin to speak the same language.
To cross this frontier, we must re-engineer our approach. We need large-scale, multi-center validation to turn candidate biomarkers into trusted, reproducible tools. We must deploy intelligent clinical AI to distill massive, chaotic data streams into simple, intuitive roadmaps for doctors. Most of all, it requires forging radical, borderless collaborations, uniting technology hubs and hospitals from India to the global stage to turn big data into actual healing.
Because at the end of every single data pipeline is a human life waiting for an answer. This is where the science meets the soul. To guide this vital dialogue, we are honored to introduce an exceptional international panel of innovators, researchers, and clinicians.
Thank you, Dr. Sanjeeva, for such a creativity and bringing on the bigger screen, because this was completely unbelievable that he would create something like this. And this shows the innovation and something which you could create being a scientist, being a inventor, and I think being a discoverer. So thank you very much. Thank you very much, the experts. It's very humbling that all of you coming from different part of world and participating in this discussion. And I welcome you all. And our discussion would be more about the multi-omics and of course the cellular therapy, that how we are bridging the gaps. So can you just please move forward? Okay, the slides I can. Yeah, so I'm just giving you the premises before we start taking the questions. Multi-omics, you understood, all of you know what exactly the multi-omics is. The idea is when we start from the genome, we land with the phenotype. And with the phenotype, we would go further about the cell signaling, because each of these omics would give you multiple molecules. And which molecule, if you can just keep adding those molecules, you will identify at the end the pathway. When we talk about the cancer, Wnt signaling pathway, TGA beta pathway, the information about the pathway coming from each of these omics. So integration of multi-omics and artificial intelligence, now we are in the era of artificial intelligence. What it is actually eventually escalating and augmenting us for the drug trials, identifying novel drugs, and trust me, maybe probably 5 to 10 years back, we have to do, it takes years of time to screen thousands of drugs. Now it is done in a day or two. So that's the importance and that's the significance of artificial intelligence. We are into the drug design, and not only the drug trials, but drug design, trial startup, trial conduct, and again to identify and interpret the trial data. TILS and CAR-T I mentioned, and these are the ones which actually helping us for especially those patients who are very advanced patients and they are giving extraordinary results. Integrating multi-omics in cellular therapy, that is where our discussion, and multiple papers in the past published which actually empower the findings, how multi-omics is helping us in the cellular therapies. Multi-omics in CAR-T, and I was actually, Dr. Sanjeeva's the work he was mentioning, we have done from the glioma, and we identify some of the target actually on the glioma cells, and again I was telling that those could be also used to identify for the CAR-T cells. So this is what you could see, how genomics, proteomics, transcriptomics, and microbiome also helping us to enrich the tumor microenvironment, to identify the target, to identify the CAR-T cell library preparation, CAR-T genes, epigenomics again, because it actually deals with all those dynamic changes in the cell, metabolomics, and here is the questions which we would like to brainstorm with the experts and get what is their opinion and how they think differently from us. So very first questions that over the past years we witnessed an explosion. An explosion means multi-omics, multiple paper getting published. And from genomics to proteomics to spatial, and I think you have seen a lot of data from the spatial and single cell technology. Yet, on a small fraction of these discoveries have transferred into the clinical actionable biomarker. So the question is very straight that what is the lacuna that with so much discovery, and we are talking in the era of multi-omics and AI, but the clinical translation is not too many, and what would be the limitations? Yes, Dr. Devasys, can you please start?
Thank you, Dr. Prashant, and I'm happy to be here. The question here is that how the current scenario of using clinical research and helping the omics in vice versa way to engage the patient to make a better outcome. Where is the pain points here? One of the pain points I do agree, see that minimum interaction between faculty researcher as well as the clinicians. This is one of the major areas, because the timing and understanding and the domain of expertise are quite complementary, yet we need to sit together and work on it. That's one of the reasons. Second point, these are the deep tech. Deep tech means that you should have a patience to observe the technology, adapt it, and bring to the patient bedside and get a result out of it. It takes time, it takes money. And unless we do have the patience to go for a marathon and trying to hypothesize that we make a cure in a short time span is really, really not possible. So sitting together with clinicians as well as the faculty research or basic sciences as well as the patients to take the science forward to the bedside requires a lot of time, effort and money. And these are the, in my view, are the major lacuna at this point.
Thank you, Dr. Devasys. I just want to introduce him. So he is a professor at IIT Bhopal, and he is doing extraordinary work in the area of CAR-T. And I think again it's a pleasure that he agreed at the last moment to join this panel discussion. So thank you very much, Dr. Devasys. Yes, Dr. Joseph.
So I think it's important to kind of step back a little bit and think about where we're coming from in terms of how we're going to get to where we want to go. So if you look at 20th century medicine, it was largely based on individual signals that predict certain outcomes, and then we would target specific targets in that context. And that's because that's what we had available to us and what we were able to test. And the notion of omics is really the opportunity for biologists and biomedical researchers to start doing what other sciences like physics had already done, which was large team science to look at much bigger amounts of data. I come from a time when I remember really big corporations like Microsoft and Google, who all announced that they had developed software applications that could act like doctors. And they could talk to people, they could interview them, and they could tell them they could make a diagnosis. And they announced this with great bravado that we had solved it, we could make medicine much better for doctors. How many of those systems have you seen in the clinic lately? Like not one of them, right? It turns out that biology and medicine is much more complicated than what these guys thought it was going to be. It might have worked for software stuff and for simple sciences, but not medicine. And that's what drove all of us here to realize that we have to collect much more data, that we can't just look at one molecule at a time. We have to look at many molecules and recognize that these are complex systems, and we need tools to study those complex systems. I think and I think we're doing that now. I think you've heard already today countless scientists talking about systems where we've developed, looked at large amounts of data and started to come with certain conclusions. And I think that's a key start. I think the reason it's not yet in the clinic is that we have one more barrier to get over. If you think about any drug or any intervention that involves actual clinical usage, right? It has to be studied in large populations. You can't get a vaccine approved without looking at thousands of people. And even then, many people would argue that's not enough. Some of the studies on vaccines have involved millions of people. And right now, we're still in a phase with omics where we can look at maybe hundreds of people. Probably not enough to draw a conclusion that's applicable to a large population. So I think one of the barriers we need to get over is how we're going to get from looking at hundreds to looking at thousands and maybe tens of thousands or more. Still remains to be seen. I think it's all doable, but I think that's a big challenge that we face.
So I concordant with your thought, and you know that many of the vaccine trials failed when it moved from phase 1 to phase 2 or phase 3, because once the numbers grew, the trial didn't work the way it was shown in the phase 1 trial. So what the take-home message, what he is telling, that it should be the larger cohort, because and cancer is very heterogeneous. So heterogeneity, keeping in mind, it's very important that the trial should be planned in a larger population. Thank you, Doctor. Yes, Yoshan.
Yeah, so, okay, I think it's better. Yeah, I think because it's a panel discussion, let's share different perspectives of the reality. I think, yeah, as Josh mentioned, in the last decade, we have, revolutionized our ability to study human biology, and been amazing to witness how many data points you can generate. But I think we've also reached a point where technology is like, a tiger chasing its own tail, right? And we're at the point maybe where the tiger starts to bite its own tail. So things have become so complicated, so specific, so high-tech driven, that we're missing the point of, finding something that's actually useful. And I think if you just think about how smartphones have become successful, is because they have been built on something that is using simple things to make things work. Every child can use an app on a mobile phone, and it's a very high-tech device. So I think if we manage to convert all this high-dimensional data into something that we can actually use, absolutely, clinically or in the research perspective. We've seen many studies where you harvest genetic, proteomic, metabolic, lipidomic information, but all the time the same candidates reappear. So I think we also come to the point where maybe it's those few things we need to focus on and really learn about them first before we dive into some exotic corner of the high-dimensional space and find new fancy ways of doing something that might at the end of the day serve very few. So I think especially if you think not only about the most elaborate clinical environment with all the unlimited resources and possibilities, I think we also need to think about making this useful for everyone. Right? And find a way that sequencing or doing proteomics is cheap, fast, reliable, so they can be applied and become actionable in places where resources are not so plentiful.
Absolutely.
And again, that is for me, the point to simplify the message, to simplify the approach, and then, to see this simplification save lives, improve lives, rather than generating more data and making things more complicated.
Absolutely. Dr. Harish, what's your view?
Yeah, I think the original question was, what are the bottlenecks to getting precision medicine to the next era with multi-omics? 3 great answers: multidisciplinary collaboration, sample size and scalability, that's really important, distilling simplicity from complexity in order to design and test a hypothesis. To me, when I think about all 3 of those things, it comes back to something that we talk a lot about in our lab and in our institution, which is this idea of translational research. Well, what is translational research? I think a lot of people get this wrong. Translational research, to me, is a way of thinking. It's not whether you use human samples, it's not whether you're a doctor or a scientist or a master's student or a PhD student or a postdoc. It's a way of thinking. And what that way of thinking, to me, is how do you generate, design, and test biologic hypotheses that advance patient care. You can do that in a cell line, you can do that in a test tube, you can do that with mass spec, you can do that with stem cells, you can do it with the protein atlas, you can do it with large arrays, you can do it with CAR-T gene therapy, but it's really that way of thinking, and I think that you to think how you can be a translational researcher. It doesn't matter where you are, it doesn't matter what samples you have access to. Translational research is thinking about how your work can impact patients. And so I think that's the bottleneck, actually, where we're stuck.
Perfect. Yes, Dr. Sandeep.
I think a lot has already been said, and I agree with what has been said. So it's not that translation hasn't happened. Multi-omics data hasn't been used or converted to clinical practice. It has, but not to the extent it should have. Why? Because better dialogue needs to be done between clinicians and basic scientists. That's the key to me. Teamwork. I want to say that we are living in exciting times. It is so easy to connect with each other throughout the world. I mean, there are so many platforms you can use to connect with each other, and people are receptive, So sharing helps. Share your data, honest reporting. So I think these are the keys to take whatever we have towards clinical world and to industry.
So very strong message what he conveyed, that we should be in the hyper collaboration mode. It's very important, and that's where you could see that the international, national, and even students sitting in the room, trust me, the ideas can come pretty early in your mind, but until you're not going and meeting your professor and telling that, okay, there's something I think differently, and this is innovation. When we come, interact, you can come and teach me, because teaching could happen at every level. And what he's trying to convey, that any such bigger goal we could achieve when we collaborate, and collaborate among the department, among the expertise, and this is where we can collectively achieve the goal. Thank you. Dr. Sanjeeva.
So very quickly, it's already been said everything very well. I think availability of good biobanks along with the clinical data, metadata, is, at least I feel, one of the key bottleneck when we are doing a lot of clinical work. It takes a while to really obtain the right samples and to get all the patient data. It's very easy for us to generate those mass spec and proteomics and multi-omics data, but how to correlate them well with the actual patient metadata set is another key challenge. Second, of course, is the affordability and the cost of technology. I wish to do 10,000 and 1 million as well, but again, the funding may not be supportive that number to really, all the cost which is involved in that. So again, the cost and big initiatives are required. And third, of course, is what we are all trying is the mindset change and collaborations you're talking, how to have the clinical side and the technology side and basic side together to design the experiment properly, then utilize those technologies and generate the meaningful data. So I think all three.
So let's keep, Dr. Sanjeeva, again I'm coming to you. Let's keep Indian perspective. And there are tier 1, tier 2, tier 3 cities, they are top medical colleges, but you are in R.D.GARDI Medical College. And I admire about this college that there are many such brilliant doctors are there. But when you go and talk to them, they will come up with the idea that there's a lack of funding, maybe infrastructure, they want cutting edge and expertise. How would such medical college would participate in something which would change, transforming technology? And what is something we should do that many such medical colleges, which even the tier 3 cities where the medical colleges are there, how can they participate in something which is mind-boggling transformation?
So right, so again, like, when I was getting trained in North America and when I returned back to India, one of the key problems which I realized is actually, when you are doing any kind of, the bacteria, yeast, those kind of biological samples, very easy to do that. When you work on the human samples, to really obtain the meaningful samples with the proper storage is one of the key bottleneck. And if while, these hospitals, they're already looking at a lot of patients, if we can have the good biobank storage and along with them, a good bioinformatician who can store that, with the barcoding and with the clinical data, that will be, huge resource for all of us, right, in the future. So many rare diseases will come out of it, so many other type of disorders will be there, and everybody can do the things very quickly. But when you are thinking something, you have to start from the scratch, and then it will take you years' time to obtain that number, which can be anything meaningful. That's why we always, start something very small and we start claiming that it's something very big, right? So we look at 100, 200 patients and we think we have found a biomarker, but that's one of the key challenge of not getting to the, when you extrapolate to the large population, it may not translate that level because it's very sampling of the small size. So I think I would really request all the progressive medical centers to initiate the good biobanking and good bioinformatics. At least one person who is actually documenting all the data. And Dr. Pramesh from PMH, I think he has taken the lead for the cancer grade, National Cancer Grade, which is trying to connect a lot of these cancer centers and generate all kind of data, but mainly clinical side. But I would go one step more and store those samples as well. Yes.
Dr. Sanjeev, that day also, it came there on biobank issue.
Okay.
And you will be pleased to know that we have a biobank.
Wow, great.
And the sample number is nearly 55,000.
Fantastic. So you are already so much visionary and you always look ahead. This is one area which I think will definitely transform not only this region but even Indian landscape, the clinical research. That's a key bottleneck I see, returning back from North America.
So the take home message is very loud that we need sample but well annotated. That's very important. Annotated. Annotation is very important. Just storing sample will not help. And the bigger challenge is longitudinal sample, which many parts of India and the hospital they are missing. If we can have the longitudinal sample, that will help us to understand the disease pathology, identify the prognostic biomarker. So I think that's very important. Yes, Dr. Sanjeev, please. How you think that India could compete
with? India could compete with?
Compete with the transformational technology, and again the question going back.
Take back this question once more because I'm also being part of PI Hub in China, and what I see, some of these things should come from the top-down level with the government leadership as well, because at the individual level I think only small what we can do, but when it comes with the kind of mandate, when there is a lot of the resources are also being provided with the roadmap of at least 10 to 20 years, then only something can be done. With the limited funding of 3 years, 5 years, when the projects will be getting struck, will be not so easy for transition path. Yeah, please.
So, I mean, it's slightly different, but I want to say something. 5 years back, I do a lot of admin work for my university. 5 years back, we started realizing that within our different faculties, we have a lot of talent, but people don't collaborate the way they should. So what we did was, we started a new program there. $5 million we thought we'll give to one team, three teams will fund in a year. 5 years grant, but eligibility criteria was that teams should be from different faculties, engineering, medicine, and from social sciences. And it really helped us to ignite new collaborations. So I want to suggest something like that. If maybe you already have it, ICMR, DST, or any other funding agencies, if they have resources, they can start new programs, team grants, but the eligibility, let's say, should be, the teams should be from north, south, and from east or west of the country. That will bring people on one platform leaders like you guys can suggest that, if that makes sense to you.
Yes, please. Yeah. Agree.
I want to stay on that topic because I think the key word isn't competition, it's collaboration, working together. To piggyback on what Sanjeev said, we have these programs in the US that are now really the best types of grants. They're called these specialized programs of research excellence or SPOREs. The idea is it brings together many people. Typically there's 3 projects, just to give you a sense. Each project has a scientific lead and a clinical lead. I think this comes exactly to your point, right? It forces those people to talk, work together, and the end point isn't just papers, it's to be a trial at the end of that funding period. The other thing these programs do is they provide a lot of opportunities for early career investigators to get seed funding for their programs. So if you have an idea, like Dr. Prashant said, you can go talk to a mentor and you can implement that and you get access to all of these great resources. So that type of team science, I think that is where the future is going, because again, physics has been doing this for 50 years, right? You think of the colliders, you think of LIGO, you think of all these great projects. It can't be done by one person. We reach the point where no one person has all the expertise.
Absolutely. Yes.
Yeah, I think I can continue because I mean, it's about learning from each other, right, at the end of the day. And also if you want to think about how India should do something like this, well, I mean, you already see how other people have learned the process and you're learning how to take this on and adapt it to your environment with the constraints and the opportunities that exist. So I think there should be an Indian version of this, right? It should not be a copy of the UK, US, or whatever place you could imagine. Because I think you have so much opportunities here, talent, energy, all the things that needed for finding a solution that works in your environment, right? And I think that might look something very similar to how other people or other places are bringing this up, but it might also look very different, right? So I guess it's important to find sort of a common goal to hopefully agree on a few common sort of milestones that you could say, okay, we cannot achieve everything, but let's agree to try to achieve these things that we can then identify as flagship projects or as sort of, ways to demonstrate that we can do it, to attract attention, to show that this is feasible, to align thinking, because sometimes collaborative thinking is not embedded in everyone, right? So, and when you manage to align people, then I think you can be extremely successful. And just to wrap up my sort of statement here, I think a lot of people think about Sweden as a very innovative country, and I think it is so. But the fact is that people in Sweden, they have a very community-focused mindset. So it's less about, pleasing their ego, it's more towards how can we contribute to the greater society, to the benefit of the greater society. And that is sort of a fundamental approach to collaboration. It's not, 100% in everyone, but the majority of people that I work with, they're super collaborative, they're super curious. And when you share, that passion to be, each of us is at some point in time the most stupid person in the room, but at the same time, you're the person that has the steepest learning curve. And if you sort of, allow yourself to maneuver in those 2 dimensions of thinking, I think then you can reach very far, and even with limited resources, do amazing things. So I think that's...
I think, we are all blessed, and especially as students, you may not believe you are sitting in one of the best convention center in the country. And you are sitting here meeting the experts. If you ask Dr. Sanjeeva, for us to access the experts, we have to go abroad. For me, going for my PhD abroad, Max Planck or Dr. Sanjeeva flew to Harvard, the idea was that time that we were not able to access. We were, yeah, of course, but in India, sitting in such a beautiful infrastructure, you have experts, you have to catch them. Your curiosity should go beyond. And if you are accessing those experts coming from abroad, you can link, you can discuss the idea. Then that's one of the things I understood, hyper collaborations. No ego between the departments, professors, you need to talk each other. Only talk. And one more request, that publish even a single case reports. It's very important that people think, who sees the case reports? And many times when we PubMed, we do not find a single case report for any such variance. So we are publishing lots of case reports. But here also I think huge potential that each of the department, every year, they should make mandate. Nothing, just 5 or 10 case report you have to publish. Because the world will only know that you are working here when you publish and they can access. So very quickly, what is your comment, Josh?
Okay, well, I guess I would say we as biologists are just learning something that everybody else already knew. If you look in the real world, the idea of sort of multidisciplinary collaboration is just part of how it operates, right? Look at any company that's out there that's successful, and they will have a research enterprise, they will have a manufacturing enterprise, they will have a human resource enterprise, they will have people doing Marcom, people doing business management. They already have multidisciplinary teams that make that company a whole and make it successful. And for the longest time, biologists held out. And for them, it was this old adage, if all you have is a hammer, then everything starts looking like a nail. If you're a genomics enterprise, then everything looks like a genome sequence. And we need to kind of wake up and recognize that if we're going to solve biomedical problems, we have to approach it from multiple angles. And I would encourage everyone here to think about structurally building your institution in a multidisciplinary way. At the institute that I come from, we don't organize ourselves by departments. If you have a chemistry department, then everybody in that department looks like everybody else. They're all people with PhDs in chemistry. And that's not going to solve real world problems. Instead, think about the problem first. Focus on what is the problem I want to search. And then ask, what are the approaches I'm going to need here to make this successful? There may be a chemical aspect to it. There may be an informatics aspect to it. There might be, an epidemiological aspect to it. But you need to kind of ask, what are the elements that are needed here? And then go find leaders in all of those spaces to contribute to the problem, to contribute to the answer to the problem. I think we need to start thinking more that way.
Can I address one of the questions which I saw?
Yes.
Regarding how to improve the CAR T-cell or cell gene therapy in this context.
Yes.
So I think we talked about the oncolytic virus therapy. And being India having a lot of virus, we can really re-engineer them. And now the AI has made it 100x faster to create a virus than what you used to do in a year. Now you can do in a month. Okay. So that gives a really good hope that we can combinatorially add oncolytic virus therapy to existing oncotherapy, including radiotherapy as well as the chemotherapy plus CAR T-cell therapy. That's one point. Second point is that, as you know, that the cost is also heavily dependent on ICU. Okay. That is where we need more automation, a Tesla model approach where one size fit to many could create a scenario where the cost of ICU will be reduced so that any gene therapy or CAR T-cell, even bone marrow transplantation, all these variety of things can be substantially cut down to the range. And third is in vivo. How to target the T-cells in vivo, bypassing the transcription and all these things. And for that we need good help of virus. And again the field is rapidly emerging. In matter of months, we'll see dramatic effect. Yeah, that's my take.
Thank you, Dr. Devashri. So I'll take one more last question because of the time constraint we would just... So it's a very generic question again I'm asking that what are the highest priority of strengthening India? Again I'm coming back to India because we're sitting in India and I want that the argumentation that it will argument the precision oncology program or the ecosystem in this country. Very short answer if you can give the very brief.
Well, I think initiative like this is one of the the right way of taking forward, and we'll come together and discuss that's the best way to move forward.
No, I would totally agree with that. What you guys are doing, this should be done nationwide. People should come on one platform, discuss, share what they are doing, and go from there. Collaborations, as I said. Funding agencies can contribute in a significant way by changing the way they are funding now. They can, like I said, they can come up with innovative ideas to bring people from different regions together. So that's the way to go.
Agree with everyone. Maybe my one addition would be to invest in the next generation. Translational thinking, collaboration, ability to ask and answer questions. I think this is really critical.
And there's often a word we use when we try to collaborate or sort of share thoughts, and that's strategic alignment, which basically is sort of that we at least, we don't need to look at the same point, but at least focus on the focus on the same point, but look in the same direction. And I think, you know, using the dynamic and energy that you have here is something that maybe can be helpful to align people and bring them together. And then I guess it is a matter of finding a trustworthy first decision maker, I wouldn't say leader, first decision maker, that would then sort of, you know, pave the way for the next generation, like I just said, you know, to basically go the way, right? Because it's not going to be the old people like us. It's going to be the next generation who are going to execute it, because they will have the knowledge to do it right.
So I guess I would start by saying, look at the amazing resources that India already has. This is one of the smartest populations on the planet. If you look at the number of engineers and scientists that are being produced here, it's astonishing. And we're sitting here at the home of the invention of zero, right? This is where we are right now. The level of mathematics that was developed in India is, you know, unmatched. We also have a country that has the largest population on the planet. So the workforce is already here. So I think a key element to this is thinking about what are the problems we need to solve. I'll come back to that. Thinking about what do we want to fix, and then start to organize around how we find solutions. And you know, the hardest part that I find with my students, they're really good at going into the lab and generating data. Where they struggle is stopping for a moment and thinking about what it is they've already discovered and what does it mean. It's really hard to stop and think, and I would suggest stopping and thinking and defining the problems, and that's going to really help you identify where you want to go with the solutions.
So every time, Josh, you come to India, please make in your agenda spending 1 week time here in R.D.GARDI Medical College, and all the experts actually, so that you will be also pioneering the effort of R.D.GARDI. But thank you very much what you have told, that India had immense talent, and we just need to leverage our talent. So thank you very much. Yes, last.
My point is a little bit different. I'd like to see more or little reform in the medical education itself, parallel to MD PhD program, number one. That too, MBBS to MD or super specialization should be seamless. There is a lot of barrier and there is restriction. We have already set our goals that, okay, the super specialization or MD should be like this, fine. But the time gap as well as the restrictions in not nurturing these young people is creating a lot of loss to the society. And that's where I would like to see a quality MD PhD program, fellowship, early stage grooming the medical doctors to go to research come faculty come practitioner stage, so that many things can be addressed. At this moment, this is highly lacking in India.
So thank you very much. I think I will end this session here, and I really appreciate all the experts sitting here for your guidance and overwhelming words which would actually help us to decide the future and what is something we could do differently, which would make a bigger mark to the society, to the country, and I think I'm looking forward for a collective effort. So thank you very much for your quick and expert opinions. Thank you very much.
Thank you for such an insightful panel discussion. Before we proceed for the lunch, I would like to thank all the speakers and everyone present here. The entire day was filled with such insightful lectures, thought-provoking lectures, and I am sure the audience sitting over here now must be thinking into omics and going into multi-omics now. Thank you very much, sir. Thanks a lot. Yeah. I would like to invite Dr. R.D. ma'am to kindly felicitate Dr. Devashish Nayak, who has come all over from Bhopal, IISER. He is from Madhya Pradesh and is doing an extensive work in CAR-T. So ma'am, please. Dr. Devashish Nayak, sir, please. And I must really thank Dr. Prashant sir for having all the faculty members over here being the bridge between them and R.D.GARDI Medical College, and I'm now sure he is an integral part of R.D.GARDI Medical College, and for such a good lecture and the panel discussion. I'd like to invite Manju Purohit ma'am, the HOD of Department of Pathology and Director of the Central Research Lab, to felicitate Dr. Prashant Kumar. The lunch is served on the second floor, so we'll have to move 2 flights of steps onto the second floor for the lunch. And we'll be back in another half an hour for a very interesting workshop. We'll be having Dr. Sanjeeva taking the workshop. It would be live from IIT and we would all be enjoying it. Yeah. So we'll be back within half an hour. Thank you. Thank you.
Which is a chromatography setup. So we are right now connecting you live from our IIT Bombay technology lab, and what you are seeing is in one of our startup company from my lab, Multi-Omics International Private Limited, where we have set up this new chromatographic method and the mass spectrometric technology. I don't know if they can hear me as well. Shambhu, Avinash, are they able to hear me? Over there or? Probably not, right? So Shambhu, can you ask them to just show the long view of the entire facility setup? Okay, so let me just talk to you. So when we think about doing proteomics, all you are having any biological sample, you take tissue sample, tissue biopsy, you take blood sample, you take saliva, urine, cerebrospinal fluid, or any other biological sample for that matter. You need to extract the protein out of it and then ask you to fix the frame. Don't move. So once you have done the protein extraction from those biological sample, which is looking at not 1, 2 protein but thousands of protein, you don't know how many protein you have. Then you want to separate those protein and analyze those protein using these instrumentation and technologies. So what we are showing you here is a test of a very new technology, which is also known as 4D omics technology. And here one could separate the complex protein mixture to a very unprecedented scale where you would have not seen earlier. So what we do, we take the protein, digest the protein with the enzyme like trypsin or chymotrypsin or lyse proteases, so that you can chop down them and make the peptides. And these peptides you are going to first separate in the chromatographic method. And what you see, if I could use a laser, or I can just go there. Alright, good, it's working. So, as I said, you are digesting your protein to make the peptides. Those peptides you want to separate using this kind of chromatographic instrument. What we have is a very specialized and advanced chromatography. Comes from the company Evosep, where you take the small cartridges and peptides and you want to pass the peptide and clean them up on the tip, and then you are directly injecting that peptide into the mass spec instrument. So if you can go back to the instrument view. Instrument view, full instrument.
Which chromatography? Please, can you
Sorry?
Which type of chromatography?
We are looking at reverse phase chromatography, C18 material, where your peptides can bind, and then you are going to look at hydrophobic base binding and eluting solely the peptide based on the gradient of acetonitrile and formic acid, and you are looking at based on amino acid property. Some will have more hydrophobicity, some will be more hydrophilic, and you are solely sequentially with the gradient you are removing the peptide from the column. Then you are injecting them in this instrument, which is known as the Timstorff mass spec. Again, if I get time and we have more interaction, I can show some basics what this instrument means. But idea is, can you separate this complex mixture of peptide and look at how many of those peptides are there from different protein using databases. So this is the data acquisition part which is happening. Now what I request TAs over there from our mass spec facility at Proteomica, that they can start one live injection. Are they already started now? So can we see the screen, official screen where... So this particular technology, there are different type of gradients. So gradient means you want to separate the peptide. So think about it, in a tube you have maybe 20,000 protein or 10,000 protein. You did digestion of peptides. You got maybe 100,000 peptides. Those you want to separate. So slowly you are separating from the chromatography, and now you are looking at those in this kind of setup. So this is, now we can operate from here. So we don't have to be at IIT Bombay or any place. Somebody has to only keep the sample in the instrument, that's all, right? After that, all the interface of the software, we can do that. And now what you see that, the peptides have started coming out of the column. So we have set up a gradient which is of 44 minutes. It means we are telling the chromatography that in 44-minute time, you separate all the peptides from the column, and this is the setup which we have made. And now you see that the peptides are slowly coming out. You don't want all the peptides to come at once. So you have to optimize it so that all the peptides are following a Gaussian pattern. Okay? So this is something which is already ongoing in one instrument. Let it keep running. We will review that again when we get time. Okay? Let me go back to some other facility, other instrument, and show you that. Can we now move to our other facility? So you are in this instrument facility, which is our startup company Proteomica International Private Limited, where we have this setup. Now I will take you to our mass spec facility, which is the national facility which I am convening. Let's remove this thing from the main screen and then add only that as a main screen. And can you ask people to show in the landscape? Okay, so again, now what we are going to show you, we are looking at protein analysis using other type of mass spectrometer, other type of technologies. And this setup what we see, this is being used for looking at metabolomic analysis. From the same sample what you have prepared, you can also extract the metabolite, and that's much simpler method. You just take any of a biological sample, lyse it, add methanol 4 times volume, centrifuge them, keep it overnight, and next day again vortex centrifuge, and what you generate is metabolome coming from that sample. Metabolome represents all the metabolites coming from nucleic acid, coming from amino acids, coming from your carbohydrates, everything at the end representing the metabolite. So that analysis we are now doing in this instrument, and this chromatography. This chromatography is slightly different. We are using the gradient, which is high pressure liquid chromatography gradient, and you already see the run has already started, and in around 20 minutes time from that given sample we can look at all the metabolites from that complex sample. So again, the sample, somebody can keep it there. If you are the one who are preparing your sample here, you got the patient plasma sample, you just simply take 100 microliter of it, add 400 microliter of methanol, just keep it overnight, next day centrifuge, you got some supernatant, freeze it, done. You are going to get, in addition to your specific test, you are going to look at every other molecule which is present in that sample. And then you can analyze it and see something is very aberrant, something is very unique, different, right? You can get a lot of new information from the same sample, not much work to be done. Just take, a small aliquot, just throw some chloroform. If you want to add even lipidomics, you can add even methanol and chloroform, and you can generate lipidomics as well, lipid analysis as well. So all this is, as I said, what we want to convey you. We can't train you right away in terms of as an expert, but what we can at least convey you, in which way along with the clinical work or other experiments, you can use them as a good, strong complement to provide you a good hypothesis. So now, this is something setup which we are seeing for the metabolomic analysis using one of the instrument mass spec what you see on the screen, which is an Orbitrap instrument, which is a QE Orbitrap, which is a hybrid instrument having the quadruples as well as the Orbitrap, and then we have interface with the HPLC. I think we can go to the fusion now. So again, just go back to Amit and tell him to show the run again. Run is happening, live run. Alright, so I just want to you to keep in your memory that one run is already happening for proteomics in the 4D omics technology timsTOF. Now another run is happening right now on the metabolomics in the QE Orbitrap. Let me now take you to the third instrument. Alright, let's move to fusion. Okay, so now we are in another instrument which is very advanced instrument, which is like the timsTOF but having some different properties. We are using the nano LC. This is another chromatography. The purpose remains same, the decision what we had remains same for how you separate the peptides. And this run has also started, which we have set up a gradient longer here. Can we show the fusion instrument? And can we show the auto sampler and do an injection? Okay, so you are just trying to inject a sample. Sometime what we do, like on a Friday, you just keep your 40 sample, 50 sample which you want to run over the weekend, and it's all automated, right? You just keep in the right positions like this. You know which peptide samples in what position, and now you can operate sitting here. You can set up a method, and you can just tell A4 well what method you want, or B10 method well what method you want. Sometime I have operated the instrument from US sitting over there in my sabbatical or doing other work. So it's all very simple, right? Somebody will just place a tube, that's all for you. Or you are the one sending your tube and somebody just place inside the instrument, okay? So from there, then this is the chromatography, will take up that peptide and then going to separate it further. Now you are going to run in this instrument. Can we have the long view, broad view, and hold on there for the fusion? Alright, let's stay there. Okay, so I want to show the ESI and stay there. Yeah, so what you see in the, oh my god, they're just jumping up and down. No, just go back. Tell him to do the same way, it is not coming good. Can you ask them to also see what they are showing so that they understand? Growth of fixed calorie, they don't have to run too much. Move, don't move, just fix. Alright, so what we see this instrument, this is known as Orbitrap fusion, which is a tribrid technology. This instrument has 3 different type of mass analyzer, very unique instrument technology, having a quadrupole, an Orbitrap and ion trap. Their goal is same to how to separate the complex peptide ions in mixture. What you see in the front, which is this type of gauge, in that it is the electrospray ionization, ESI. Your peptides in solution form is coming to the ESI. It is being bombarded and getting ionized with the very high voltage, reaching to this instrument, the black box, which is the mass spectrometer, and then you are going to separate these peptides with different physical principles of Orbitrap and then generate data which you can analyze using the different databases. So what you see on the screen is another run going on, right? The third run is going on right now, for which is live. So we did one proteomics in the 4D omics, one proteomics sample we are running in the Orbitrap, in the fusion, and one we are running the metabolomics in the instrument which is the QE. Are we showing something on the artist as well, or? Alright, good. So now let's say you have identified some target. What we are looking at, we are doing screening, we are doing fishing. We do not know in that given sample or the tube what all protein or metabolite we have. But let's say you have run your 100 samples by now, 50 control, 50 disease samples, and you generated a huge list of the protein and metabolite, and now you look at some data analysis in which way which proteins or metabolites are most significant or differentially expressed. And that's something which we'll show you shortly here, right? How to analyze the data. So now you have identified the target. You are only focusing on, for your clinical work, you don't want to bother discovering all the time. You want to only look at which target to screen for the patients. So then you will go for the targeted proteomics. And now what we are looking at, this instrument, which is a triple quadruple mass spec, where we will define to the instrument only specific metabolite or peptide, their ions and transition you have to monitor. So that's something we are going to look at in this instrument technology, which is known as the triple quadruple based mass spec or targeted proteomics. So this is another run going on. I'll take a pause here, and I hope I have at least told you that it's very easy to run the samples. These are just different instrument boxes. Where you place a sample, they will acquire data for you. So multiple data acquisition is in process right now. Let's talk some fundamentals. All right? Are we interested in doing that, or you just want to analyze the data? Okay, so let's do some data analysis first. Okay, if we have time, then I'll come back and talk more basics. So what we'll do now, let's say this is instrument generating data. Now what is next, right? How to look at this data and analyze this data. Truly speaking, it's very straightforward, very simple. If you have laptops, please open it. We made it as a workshop, not just to listen to story. So if you have come here to learn something, do something. Otherwise, we'll tell you a story and we'll go back. Please open your laptops if you have and do some hands-on. These are some very simple softwares which you can beautifully use it and you will feel more confident. If you don't have, you can just align with somebody else next to you who can do that analysis, right? Okay, so there are different softwares which can utilize their abilities to help you out to look at the complex data. There are two types of data analysis. One is a primary data analysis. One is secondary analysis. Primary means when I'm generating data from the mass spec instrument, I need to first convert that data in a readable format. That's require a lot more specialized server and much more computing power. Not that you can't do it, but if you have 200 samples, you need much more computing power to process that sample because every file is some GB data files. So then that takes few days time, some time to maybe a day time, depending on the number of sample, and that will be not taught right now because that will be much more longer process of doing the primary analysis. But primary analysis is very straightforward. That comes mainly with the instrument softwares, which is having the set defined guidelines. But then that will generate you list of the protein. You know the name of protein or metabolite, and from that you want to do that which proteins are disease-specific biomarker or most interesting candidate biomarker or target. That's something which we are going to show you in this software named MetaboAnalyst, made from one of the Canadian researchers from University of Alberta, David Wishart, his lab. This software are primarily made for looking at the metabolomics data, but we are also utilizing it for the proteomics analysis. Of course, like this, there are multiple softwares. Perseus is another good platform, and there are multiple platforms. You can use in your laptop and do a lot of analysis. So here we will take you one mass spec data file or few mass spec control and disease files, and first walk you through the demonstration about showing how you can analyze your data file. All right? One round, let's do that. And second, if you want to do that, we can give you some file to test out, and then you can do on your own. So I'll now request Avinash Singh, he's my PhD student, going to graduate in 2 weeks' time, to give you a demonstration for this thing. And if you can't do from here, you can do that from there and speak. Because
okay. So thank you very much. Okay. So okay, we can. So before going to the first analysis, I would like to show what is the output of the primary analysis. So we got the raw data from the instrument, and that instrument just have the information of the spectra. So they have the just mass to charge ratio. But after that, we need to understand from which spectra what intensity of the particular protein or peptide is. So we do use different kind of software, as Sir mentioned. So we are not going there in depth. So basically those software just give you the intensity of the particular protein in your samples. So I will just show you how the output file of a primary analysis looks. So Sandhu, can you open the file? Yeah, so open the test one. Yeah, so as you can see, the first column is basically your gene ID, and the second column is your sample name. So just go up. Okay, So what you are seeing here, so basically, okay. So here we have the two group where the control and the Cushing. So there one is the disease and one is the control samples. So first column A is showing the protein or gene name, and the second apart from after that we have the samples, which is our like clinical samples. So each column, like they are showing the intensity of a particular protein in the different samples. So now we have the two group of samples and the intensity of those proteins. Now this raw file we will take and we will do the analysis. So now we will go to the software for the secondary analysis. Yes.
So it is good, they want to do it.
Okay, okay,
and
great.
They provide this file to them.
Okay, great.
That will be the work.
So how to so anyone like,
So I'm just asking them to share the WhatsApp and they will share their.
okay, great, great, great. That will be better.
So you can just share. Yes, exactly. This is exactly what we want to do.
So while we are transferring the data, I will just quickly review through the how the sample preparation workflow. In the meantime, Sambu, can you open the slides? Yes. So this is just an overview. Just go to the next slide. Yeah. So this is the overall different steps that we follow till reaching from the clinical sample to the mass spec. So there are multiple steps, like in the many minor steps and major steps. So just go to the first slide. Yeah, so as you can see, the major steps are there basically the 4 steps. The first is the sample collection, and then the preparing the lysate, and then extraction of the protein, and followed by the estimation. The first step is basically to get the protein from our clinical samples, whatever clinical sample we have. So you can have the serum, saliva, tissue, and different kind of samples. So first step is to get the proteins. Now the second step is basically, and also followed by, we have to quantify those protein, how much protein we have got from the different samples. So after getting the protein, now we need to digest the protein. So as the protein is a very complex structure, they have a different kind of bond and interaction. So that's why we need to make a linear or open structure of protein, then where trypsin can go and bind to the specific sites. So the second step basically is the digestion of those peptides. So trypsin basically cleave at the particular sites, and that it create our pattern of a fragmentation. So the second step is basically digestion, and after digesting those peptides, we clean them and then we inject in the mass spectrometry instruments. So after injecting in the mass instruments, we already gone through the review. And now after that, the fourth step is the data analysis and interpretation, that what we are doing right now. So now we can go to the next step, next slides. Now, these are the major minor steps that we follow till reaching to the data analysis part. So, I hope everyone get the files.
So let us keep doing it in the meantime.
Okay, okay. So, just to brief that, so the first thing is, as I mentioned, is sample collection. So you could have a different kind of biological samples, like a tissue, cell. If you are doing the cell culture, then cells, and then the serum, plasma, urine, saliva, CSF, or any other different kind of sample. So the major step is the first is the collection of the sample and making the keeping the integrity of the sample. So we need to store the samples in the proper temperature to avoid any kind of degradation and avoidance of a sample degradation. So after this first step, now the second step, as I mentioned, so cell lysis and protein extraction. So, and the second step, as you can see, that we need to extract the protein. So in the tissue, the protein are enclosed in the cell membrane and within the cell. So that's why we need to lyse them. But in other biological fluid like serum, urine, or saliva, they are already in open thing. But problem is that in the some biological samples, we have a very concentrated protein, like in the plasma or serum. But in saliva and urine, they are very less concentrated. So in some case, we have to concentrate the protein to get from our samples. So after getting the protein, we basically, as I mentioned, the protein is a very complex structure. So we need to unfold them so our tryptic enzyme can go to the particular site and cleave them. So yeah, so now we first extract them. After that, now we got the protein by using the lysis and different kind of using the different kind of methods. Now we have the proteins. Now we want to estimate or quantify the protein, how much protein we have got from our samples. So in this case, we are using different kind of, we have the demonstration. If you want, we can discuss those things as well. But so there are different way of quantification of proteins. So some are the Bradford, PCA, and other kind of Fallory, so those are some conventional method. But for our samples, we usually use the Bradford and BSA for the protein quantification and estimation.
Are they ready with the file? Okay, then we can start.
Okay, yeah.
So again, you can send some samples to us, which you think you want to take, acquire data, and we can send you data, and we can analyze data here. So this is the best part what you will learn, how to analyze data and interpret data. And running data is not very difficult. That will be something to be sent.
So now let's start. Now everyone has a file, so then we will first go to any browser and just type Metabo Analyst. So network is quite signal is quite weak here, but it's okay, we will do that. So now we will click on the click here to start. Then we will go for statistical analysis one factor. So please follow what I'm saying, and also you can look in the screen as well. So we have the peak intensity of each protein. So that's why we will select the peak intensity. And now, if you see, if you can go back to the our input file. Sambhu, yeah, just go to the yes. So now here you can see that these samples are not paired. Yeah. Okay. So I'm just showing there's a few drop down. So the what drop down we have to select. So before that, I'm just briefing what. Don't go to this page. This is your raw file, that input file. Yes, Metabo Analyst. Okay. Vishal. Okay. Okay. Yeah, he will. Okay. So if you drop down, go to the drop down format. There are different options given. Your sample in raw, sample in column, is a paired, unpaired. So you can select whatever sample type, how you set up your samples. So in our case, our data is unpaired, and we have kept the sample in a column. So we will select the second option, samples in column, and these are unpaired. Okay. Now we will select our file. So this file should be in either CSV or .txt format. So we have provided the file in the CSV format. So you can select that. Go to the APT 2026. The second one, the Gen. Yeah, second one, yes. So this is the file that we have given to you. Now we have selected is so everyone just cross check it is the 526 4 KB file. Now we will go to the submit.
Wherever you are slow, you can raise hand so that we can stop.
Okay, I hope everyone is in the same state. So this is just starting. Okay. So sometimes we get this kind of problems. So there is many duplicate proteins. So we will select the. Okay. So what happened basically? Some proteins they have with a common gene name. So when you are selecting the gene, then it will come as a duplicate. In this raw files, basically we have to remove the duplicates. So we will share it to another file to the group. Just share another file. Otherwise, it will take time to remove the duplication. So we are sharing the another file. So it can be done if you want to remove the duplicates. Just you can select a column and you can type in the Excel the remove duplicate. You can do that. But to save the time, we are sharing the another file. Okay, so meanwhile, anyone have any questions they can ask? Any question regarding sample preparation, anything? Yes. No, this is the data then we are using after the primary analysis. So we got the .raw file or .d file according to the instrument type, and this file is going to the primary analysis software. Those software gives the output and they will give you the protein and intensity. So basically these are the our primary analysis output file from the software. So many software like the FragPipe, Dian, Proteome Discoverer. So these are the multiple softwares. So I hope, have you shared the files? Okay, so we have here in the group, just download that file. So it might be you have a different file, but we will follow the same step and you will get the outcomes as well. So now we will click on the submit.
Data can be generated so live. We have a gradient which is only 5 minutes long gradient. Okay, and in this workshop which we did yesterday at IIT Bombay, we were able to show them 6 sample runs, 3 control, 3 treatment, 5-minute long gradient in just 30 minutes time. We had a very tight day and we sent them for tea break, and by the time they are back, data is already all acquired. This gradient is much more longer, comprehensive gradient, 45 minutes. Again, what this means, gradient means, you have too much peptide from that given sample. If you allow longer gradient and optimize it, you can get more number of protein. If you provide shorter gradient, you will get less number of protein because the protein will be very coming fast, right, from the ESI and the mass spec. So therefore, if you have time, it is a good idea to allow for the best possible longer gradient, and that's why we usually do around 45 minutes. And typically in previous instrument what we have, which is Orbitrap, we used to run for 2 hours long. But with the timsTOF, that is the longest is the 45 minutes. Additional thing which you should know that you don't need a whole lot of sample to be actually injected. What we are injecting is only 200 nanogram or 300 nanogram of peptide from a given complex sample. And previously we used to load 1 microgram peptide. What does this mean? If you are starting given 20 microgram of protein in a tube, very minimal, you just have the tissue biopsies or some of the liquid biopsy, small aliquots could be just less than 5 milligram, although sometimes they're getting that which is difficult in the actual patient sample. But that is also sufficient to obtain the sufficient proteome data. But I would say metabolome is much simpler and easier to do, given that you don't need a whole lot of sample. Even a small amount of sample, you just simply have to lyse them, or you take the serum or plasma sample and take a small volume and add 4 volume of methanol and extract the metabolite. So these are just ways of getting your peptide or metabolite out of your sample. Then the chromatogram which we are showing, you can separate those. Again, if you guys have time, we can show the QE as well. Shambhu. If we have some time, could we show the QE and the live run of that? Unless you can bring all up gradient possible. And that's where everybody don't need to reinvent the wheel. You don't have to spend so much time. If you are the medical students or the clinician or the medical aspirant, let me keep talking to people in the back as well, where they're learning so far. So, in the back as well, where they're learning so far. So, all right, I'm here. JEE, I teach first year, now I'm teaching, very good. So, क्या देख रहे थे वहाँ पे, क्या चल रहा था? What lines were going on? किस करो कुछ हम लोग प्रोटीन कोर्स के लिए कुछ होगा. All of you have done at least bio, right? 12 bio. तो क्या रन कर रहे हैं हम लोग? न्यूक्लिक एसिड, डीएनए, आरएनए, क्या रन कर रहे हैं? प्रोटीन, गुड. प्रोटीन डायरेक्टली हम एक्सट्रैक्ट करके रन कर रहे हैं और डूइंग समथिंग एल्स, क्या करके कैसे रन कर रहे हैं? सोचो, आप लोग बताओ, आप लोग ज्यादा नहीं तो कर रहे हो कम से कम. चल गेस करो क्या क्या चल रहा है. इतने पीछे क्यों बैठते हो भाई? यू आर बेस्ट माइंड ऑफ इंडिया. हम लोग का टाइम आईटी से क्यों वेस्ट करके हमें आपको बात करने के लिए? बिकॉज वी थिंक यू गाइस आर द बेस्ट पीपल टू इन्वेस्ट टाइम इन, राइट? अगर आप ये टेक्नोलॉजी सीखोगे, यूज करोगे, देन आई थिंक यू कैन मेक मच मोर डिफरेंस. इसको सीखने के लिए यूएस में आने क्या जरूरत है? प्रोग्राम में यहाँ बैठे सिखा दे रहा है क्लासरूम के अंदर. चलो आगे बैठो, बड़ा एक्टिव रहो. इसके बाद हम लोग क्विज भी करेंगे और सर्टिफिकेट तभी देंगे डॉक्टर मंजू व्हेन दे विल एक्चुअली आंसर आवर क्वेश्चन एंड डू द असाइनमेंट. विल नॉट गिव सर्टिफिकेट विदाउट फिनिशिंग इट. आई डोंट गिव एनी फ्री सर्टिफिकेट्स. चलो तुम लोग आगे आ जाओ फटाफट. क्या रन हो रहा है इतना भी नहीं मालूम. इतने लोग स्माइलिंग में बैठे हैं, सर लोग बात कर रहे हैं. इतनी कहानी हो गई. यू गाइस हैव नो आईडिया. चलो बताओ क्या कैसे डाइजेस्ट किया था आपका प्रोटीन को? कुछ गेस करो. मतलब एमबीबीएस स्टूडेंट में तो इतना तो पढ़ते आए बेसिक. चलो बताओ कैसे डाइजेस्ट करते हैं? चलो अरे भाई चलो 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 ऑलराइट. हु विल हेल्प मी? जो नहीं हेल्प कर पाएगा आगे, जो आंसर दे देगा क्वेश्चन का वो बैठेगा अपने प्लेस पे स्मार्टली. आई लास्ट मिनट नेक्स्ट क्वेश्चन. Okay, अभी क्वेश्चन करी है कि what we are running is not the protein, but what we are running, what we are injecting, what you see is not protein, but protein in some other form. What is that?
Peptide.
How did you digest that from protein to peptide?
Peptidase enzyme.
Into a student. And where that works? नहीं एंजाइम कहाँ पे काम कर रहा है? You have बॉन्ड पे कहाँ पे क्लिक होगा? पेप्टाइड बॉन्ड क्या होता है? चलो बिल्कुल एक्टिव आप लोग ये विकिंग इफेक्ट गुड टू अमीनो एसिड के बीच में बॉन्ड होता है, पेप्टाइड बॉन्ड है, वो क्या है? चल केमिकल हुए, व्हाट इज द पेप्टाइड बॉन्ड? CONH, राइट? CONH बॉन्ड है. अच्छा, वो जहां भी मिलेगा वहां पे क्लिक कर देगा. ट्रिप्सिन, डू यू नो, और डू यू नो एनीथिंग स्पेसिफिक वेयर इट क्लिक? स्पेसिफिक प्लेस पे जहां पे लाइसिन अर्जिनिन होगा वहां पे करेगा. ये कैन इट डू ऑन बोथ एन टर्मिनल सी टर्मिनल? नहीं, इट डज मेनली ऑन सी टर्मिनल. ठीक है, चलो सब द बेसिक्स. Based on this, then now your complex protein which was very long have been chopped down to some small polypeptides, right? उसको हम जो है रन कर रहे हैं, इंजेक्ट कर रहे हैं इंस्ट्रूमेंट में, and that's what we are trying to separate. Now think about if you had 10,000 protein, 10,000 protein को डाइजेस्ट कर दिया आपने, कितने पेप्टाइड आ गए? How many peptides do you think you have? आप लोग सब सेम क्लास के हैं? हाँ हाँ, तब तो और तेज होना चाहिए आपको, तो और बेसिक अच्छा होना चाहिए. तो कितने पेप्टाइड हो गए 10,000 प्रोटीन? Come, go, front, चलो, आंसर नहीं पाया इधर. They are going. You guys are enjoying your back seat. चलो go first. आप लोग आंसर करो. All right. Well, please be vigilant. Respect our time. Why we are spending time? We are also tired till yesterday late evening. We finish our workshop and travel till 12 o'clock here, and I'm coming here to teach you. So make use of your time and our time. So what we are showing you that and why we are giving you the motivation. As I said, I'm very active in my class. I've also got the best teacher award at IIT Bombay, the highest teaching awards. And I interact with my last student as equally as to the first student. And they keep asking questions where I see the activities more, I go there more. देखो वो reason थोड़ा active हो रहा है ज़्यादा. इधर आ जाओ आप लोग, नहीं तो फिर क्वेश्चन ज़्यादा आएंगे उधर से. Please come in the front, come on. सेंट्रल डॉग ऑफ लाइफ में 1:1:1 यू विल थिंक कॉन्सेप्ट, राइट? DNA से जो है इतने RNA बनेंगे, प्रोटीन बनेंगे. बट ये ट्रू है या फॉल्स है? आप लोग she says central dogma of life में one DNA will not make one RNA or one protein. Why is true? Come on. इतने ब्राइट स्टूडेंट जो है इंडिया के, कैसे जो है से बैठे हुए हैं यहाँ पे? क्या लंच का इफेक्ट है? What is happening? So from the gene, when you are doing, you're making the RNA, what is the process known as? Transcription. So what can also happen? You can have different type of splicing can happen during that time. So same gene may not be making the same RNA. It could make different combination of RNA depending on different splicing and intron exon, right? So now think about complexity. We are looking at in human body we have 20,500 or 300 genes, and each gene can make different type of transcripts. And each transcript now will make ideally one protein. But given that combination of random and alternative splice, you do not know your one gene has made 5 alternative splice form or 10, you don't know. So could be eventually making 10 protein or 5 protein, you don't know. Now each protein, once you make the protein or protein is being synthesized, do you think that protein will be only one protein, or that protein can be changed to some other form as well? What do you think? This group I have not reached here, but it's looking very studious group. Okay. So from that RNA, when the protein is being made, can there more changes be happening, or that will only make one RNA will make one protein?
Changes can happen.
Let's go in the front. Move, let's move. आप लोग बहुत साइलेंट इस साइड में. तो बहुत ही कोजी प्लेस है भाई, यहाँ तो एनीबडी कैन स्लीप, यू नो, कम ऑन। वर्कशॉप कहाँ होता है यहाँ पे? तो ओनली यू कैन लिसन वॉच मूवी, इतना अच्छा थिएटर है। कम ऑन, प्लीज कम इन द फ्रंट, यू नो, बी एक्टिव। आप लोग लैपटॉप लेके नहीं आए, वर्कशॉप करने क्यों आए हो फिर? मतलब व्हाई यू गाइस केम एंड वेस्टेड द टाइम? We told her I prepared 40-page document myself, despite I was so busy in preparing for my APT conference, all right? उसमें इंस्ट्रक्शन लिखा हुआ है साफ साफ, आपको हैंड्स ऑन कराना है, you guys should bring a laptop. उसके बिना आप यहाँ पे क्या देखोगे? मतलब you just see some people are doing something here and there, making some graph. मतलब you guys are all tech savvy ना, मतलब आप लोग इतना तो यूज करते हो कंप्यूटर, मोबाइल। चलो who will answer? कि जो है वो जो हमारे जितने भी from 1G we got many splice form of RNA, and now are we going to make only that many protein, 10 protein from those 10 RNA, or can be more forms?
There can be structural changes.
And in which way that change can happen? What can trigger that change?
External factors.
What additional factors? Or do you think something else can add on those protein to make that protein change something drastically?
Translational changes like acetylation, methyl.
Fantastic, fantastic. So now you can have lot of post-transition modification. Just by adding one sugar moiety, a glucose molecule, protein can change to glycosylated form. Just a phosphate moiety being added to the protein makes it phosphorylated form, right? So same protein has become drastically different now, right? What we are looking at now, same protein but with a very different activity, become more kinase, become much more functional. So now a lot of cell signaling which happens, it happens because the way this small modification happens, which we say the PTMs. So all that is happening at the proteome level. So your same gene has resulted into your few numbers of RNA, and that's in many form of protein. And you, how many you don't know, and we don't know. That's why we are looking at the technologies which can capture the dynamic proteome to as much as possible, right? And then you are not only looking at static information, you are also looking at dynamic molecule changes which may have happened. Eventually all that can help you to understand they are triggering towards which type of pathway, which type of biological signaling is being induced. Okay? So what you are seeing here is the data analysis step. What I was showing you how to acquire the data of the peptides. We took the proteins, not one protein, maybe 10,000 protein. You don't know how many protein you have in the tube. All that being digested, maybe you have 100,000 peptides. Everything is now running in that 45 minutes and you are separating those. After those separation, then now you get some value for every peptide. What is the intensity of that peptide? You are getting that across all the samples. Then simple statistics, right? You are looking at eventually is it passing the p-value? Is it going to pass t-test? Is it significant, not significant? If it is significant, is it following a pattern? That's what we are going to walk you through now. Alright, Avinash, continue.
So, sorry for the
as many as you have laptop, please open the laptops and do something at least, no? Can they do on their mobile? They can't, right? They can't upload the files.
Okay, so I think they can do it in a mobile as well.
Really?
Yes, they can.
We'll try.
Okay, so we have shared the new file with the name of data.original. So sorry for the previous glitch.
Okay, how can, that was pretty good actually, good to have some time glitch. It brought everybody at least in the same forum, you know. So, right. So I teach all the first year undergrad, you know, these are 350, 400 students like them. Keeping them all engaged is not easy, but this is how we do. So can we get all of them to share the file? Can all of them access this file? And even they can try the mobile. I think it's watching the show will not help, right? It's not a movie which you're watching. You will do something that you will learn. So if anyway some of their class representative you can share, so then they can have access.
So now we will go back to the metaboanalyst. And I hope everyone got the file.
All of you will do, but nothing else other than this data analysis, okay? Because questions will come. Because
so now we will start again. Yeah, okay. So now we will start from the starting. So first we will select the peak intensities. As I mentioned, we have the peak intensities, our samples in the column and unpaired, and then we will choose the file. So the name of the file is the data original that we have shared. Now we will just click on open and then we will submit. So
at least everybody can reach the metaboanalyst link, metaboanalyst.ca. What is that link he's showing?
So how many of you reached to this step?
Just search in the Google Metabo analyst.
Okay,
You will find
great.
out and click the link. That's right. Yeah.
So here, what you are saying basically that we have selected the data file and
Go
we
back to
Yes.
previous page so that they can orient and then click.
We will again go to the data upload file. Okay. Now we will select the peak intensities, and here in the drop down, the way we have our arranged our data. So our samples in a column and they are unpaired. So we will select them and then we will choose a file.
So Avinash, just I'll pause here.
Yeah.
So from the chromatography, when we are separating them, followed by running in the mass spec acquiring data, all we are generating at which retention time, what is the intensity of each of those peptides. So what you are obtaining the values, some intensity values, right? And then based on that, you are going to try find some pattern. It was very simple and very interesting puzzle. You have a lot of values. Is this making any pattern which is significant across large number of samples? Right? That's what we are going to do that. It's very simple because you are doing 3 control, 3 treatment. In your actual patient scenario, if you think about it, it's not sufficient number. You have to do maybe 300 or 3000 control, 3000 disease samples, right? So the data size will be much more.
Okay. So we will choose the file. Please select the file. Yeah.
Everybody is on the same page, reach mobile at least, on the Metabo analyst page. By the way, after this session, we will share you this file. Anyways, you will do that.
Yes.
And we will share you some assignment. Okay? Those who can do assignment and will submit assignment, we will only sign the certificate for them and share the certificate of this workshop. Others can, of course, welcome to watch and see what we are doing. But I don't think I can be comfortable signing that you have done workshop if you have not even analyzed any file on your system, right? So that will be unfair.
Internet is quite slow. You can try my. Okay. Now, what you are saying here after submitting your samples in the first step, here you will give you all the information what you have given. So this file have the 12 samples and there are 2 tubes and there is almost 6% of missing value. So this is just give the brief preview of your what sample type you have submitted, and if you wanted to, like in this case you wanted to again cross check that your data annotation or group annotation is correct or not, you can just click on the edit groups.
Avinash, I would go first round slightly faster.
Okay.
And reach till some output, then I'll come back
Great,
and do
great.
it slowly. Yeah?
Okay, so this is just the quality check and then we will to proceed.
I just want to walk through a little faster first step so that you know where we are leading you to and what output you will see.
Yes.
Then again we'll come back and then go slowly so that everybody can see it.
Yeah. The next step is missing value exclusion and imputation. So the missing value is basically when we are acquiring the data, we found that many proteins they do not have the intensity. So as you have seen in our input file, there are many blank spaces. So we have to remove the blank spaces.
Don't go in detail, right now just
Okay. Okay. Now we will select on the enable and then click on the 30%.
So many of these criteria are very important. We'll explain sometime what this means. Why we are choosing that 30% number or missing value imputation. What method should be done. But I just want you to walk through one round so that you see what we are trying to achieve.
Yeah. So we will remove the missing values and then we will also check the use group with the threshold. Just go up. The top right side. The group check. Group threshold check. Okay, so we will remove in the both groups. So basically what we are trying to find, we wanted to keep those proteins and that have almost 70% of data. So we will select that and then after that we will click on the submit. After submission, basically in this step what we have done, so some protein, they are present in only, suppose they are present in 70%, 100%, so they could be present in the number of samples. So we kept some criteria for each group wise, at least that protein is present in the 70% of that group. So that's why we kept this filter criteria. And now after that, is there a network issue? Okay. So I hope, so after that you must have get the data of the missing value imputation.
Again, some of
Okay,
you are not
yeah.
able to catch up. Please raise
Submit,
hand. We can
submit,
help
submit.
you to reach to the right link.
Okay. Now, yeah, it's taking a bit, it will take some time. So when you have the larger file, it usually takes some time, and also there's a network is quite slow. That's why it's taking too much of time. So the overall criteria in this step is basically we wanted to remove the files that have less than 70% of data. Then the next step is just proceed. Okay, and then the next step is basically now we have selected those files that have 70%, but still the 30% data is missing. So that's why we need the imputation. So now we will impute all the files. The reason behind imputation is that suppose in your out of 10 samples, your 7 samples have the value, but 3 have the 0 value. So in that value, if you go for the analysis, it could create a biasness. So that's why we need to impute some value there. Now the question is that how to impute the value from the blank area. So there is a different algorithm. So one of the common algorithm which that we use is the KNN. So KNN is basically it's take the average of the near value from the same group and you put the value there. So basically all the value and all the cells get filled. Okay. Now the next step will be there is a network issue. Sorry for that. Yeah, so this is the file, and now in this step we will talk about the imputation. So just go down and we will select on the multivariate statistical method, and here we will select the feature wise. So there is a two option, one is feature wise, sample wise. So we will select the feature wise, and the feature basically it is showing the particular gene, and if we select the feature wise, it will take the value of the same gene in the different samples. So that's why we will select the feature wise. If we select the sample wise, it will take the value of other proteins for the imputation. So but the best way, we wanted to put the value of the same protein from the same group. So that's why we have selected the last one, the first one, feature wise, and then we will submit and the proceed. Yeah, now proceed. Okay, now we have done the filtration, imputation. Now we will move to the log transformation and normalization. So now here we will select the median normalization and log 2 transformation. That's all. So now doing of median normalization is that we wanted to keep all the all the data or samples in the same scale. So now we will after that we will collect on the normalize. Please click on normalize. Okay. So I think it's already normalized. So now you can see this is the Gaussian distribution what we got for the overall data. So this is one of the data, it's showing the how our data is median normalized and how the distribution of our data. Now we will move to the next step. Just proceed.
So don't worry, we'll come back to each step and explain you. I'm just trying to give you a quick glimpse because the internet is very slow.
Yeah.
So that you know what all is going to come.
So now we will go to, so I'm not going to all statistical thing, I'm just going to the main file that we needed for our purpose. So that will be the volcano plot. So now we wanted to find those proteins, those are differentially expressed. Differentially expressed, that means their value is different in the two groups. So we will try to find that. So now that's why we will go for the volcano plot. So just click on the volcano plot.
TAs, in the meantime, TAs, Sai, can you just run around here and help any student if they need some help?
Okay.
And Vishal, also, please do that. Avinash, you keep
Yes,
going.
yes, yeah. So in the volcano plot, if you have a better network, you can just select the log2 FC2. There is a fold change drop down, just select the 2. In the p-value, yes. So in here you can select the fold change. So fold change, that means how much difference between the two groups. So what the fold change, how the expression has been changed. So in, how much fold has been changed. So we will select the 2 and then we will select the comparison criteria. So if you wanted to disease versus control or control versus disease, that kind of to select. So here we will select the disease versus control. So here our disease is basically the hippocampus samples. And here we will select the p-value as a 0.05 as a raw p-value. Now we will just click on the submit. So basically for the getting the significant protein, those are actually differentially expressed. We will select the fold change of 2. If you convert the fold change 2 into log2 FC, that will be log2 FC1. So here we have selected the fold change 2 and p-value is 0.05. So this all protein are differentially expressed proteins. So any of these proteins could be your marker, and so basically it is showing the differential expression in your two groups. So Here you can see in the drop down, so if you see it is we have got the few significant down-regulated protein and few up-regulated protein. So here this is just test data. So here we are getting just 2 down-regulated protein and 8 up-regulated protein. So this number is quite low here because this is just a sample data sets. So in the same way, according to your biological condition, you will get the number of protein that are differentially expressed. So we will just click on the table and we can also download that. So just so how to download that? Yeah. Okay. So there is a table, left one. Yes. So there is a table that will give you the information of all the differentially expressed protein. So these are the basically protein that have the different expression in your groups. So we will select that, and after that we will download that and just open that. So now we have got our differentially expressed proteins. So this protein could be your marker, or if they could play a very significant role in your data. Yes. Yes. Go to the static analysis. So, so this is basically what we try to find. But before going to this step, we do the multiple group. So basically, we check for the. Just go to the statistics. Go to the statistics. Just click here. Yeah, up. Statistics, upper one. Yes. So here you can see that there's a different kind of setups and those things are there. So basically we also try to find are there a difference between two groups or not. So that's why we use a different kind of plot, correlations and the heat map to check actually there is an actual difference or not. Like in this case, this has a very significant, very less significant changes in the samples. So now we will go to the PCA, just click on the PCA. Yeah. So PCA basically is a principal component analysis. They try to classify the two group on the base unsupervised way. So it will give you the information and how your data is actually different from each other. So it will give you the percentage of reduce the dimension, and it will give you the idea of how your data has been segregated. So it's taking some time. Yeah, the same way there is a multiple thing like the correlation heat map. So then correlation heat map you can get the how these proteins are correlated to each other. So we have to check the correlation because so those correlation PCA, these are the quality check parameters. So suppose your sample has been collected from the same cell type or tissue type from the same sample. So ideally they are similar to each other. And if you suppose, and they will have the similarity of 0.7 to 1. So this parameter we take to check the actually how the data has been acquired. So if you will correlate the brain cells with your pancreatic cells, obviously their similarity will be not less than, it will be greater than 7%. So overall, we just wanted to check the, yeah, just go to the 2D plot. Yeah, so here you can see that as I mentioned, so our data is not very much correlated in this case. So in the same way, we have the two group which is control and MHE. So here also you can see. Yes, sir.
I think some students from IIT Indore, they don't have the file. They are still sitting and they are hesitant to even ask.
Okay.
Dr. Manju, आपकी कोई student उनको just share कर दे file. All the IIT students are sitting there.
So please share the files with them.
One of you can just go and get the file. आपको चाहिए file? So please go and ask. Guys, don't hesitate. And as I said, if you do something, you will find so easy. These things can be done. बहुत simple Excel पे play करना है and just keep clicking. To try out the file access नहीं है, please ask us and try to access the file.
So actually this software is very easy to use. Just 4 to 5 clicks and you will get all the data. And after that you use your mind and you can just check how the data quality, and then you can get the differentially expressed proteins. So
keep going, I'll show the different plots.
Okay, now the same way we will go to the other plots. One of the plot is the correlation plot. Okay, so this is the other plot of the same kind of thing, which is a PLST. Here we are using the supervised clustering. So we are so here we wanted to just to check how the two samples are different to each other in the supervised manner. So it will give the similar kind of plot, and also there is another one is a correlation plot. We can go to that one as well. So yeah, so just go to the 2D plot. So here you can see it when we are doing the supervised clustering. So that what we are saying, basically giving the algorithm, just consider two different group and then try to differentiate them. So it will give the 2D plot. Now if you go to just show the 3D plot.
So stop here. No, show 3D.
Yeah, just show the 3D view. 3D, synchronized 3D plot. Yes.
So again, students, like
Yes.
what you want to see, your healthy individual versus disease individual, they have something different, right? And can you find out some protein signature based on which you can differentiate them and put them into different clusters? But what you'll find always is that not everybody is able to fall into the same clusters. And that's where you will see sometime we have done some work on the meningioma where we could see that we are looking at low and high grade. But within high grade itself, there were 3 subtypes. So you will see that those cohorts are pairing in different principal components, and that is helping us to tell that okay, there are multiple subtypes. Can you move around and then explain 3D?
Yes. So now if you wanted to check actually which sample is not clustering to the other sample, so you can just drag them and you can check where those samples, and you can also hover your cursor, you will see the which sample is that. So just drag them, just drag and show the different view. Yes. So in this way, in the 3D way, you can actually check which sample is actually correlating with your clusters, from the control cluster and disease cluster. So it could be a biological, technical, anything. It could be also possible that from the same group they have the different kind of like comorbidity and those kind of things, the different problems they have. So, and also it could be also possible this sample has been not acquired properly during the sample preparation, other things. So in this way we can get the idea how the data is actually clustering with each other and which are not clustering. So if you find any samples that is not that is showing a very far from their cluster, and the same have been observed in the different kind of plot. So we have checked the PCA, PSTA, now we will check the another plot which is a correlation. So just go to the correlation plot. Yes. So here it will give, now here we will just check the heat in the same thing in the heat map way, and yeah, and after that if we found that the correlation of those samples is less than 0.7%, then it could be possible that there is some problem with the one of the samples. So this filter criteria we just use to check the clustering and also to find the outlier samples. So it could be possible that there are many samples, they are not correlating and there is technical issues, or sometimes also there is a biological issues as well. So we can find in there that way. So it's taking with time. So in the same way you can say there is a multiple plot and then multiple algorithm to find and understand your data. Yes.
So see, like once you have done some basic filtering, then you have just way to plot and visualize the data, right? What we are looking at now, visualization of data in different ways. In which way you can look at the heat map. For example, each protein-wise expression change, is that making any kind of cluster or pattern? You can of course look at principal component, you can look at PLSDA, you can look at showing you the correlation plot. So once you have done the basic steps of data, these are just different with the visualization. But again, these softwares have also some machine learning based setup where you can pick up the best or most important features which are best for classifying the control versus disease. So there is some VIP score here which is using some machine learning algorithm to give you top 20, top 10 or top 25 which are the best one to classify that.
What is the VIP? So there is a network issue that was taking a longer time.
But all the left pan, what you see, can you zoom in a little bit on the left side? Is it possible to
just zoom on the left side?
Just yeah, correct.
Yes.
So please keep it like much bigger like this because in the back.
Yeah. So from the starting, you can see there is a different kind of plot. So first plot is basically the first statistical thing is the fold change. So it will give you the how fold difference from your expression of the particular protein. Yeah. So the fold change, we will get the idea of how the differential expressed or expression of the particular protein in the different groups. So it will give you the idea of all the protein and their fold change when we are on the basis of your comparison type. Now the next step is basically the t-test. So basically t-test help in the find. So basically it's give us the confidence that the protein that we have got is actually they are not coming by chance. So that's why we are keeping the value of 0.05. So only there is a 5% chance that it will come. The sample protein has been selected by chance. So this is just to reject the null hypothesis. Now the next thing is a volcano plot. So as I mentioned, the volcano plot to get the differentially expressed protein. Now the another is a ANOVA. So when you have, so in this case we are using the differential analysis for the two group. Now if you select the ANOVA where we have the multiple group, in that case we can use the ANOVA to find the difference in the different groups. In the same with the DCFB and the pattern hunt, so basically it gives, so correlation is the important one. Now we will just click on that correlation. In the same way, just go to the correlation plots. Okay, it's going on. So in the same way, the correlation, PCA, PLSG, SPLG, also PLSG. So these are the different algorithms for the clustering of your samples. And the few are the dendrogram and heat map. So dendrogram basically they classify your group according to their similarity.
I think it's good to show dendrogram if we can.
So it's taking time. So yeah, dendrogram will be open easily. Just open that.
So you want to see like is there any kind of cluster appearing, and sometime when you actually tell them these are 3 control and 3 disease, you're already biased, right? So that says like you're doing supervised clustering. But ideally you should do unsupervised, you should not tell. And let's say, is it able to form 2 trees? And I look at, tell you all these 3 control, 3 disease are different. Or it may happen that some control will fall into disease, right? And that's actually usually happening in reality. And also some of the, within the disease group, you will find some are at the maybe low grade and they will form a cluster. So you will see this kind of dendrogram pattern. Is that your Wi-Fi here or your mobile data? Why is it nothing here? Is everybody having a problem with the network? So that's another feedback which we will request here. If we want to do a workshop, we should do in a small room, not in the big auditorium. Because, and Wi-Fi will not be effective in that way. 25-30 people who are actually keen, we can do more intense thing with everybody laptop. Laptop should have mandatory. Allow नहीं करना चाहिए किसी को. You're doing a workshop computational analysis, and I can just give you 1 hour lecture and I go. So that will not help because बहुत से lecture already देखते रहते हैं. So use this opportunity to do some analysis and get comfortable. What we are trying to do cannot make you expert, right? But all we are trying to break the barrier. You can do it. अगर आप analyze करोगे, तो you will see कि all this is very easy. Excuse me.
Refresh. So I hope if you have a better network, you can open that. So anyone is the network is working very well? No? Okay. So yeah, so this software takes some time to load all the files. So this is a network issue we are not able to open. But ultimately, the final goal is.
Do you have any assignment file from which we have the reports, which you can show?
That. So sometime if we taking longer time, you can just close all the files and you can just open again.
Okay, now while they're doing it, let's talk about basics again. So one thing which we were talking in the beginning, you have data coming from the instrument data, and you are analyzing for different patient and different healthy individuals, supposed to be healthy individuals, and you are thinking about 10,000 protein you have in tissue sample, right? Let's say you're looking for the cancer type, 10,000 protein estimate, and you're going to analyze them across large number of control and disease samples. So your goal will be for each peptide, for every protein, you should obtain some value, some intensity, right? अभी आगे क्वेश्चन हम पूछ रहे हैं वहाँ पे क्या बात हो रहा है? All right, this group, what I'm talking? बताओ, repeat करो last sentence what I was talking. अगर यहाँ तक आवाज नहीं आ रही मेरी तो पीछे आगे चलो ना, चलो let's go. You can't hear me properly, तो चलो आगे बैठो. क्यों पीछे बैठे हो? Just आगे, those who are not able to hear, go in the front. आप लोग clear थे क्या बात कर रहे थे? We are talking that you are trying to acquire data for every peptide, right? From the instrument. But practically it is not possible to obtain value side में. No possible. Vishal, take a pause. So in the basics of what we are talking, that your goal is, in ideal situation, for every protein and every peptide what you have, you should obtain some intensity value. But in reality that does not happen. Because somewhere the ionization gets affected or the instrument is not able to capture that value. For few peptides, you will see in control you have some value, you don't have value. In some disease you have value, you don't have value. So then, ideally you have missed out some actual data acquisition from the instrument, and that is a missing value, which is actually an artifact, right? So you need to decide, are you going to keep that data or going to remove the data? So then people use the filtering criteria, that up to what extent I can use the data. Even if I had, let's say, only 7 data points out of point 10, can I keep the data? And if I have maybe less than 5, then I'll throw the data, right? So that is the filtering criteria which we use. What is the missing value? Anybody here? Missing value, क्या होता है? हाँ? नहीं idea हम वही तो बता रहे हैं. इसमें idea कहाँ होना चाहिए? I'm not asking you something theoretical. I'm just telling you that instrument is not able to acquire data to some place. That's a missing value. ठीक है? So we have to decide कि उसको रखना है या उसको remove करना है. So sometimes we make some threshold कट ऑफ कि अगर 70% डेटा पॉइंट है हमारे पास तो हम यूज़ कर लेंगे, उससे क्या बात नहीं यूज़ करेंगे, राइट? सो इन दिस डायरेक्शन देन यू गो एंड से कि 30% जो डेटा नहीं है जहाँ पे, कैन आई यूज सम ऑफ माई कंप्यूटेशनल स्किल के नियरेस्ट नेबर एंड यूज दैट टू पुट दैट इन पॉइंट वैल्यू पॉइंट इन दो एक्सेल. आइडिया इज नॉट करेक्ट आइदर बिकॉज इट इज टू हाई द मिसिंग वैल्यू. इससे आपको जो फर्स्ट स्टेप होता है, यू विल लुक एट दैट कि कैसे मिसिंग वैल्यू हम इंक्लूड करेंगे या नहीं करेंगे. ठीक है? व्हाट इज मिसिंग वैल्यू इंप्यूटेशन? दिस साइड यू गाइस आर वेरी सीरियस. मतलब ये तो खाली इंग्लिश में हिंदी बता रहा है कि मिसिंग वैल्यू उसको इंप्यूट करना है, बट कहाँ से आ रहा है मिसिंग वैल्यू? You are not able to acquire that data from mass spec for every peptide, right? Because of some technical issue, sometime could develop also sometime. So that's why we are doing filtering of those. What you are seeing now is a correlation plot. You are looking at how different samples, the data what you acquired, are they correlated or not correlated. If there is one sample which is odd, आप एनालाइज कर रहे हैं सारे कैंसर पेशेंट सैम्पल्स, उसमें एक पेशेंट ऐसा है जिसको डायबिटीज़ भी है, ठीक है, उसको हाइपरटेंशन भी है, इट्स वेरी डिफरेंट एंड हैज मेनी थिंग चेंज, तो वो आउटलायर हो गया आपका. If you are going to use the same cohort, it's not even the same. So your this data when you are looking at it, जैसे एक blue वाला दिख रहा है आपको ना, it's looking like is there something coming odd. So you are going to look at when you have large number of samples, कि उसमें कितना correlation है, कितना correlation नहीं है. And based on that, अगर 1 or 2 are outlier, you will go back and look at the clinical data. Again, why we are teaching you guys? Because you can go back and look at what that patient was. उसमें कुछ peculiar था, कुछ different था कि वो same cohort का था. When we looked at meningioma, high-grade patient उसमें subtype और different थे, we sent back those detailed Tata Memorial and they looked at each patient history and found out कि वो subgroup में problem था, different था. So this is where you have to think about कि when you see outliers, don't throw out the data, go back and look at clinical metadata and see is it making sense or not. ठीक है? So now show some of the pattern if you can show. Or you want to talk on this one?
So I think now we can switch to the pathway analysis and those things. So what we are saying here, so basically this is the correlation plot as sir mentioned. So this they just try to correlate all the samples. So as you have seen,
can we also see the live run? Is that
okay? Live run.
In mass spec also, can you WhatsApp me guys? Okay. अच्छा ये तो हो रहा है, चलो good. So what you see right now is the live run is still continuing, है ना, जो 45 मिनट gradient का था, almost finish हो रहा है. So it's very simple. You are doing the workshop, you are also generating the data, and sometimes we have given the live data to the participants so that they can analyze the data we are acquiring right away, right? So instrument part, what you think is a proteomics and you feel afraid कि हम नहीं कर सकते हैं, that is not true. It is very simple. That happens elsewhere. What actually happens at your end is the experimental design. क्या experiment है? What we really is a question which we want to address. और उस experimental design के लिए हमारे पास क्या control होने चाहिए? Again, why we are spending time with you guys? because you are the one who will think the best way कि ये disease अगर है, इसका ये control है, healthy versus disease is not a good comparison. Disease versus disease is a good comparison. If you are comparing somebody having fever coming from malaria and you compare that fever with the dengue, compare that with Zika or look at COVID-19 or look at typhoid, look at all the different type of causative agent which provide same type of febrile symptoms, right? Then you are looking at underlying mechanism same, acute phase response will be same. Then what is different is unique coming from the pathogen. Your goal was to look at pathogen-specific or infection-specific response, not the body's own defense system changing in response to any kind of infection. So, an ideal experiment will be a disease comparison with another similar type of disease. Okay? And this will come with your insight. When you become more into your medical career, you will have a better idea for this disease what should be my better control. Sometimes for a specific disease, we go to another disease type and use that as a control, as a disease control. If you look at healthy and disease, you will have day and night difference anyway. You will feel very happy with बहुत अच्छा identify कर लिया, but it does not mean anything. So, what we are trying to convey you, good experimental design is the first important part. Second is looking at the statistics. What is the power of the stats? What kind of number of samples which you need to analyze minimum to obtain any kind of actual meaningful data from that. That's known as power analysis. Right? What type of negative control you should have. And of course, a lot of clinical information which you will obtain for all the sample will be important. So first thing is experimental design. Good clinical experimental design is not straightforward and will come with you guys because you are only day and night doing that. So that's why we want to teach you these things so that you can do better. Okay? So if you have done good experimental design, you can get those samples, even you are very busy, it's very technical, very simple, one could prepare the samples or send a sample to us and we can do the running this instrument and provide the data. Data generation, one day we can run with this kind of gradient, we can run at least 24 samples with this gradient. But when we run this 5-minute gradient, we can go 60 SPD, 60 samples per day. Now they are coming with more shorter gradient which one could do even 200 samples per day. So acquiring data will not be a big problem, right? And of course we look at many things at the technical end, is it good data, bad data situation or not, but that's not within your purview. After that, if you are comfortable in looking at data and you can analyze the data or at least interpret the data, then that's what we are going to look for again, that are you finding something peculiar coming out? And if you see an outlier, you will go back and look at it and make some clinical sense. The best projects in my lab which have happened, there is some of the MD students who have been in different medical schools who have come for the part of MD biochemistry projects. They collaborated with our PhD students, came to IIT Bombay. Our students are helping in the proteomics and multi-omics, and they were providing the clinical and metadata information. Together, mindset was able to come back and look at much more important clinical information from the same data, which nobody else could have done together, like separately. Right? So that's why I think it's important for us to talk same language and at least give you some idea that how to analyze the data. So this is you see data acquisition is very simple. You don't have to worry about the instrument and how to acquire data. Those are all very straightforward way of reading, and one could acquire that. You have to just send these things to us. All right, let's come back to the analysis part.
Yes. So this was the correlation plot, and now we will go back to the after the
heat map.
Okay. So now we will show you the heat map. So basically it will give you the protein list that are differentially expressed. So here you can visually see which protein are actually they have the different expression in your what biological comparison you have. So yeah, so just click on yes. So here we are just setting the few parameters, and after that we will plot them. Just go down. So this is the top 50 proteins.
So stop. Yeah. All right. Now please take an observation and I'll ask you your interpretation. Okay. So देखो result. Very simple. Please don't keep audio on here if you are not doing the analysis. ठीक है. What do you guys think? इस पे क्या आपको observe हो रहा है? Just वो बहुत simple है.
Make it large. Yes. Fine.
So red and blue. Yes, sir.
But is that all uniform?
No.
It's not, right?
Yeah.
At least we made some observation. What you see, ideally, and that ideally should not be ideal, because in the biological situation you will have differences. If you see everything is same red and everything else is all blue, it means that can't be human, right? So only when you see experimental model, when you take some very tight cell culture based studies or something very tight experimental model, and you are giving a drug treatment or inducing something, then that can be much more day and night. But human sample, biological clinical sample will always have variability, and within that can we find some pattern which is much more pronounced. So now let's keep moving from the top to bottom slowly.
Yeah, this good.
Right. So many places what you see is still we can see some of the white bricks, right? White missing data. So there was no pattern observed because there was no data point there. That's why we've been talking about missing value. And then you have many places very intense or light to dark blue, or you have some of the the reddish color. So you are looking at differential expression of the genes and make the proteins made out of them. But sometimes patterns are not very clear and good because there are some missing values. Everything is not able to obtain the complete data point. Now one of the patients in the center you see very intense red, right? Can you go till up? Go up. वो बहुत डिफरेंट है ना, उसमें बहुत चीज़ें डिफरेंट हैं. Lot of missing things are there, बहुत सारे रेड हैं, very intense. Come down, even नीचे blue भी था उसमें, same patient में. Keep coming down. Right? तो ये peculiar है, थोड़ा different है. It will be interesting subject to see कि इसमें क्या हुआ, मतलब why this is behaving like this. Some of the patients, like I think if you go from the top right, first three काफी uniform लग रहे हैं. उनका pattern जो भी है, that is more uniform than others. So this kind of heat maps just gives you visual feel of how the protein expression is happening across my samples. But of course, this is not done analysis, not done perfect way. I had just walked through very quickly. But ideally, we should have fixed the missing value part to begin with. Filtering करके उनको निकाल देते हैं. जो भी हमारे पास जो missing value लेते हैं, उसको remove कर देंगे. And then when we are selectively doing, then उसके बाद जो normalization करते हैं, then what we look at all the data for the thousands of protein to several thousands of peptide, सबको normalize कर रहे हैं, so that they can come on the same scale, right? उसके बाद then we are going to look at the differential expression. Now, actual real science, the challenge comes. Think about COVID-19 time. Patients were coming, and technicians are not going to keep the sample in the proper way, right? वो patient को देखना जल्दी उनको, they are not going to store sample in the freezer or 4 degrees. कुछ patient का sample उन्होंने 4 degree पे रख दिया, plasma sample. कुछ का room temperature पे छोड़ दिया. For few patients they kept in -20 and some they left in -80. हो सकता है ना? What do you think? Is that going to make any difference? COVID-19 सिचुएशन से बोले कि बहुत हरी था, बहुत रश था, और उस समय जो है टेक्नीशियंस का माइंड सेट विल नॉट बी और डॉक्टर्स विल नॉट बी रिसर्च. बट लेट्स से वी वांट टू डू रिसर्च उन सैंपल्स पे, एंड वी आर लुकिंग एट सम पैटर्न ऑफ द माइल्ड एंड सीवियर पेशेंट्स ऑफ COVID-19. And we are collecting plasma samples. So जो left plasma sample है या जो very soft sample है, just give it to us. But those samples were stored in 4 different conditions. कुछ patient के sample room temperature छोड़ दिया technician ने. Every technician not very sincere. Every student not so sincere here in the same audience. So how you will, all your doctors will be aspirant doctors, how you will expect a technician will be all same. So some will be storing, some are very judicious, they will give -80 if they have access. Some will give -20, some 4 degrees, some room temperature. Do you think that will create any problem, any challenge? Common sense है ना, कुछ होगा problems में, what problem can happen? क्या हो गया? Your time is so precious, I don't have to waste your extra time unnecessarily. You're here, please, you make use of it. बताओ आप लोग तो sincere लग रहे हो, you are following up as well. What do you think?
So definitely the results will be completely different for each of the 4 situations, since the external factors around the sample are different. So even the
Let me ask somebody else. I'll just ask one of you guys in the back. जो उन्होंने बोला सो रिपीट कर दो आप, ठीक है? All right. इतना अच्छा उन्होंने आंसर दिया, आपने सुना भी नहीं। मेरा नहीं समझ समझ में आता है, चलो ठीक है, आई स्पीक डिफरेंट लैंग्वेज, बट कम ऑन आप अच्छा आप लोग ये वर्कशॉप में आए ही क्यों? अगर मतलब कोई इंटरेस्ट नहीं था या सुनना नहीं था तो भाई और इससे सब क्लासेस जाके बैठते कुछ और एनाटॉमी फिजियोलॉजी पढ़ते। व्हाट यू आर टॉकिंग कि experimental design and sample corrections are very important. Even sample correction, what you see here sometimes, differences may not be real, may not be biological. That is just coming कि कुछ patient आपके जो थे, they were all same patients of COVID-19, but some samples were stored in the room temperature, some were stored in 4 degrees refrigerated, some came in -20, some into -80. उतने के नाते ही प्रोटीन इतना डिग्रेड हो गया, चेंज हो गया कि now when you analyze it will give you 4 cohorts, 4 different type of colors of पैटर्न आ जाएगा। आप कहोगे हमने इसमें जो है बहुत नया डिस्कवरी कर लिया है, नो, but that is not real because your storage conditions are very different. So you need to know that when you are doing these experiments, experimental design, experimental conditions, how you are storing the samples, how you are preparing samples are all very important, और वो सब रिफ्लेक्ट होता है फाइनली यहाँ पे आके। इसका कुछ भी बायोलॉजिकल या क्लिनिकल सेंस तभी बनेगा when you have done the good data acquisition from the right type of samples with a good planning. Obtaining 10,000 samples is not a big deal, but obtaining even good 200 samples which are properly collected and stored in the flash frozen condition, well annotated, is actually not easy. So, but that's what you are looking at. ठीक है, otherwise आपको जो है पैटर्न बहुत सारी तरह के नजर आ सकते हैं, that will be of no meaning, no value. So, I don't know like how far you want to go, but what you can do.
I think we can show the pathway. This is part 1. So
एक मिनट, इसमें जो है इनको वो दिखा दो, VIP वाला top.
Okay, just go to the PLST. PLST, yes.
So one thing which is coming forward more as you see, like AI, machine learning, right? But we are trying to find some good pattern. Finally, when you become doctor, on your patient, what would you like to test? अगर आपको टेस्ट करना है किसी डिजीज के बारे में, would you like to test just based on 1-2 protein or based on 100 proteins? क्या प्रेफर करेंगे आप? एक साथ 100 protein टेस्ट प्रेफर करोगे? नहीं, what we are trying to discover is different thing. Discovery में हम 1000 चाहते हैं, लेकिन फाइनली आप इम्प्लीमेंट करते हो पेशेंट पे, आप चाहते हो मिनिमम मेजरमेंट में ही हमको प्रॉब्लम मालूम हो जाए, राइट? जैसे प्रोस्टेट कैंसर के लिए PSA यूज किया जाता है, ऐसे बहुत से डिजीज के लिए आप जो तमाम मार्केट यूज करते हो, all that किसी क्रिएटिनिन insufficient हो जाता है, but some evidence is there, many markers which you like to measure, you would prefer to use less number of protein or analyte to be measured, but that should be robust panel. वो पैनल आपका रोबस्ट होना चाहिए। अभी US FDA में based on the proteomics and biomarker discovery, they have approved many biomarker tests. In that, one of the tests is known as OVA1 test. This OVA1 test can help in the women 40 plus to, if they are having suspected ovarian cancer, whether they should do the surgery or not. And to make this decision, they just do simple plasma sample analysis and look at a panel of 5 protein. They look at the panel of 5 protein to determine that is the patient is high risk or not. Because just think about it, like you have and that's your real situation, right? There's a clinical dilemma always. You get a patient, you have to decide, are you going to do surgery or can you wait? Or your start treatment or should you wait to recover? Who can help with decision making? You can't see the patient. These proteins and biomarkers and molecules, that's what you will measure. So, if you can have robust panel, which we say the classifier and the markers, that's what we all trying to achieve. So this marker test, which is OVA1, you can search in the Google, that is based on 5 protein analyte tests. Based on that, if the score comes very high, then they put in the high risk and they will go for surgery. If that is low, then they will not do surgery. Think about somebody's life and this surgery depends on a simple test. Otherwise, they will do everybody's surgery because everybody is suspected. So this is where the biomarkers and a panel is required. Sometimes one marker may give you wrong answer. But a panel which is maybe 3, 4, 5 protein together will be much more robust. And this is where machine learning is going to be more powerful and going to be more useful for us. Now, even these softwares have to some extent linked to that. It will give you at least very important protein based on analysis. But you can always take this data and export the data and now go to machine learning softwares. One of that is Orange, again freely available. And now you can look at, based on different type of machine learning, what pattern it can predict. From your protein list only, it will tell, okay, if you take protein 1, 5 and 9, you have maybe 95% accuracy. But if you take protein 1, 5, 7, 9, 10, you have 99% accuracy. You understood? So within all of that, you are trying to look for the best panel which can give you highest accuracy. And of course, your negative control and disease control will tell you false positive, false negative, etc. Are you seeing anything there? Okay, you are trying?
So actually it's not getting open, so yeah, it will, now it will. Go to the important feature, yes.
Correct. So now you see it has ranked that among all of your protein what you have analyzed, some proteins gets more high importance, some gets less. So now next thing will be based on your clinical knowledge which you have already acquired. You would like to read your physiology book again, you would like to read literature and say, okay, is this protein making sense? Is this kind of thing is actually relevant? What patterns I am seeing? And the combinations of those is statistically how powerful it is. Once you do that, this is only discovery so far. Then you need to validate. Then you need to go, okay, let's go beyond Ujjain. Let's go to Indore patients as well. Collaborate with 3 more hospitals and then take some samples there and see the same pattern observed. Now you say, hey, I have discovered something big. Bharata Zagapura, I can, and a lot of money now. You will go now Maharashtra also, so to TMH we offer karte hain. And if that works there also, you say now pan India. But it's very difficult, given heterogeneity of population. Same sort of marker, whether it work out in all type of samples is very not easy. But the cohort should be much larger size and from different regions. For us to claim that we have found a test, developed a test, which is generic and having strong biomarkers. Right? I'm sure you will all agree. You don't want to develop a test which only works regional. But if a problem is regional, somewhere some cancer is very high or somebody is very high, then of course that may happen. So this is where I think, your intellectual journey, what you are already achieving, and I had no way of demoralizing you or telling that you are not understanding anything, but just it's a different mindset, different thought process. You are very focused on about only one way of learning. But what you have to move on to open up your learning and see that when you broaden your things, you can give your patient much better way of treatment. Dr. Sevanti Leme, which I think you might have seen or heard her name, she is at Reliance Foundation. She is of course a medical doctor, oncologist, but they are so well read and so much well researched clinician. Right away they do a lot of research-based investigation on their patients. Also connect with a lot of US doctors and researchers and provide the patient the holistic picture. That's why tumor boards are made now to look at and discuss the data for patients. It's not so objective, It's not easy to just tell that this is the magic bullet and you give and somebody will recover. So your understanding and looking at these kind of things can be helpful. And if you can analyze on your own laptop, which you can, you'll see it's not that difficult, just a couple of clicks. We'll also share you some links of, the tutorial we have. Internet here is a problem, but you may have, you have also much better. So you should try to analyze some of these things. And if going forward, you can analyze your own data and you feel that you are able to find some pattern, it might be helpful for you to think about how to use that for your patient care better. Nevertheless, I must tell you, any one of you in this room, if you can do some research and publish some paper, your career profile will be very different. Not that I want to make you scientist, I want you to become a doctor. But even doctors who are able to do some research, they are much more in demand in the worldwide, everywhere. There has been medical students, MD students worked in our lab, have been very good, published paper. One of them became faculty at Harvard, some became in Imperial London, UK. Very, very good positions. All they did just a couple of small projects, but continue doing that. But substantial work, which has gained some new knowledge. So what will make you different is something, you have to go beyond just the classical knowledge and integrate it. And now you have so many gadgets and everybody is so comfortable using mobile and computer and internet, right? So make use of them. Alright, so we have shown you at least a gist of it. Again, the response was not so great here. Neither the computers were here nor we had internet. So this is the only thing which minimal we can do. But now let's say we got the list of the protein from the VIP score or machine learning, whatever we have. You would like to know these proteins belong to which pathway, which biological pathway these proteins work, so that now you can find a drug and you can treat a patient. Alright? Okay, so now let's take this data and do the next pathway analysis.
Yeah. So the previous steps we have shown you we have downloaded the differentially expressed protein from the volcano plot. So I will just show you once again if you have not done that. if your computer is working fine. Otherwise, we will directly go to the file that we have downloaded. So just go to the CSV. The CSV that we have downloaded is the volcano plot. It's fine. Just open that CSV file. Just go to the downloads. Yeah, so we have downloaded a file, right? Volcano, the next one, yes. So from the volcano plot, you can download the list of filter. After the filtering criteria, there is one icon. You can just download the proteins, and now you will have the list of the proteins. So in this case, it is obviously the list number, but still we will show you how we try to identify that they belong to which pathway and how we can understand how they are biologically important to each other. So we will just select the proteins. Just copy, and now we will go to the other software. So there are multiple software, so I will show you one, the very simple one that give you the information about the multiple databases of the proteins. So just go to the Reactome. So in there's a multiple database like the KEGG, Reactome, and the HSA, and the other gene ontology. So this is one of the important database where you can get the very in-depth idea of biological pathways. So it is very easy to use and open software. You just have to go in the Google and type Reactome. It will get opened. And another tab we can also, so just...
so hold on, so I think all of you can at least make this much effort. Go to reactome.org and open that. And if you had any protein list, now you are not looking at a big data sheet, you are looking at maybe 5 protein, right? Because you want to, or 10 protein or 20 protein, which is the most significant. And what you are looking at, these protein belong to which biological pathway, and can I have any drug known in the drug libraries or in the available anywhere for clinical practices, which I can use and do repurposing as well. Right? That was the thing done in the COVID-19 time when we did not know what drug to use. So we are looking at which pathways are actually getting perturbed and use those drugs to control those pathway deterioration. Okay? So based on this, now we are feeding this data to this Reactome pathway software.
Yes.
Likewise, there are many string is there, and you have HPA, human protein atlas, and there are many IPA, ingenuity pathway analysis. Many softwares are there. I'm sure you guys can do much better than me. All of that. Once you know that you can do one, you will export to 10 other things. But hesitation of trying first thing is what we want to break here.
Yeah. So now we are, after opening the Reactome, there is a one icon, the analysis tools. Just click there, you will come to this page. And then just paste the list of the protein that you have. So ideally to get the holistic view, you have to good number of protein, but in this case we have the less number, so just for the demonstration we are showing how the pathway analysis we do. So after just pasting the list of the proteins, just continue and then again analyze. So now it will give you the list of the pathways, top 25 pathways, and all the pathways those are coming from this protein. So you will get this kind of a database. If you want to understand how they are interacting to each other, you can zoom in and you can try to understand which pathway they are coming. And also if you wanted to if you wanted to look which pathway they are coming, you can look visually, and you can also download on the file. Go to the analysis.
So hold on. Yeah. So this is, again, the databases have all the protein information, and people from the biochemistry experiments, they have already established that all these proteins belong to the Recidase pathway or JAK-STAT pathway or any other pathway, right? So based on the occurrence and the significance of this protein, now it is trying to achieve that which biological pathways are most perturbed. And that's what already given you rank. Just took only a few seconds time. It already gave you idea that which are the biological pathways these proteins belong to. Okay? So once you have actually established this, and now you feel, okay, you have found a biological pathway which looks very interesting, and many of my proteins belong to that pathway. Now you can look at various interaction network of these proteins. This protein belong to which other protein interactions. For example, proteins, as you know, they don't work in isolation. Many times proteins form the dimer or heterodimer, right, or tetramer, or they look at more partners to function something differently. So then you look at protein-protein interactions. Again, there are some softwares which can look for those type of possibilities. But this can be at least basic. Go through MetaBoAnalyst, go to Reactome, and at least reach out to significance protein, and then try to plot them to find out which biological pathways they belong to.
Yes. So now it will give you the list of the pathways, so you can download, you can directly go to the analysis and you can click on, after that you can click on the downloads and then this kind of portal will be open. If you click on this, it will give you the PDF file and also the CSV files. So analysis report, it will give you the PDF file, so just click here. Just go back. Yeah, again, click on the analysis report. Yes. So now if you download this file, it will give you the top 25 pathways from which pathway are very significant according to the list we have given. You can also download the different formats and it will give you the overview of top 25 pathways and how they are linked with each other. So while it's getting download, I will show you one more pathway, which is a string that is very useful. So this is the one, only the one pathway which is a reactome. But string is basically, it's giving you the protein-protein interactions and also the different pathways like CAC, reactome, gene ontology and all kind of pathway. So meanwhile, with our file is getting download, we will go to the another tab and we will search the string. Yes, so here in the go to the multiple proteins, so this is the same, just 2 clicks or 2 or 3 clicks you will get your pathway and all the list. So after that you can select, just paste the name of the protein and then select the organism, Homo sapiens. Okay, it is rat, okay, fine. So this was from the rat data, so that's why we will selecting the rat. The same way, if you have the human, you can select the Oliver. Now we will just go on the continue. So, yeah. So this is as, because in this we have the very less number of proteins. Now go to the analysis one. Analysis, click on analysis, yes. So if you go down, okay. So these are very less number, so that's why it's not coming. Just go to the previous one. The PDF that we have downloaded. So overall what I'm trying to say, basically if you select the number, you have the significant number of protein, so then you can get all the pathway information from the string. So string have the basically the result from all the databases. The gene ontology, we have the different kind of information. Some are the biological process, molecular function, and along with other cellular components. Yeah.
Can you show another figure?
Yes. Just go down. So this is the PDF file from the reactome, and it will
stop here. Stop. Yes. So these are many pathways which has come out from this analysis. Click any of those.
Just click on first one.
So see, like this software is already so good. It's able to give you visually that many of the protein from your data, they belong to which pathway, and it is now giving you the information that what is also missing, which is not from your data. Now these figures actually are very much fine for you to publish right away as such. You just have to put in your acknowledgement in the figure legend that this figure and data has been acquired from the reactome, that's all. So now you can make so nice figures from your, even if you're doing a small project, 50-20 samples of each 2 different comparisons, you obtain the data, acquire data, analyze it, make pathways, and you are generating something very substantially unique biologically on your own, and you can publish. Alright?
So as you can see, it is also giving the brief information about what it is, idea, the biological information about that pathway, and how many, and the few reference paper as well, and it will give you the list of the protein that has been matched to this pathway. So this is a very good software where you can give all the holistic view, and according to your hypothesis you can proceed.
Can we show a string as well briefly?
Yes, so just go to the string, select any.
So
this time, this way.
You can see networks of this. All these are open access software. Good thing is you don't have to pay for any of them. They are working on very less RAM. You can actually use your laptop. And even if you are not able to generate your own data, but think about you have obtained some data from already available data set from somebody else, published data set, and you want to reanalyze that. Or you are looking at maybe 3-4 different data sets and you want to make the better sense of that data together. So you download all the data, analyze that file, and come up with this kind of pattern. Eventually, you just need to validate that. So the learning process, whatever you are learning, can be implemented in different ways. You can do project, but even if you don't want to do project, you can always use the information to just take the analysis to the next level. You don't have to generate it all the time. Okay? But if you can do small project with your own population for a given problem of interest, your chances of obtaining some original data sets and then filing for a patent, getting a publication can be very high, and that could be very substantially different. But that may not be goal here, but still that will help you to think about how to you can make more progress eventually. But nevertheless, what the basic thing you will get some idea which you can use to implement in your patient care. If you can get some of these things, you have a known problem of which you are treating for a patient. Now you have reached out to this is the pathway, which is the culprit. And when you go back to your different other diseases, you see, okay, for this pathway, some drug is available. Can I now use that drug and try on this patient, and that might be able to inhibit this protein and work better. So there is a very interesting, actual story or the actual incidence of an individual in Washington. He was suffering from the leukemia, and he was a researcher of genomics. He wanted to do his own genome sequencing because he was comfortable doing that. But in US till that time, there was no insurance scheme available to do the sequencing of the individuals. It was not paid by the health insurance. In India, again, every test we pay for it ourselves is very different, straightforward. In US, everything is paid by the health insurance. It was not covered, but university allowed because the patient is at the terminal stage, and he's a researcher, scientist himself. So Lucas Wartman is his name. So this individual was allowed to sequence his genome and look at his proteins, and all he analyzed, and they found some very unique changes, which was reflecting that maybe a drug sunitinib, which is available in literature, which can inhibit that kinase, could be used. Now sunitinib is used for renal cancer cure, not done nowhere for the leukemia. So you cannot just go with the disease name and the known drug name. You have to look at what is getting changed and what is actually going to specifically treat that. In this case, as soon as they did the sunitinib treatment, he totally recovered. He's an assistant professor and reached out somewhere. I feel very happy and impressed, and see, this is your knowledge can at least give you life. And your knowledge can give you 100 people life, if you are knowledgeable. That's why we are putting our effort to make it knowledgeable. Okay? So, if you look at more specific, everybody who comes for leukemia, you will give the same ABC drug, they may not respond, and you will let them go to, do whatever they want to do and follow chemotherapy. That's not the case. Then your intellect and looking at omics and multi-omics can give you much more unique information. You can try something new, and you never know that is something which is going to totally, respond to the patient. He had the recurrent leukemia, so he has 2-3 rounds already. He was almost going to die. But this thing has totally, recovered him, and something which was never heard of. And then n number of, interesting examples like this. This is what we are saying. We are moving towards personalized medicine, which you all hear the term. But this is why it is important. You look at one individual's multi-omics data profile, look at the data in totality, look at what is changing, and then use your judgment, go back and look at literature, what is the drug repurposing can be used. You do not try something totally new, but at least what is it? Approved drugs, within that something can be tried. And that's what I think is what this kind of a small training can help you to take that decision forward, right? Again, this is the software stream.
Yeah. So as you see, this is the interaction network, so it is showing how the protein are interacted. If you go down, you will also find, as I mentioning, you can link, you can get the information of different biological process and different databases. So as you can see, it is giving information about the biological process, and if you select any thing, it will highlight it according to that selection. So it will give you the how this pathway and how many proteins are linked to the particular pathway. So it is very good software to get the idea from the different kind of databases and how they are correlating to each other. In the same way, if you go down, just go down. Yeah, you will find that there's the biological process. So this is all the list of the protein that we are getting. Yeah. So here you can see this from the same ontology. We can look for how the molecular functions are there, cellular components are there. Just go down the same way. How many the reference publication are linked from this proteins.
So hold on. Yeah. Make something bigger and then only show that.
Just zoom it.
Too fast and too faint for people to follow.
Okay.
So zoom in, and just only then show one.
Yeah. So as I mentioned, so different databases, the same, the gene ontology, molecular function, cellular components, and how many reference publications are belong from these pathways and these proteins. So it will give you the information of all the proteins and how many clusters are there according to what number of proteins you have selected. The KEGG pathways, it will give you all the metabolic related pathway informations. Just go down, and also it will give you the reactome databases related information here and the wiki pathways. So these are the different kind of databases. Yeah.
So again, like, you got some clues from your data. Now you are trying to generate associated information, which these softwares can try to give you. This may be relevant, may not be relevant. That's when you need to use the literature and your reading and judgment to decide, because they are giving you multiple possibilities here, right, from your data set. And that's why we always do validation. Whenever we generate this kind of data, omics data, which is large data set, that's not always going to be very conclusive in the first round. You have done analysis, you have found some pattern, but that has to be validated. So you go to independent set of cohort of samples and you do validation. As I was saying, you are very happy that in Ujjain population you found some solution and you develop a test. Is that test going to work on the Indore at least? Or is that going to work in Bhopal? If not in Maharashtra, can it go even pan-India? Then you have at least something substantial. But if you say it is a global biomarker, it should work in US, it should work in Japan, probably everywhere, right? Which is very difficult. That's why not unified biomarkers we don't have for every disease. So that's the need for looking at more population-specific biomarkers, more region-specific biomarkers for different diseases. And eventually, when the technologies will become much more accessible and much more cost-effective, more personalized omics profile. Now you have been hearing like at least the terms like digital twin, which I also alluded. Idea is like, now each individual, each one of us, have our own genome profile, proteome profile, metabolome profile, lipidome profile, all omes are done, and you make a cartoon 3D model which is having all the data. So your own digital twin, your own replica, which is having all these clouds of the big data. And now, if you are acquiring that data longitudinally, let's say after 3 months, 6 months, or 1 year, you are then trying to look at the fluctuations of these networks, what is getting changed. Earlier, our ability to analyze this character was not there, but now you see our ability is totally changed. We are looking at much humongous data sets, and again with AI tools, you can do things much faster pace. So these are going to be reality, and going to be future, where you are not just going to look at few measurements from the patients and just decide the right medicine. And if you keep doing it, you will be very much isolated. You will be very much in the back line. Where things are going to more progress, patients will be looking at much more personalized treatment. And of course, that's already started moving, in many of the big labs in the other places. So just to take this knowledge in the beginning, what we want to do, not to make you expert, because a lot of other experts are doing this work, but at least to motivate you that in which way you can use this information to take some of the actual patient problem which you will be handling. And if you are trained, then I think you can do better justice to it. At IIT Bombay, to all our first year undergrad, who are all engineering students who come from the competitive JEE exam, all of them take bio as a core course. Can you believe it? They take physics, chemistry, maths and mandatory bio. Very disappointing, right? Very difficult for many of them. But it is mandatory course. Why? Because biology is the future. And IIT Bombay, and you go to Stanford, MIT, all of these top institutes, they see the value. So what we want to do, we want to develop the interdisciplinary cross-pollination, the ideas and the discussion and thought process. And we don't want to make all the IIT Bombay engineers as biologists. That's not the goal. And we don't want to make all the R.D.GARDI medical students engineers or the technologists. But the idea is, can you actually speak some of their language and can they speak your language? Then they can do better to do your analysis and you can do better to provide them the right samples, provide them the right guidelines. Okay? If I collaborate with you guys when you become doctor and you are providing a sample, as I said, you are giving 1,000 samples stored in all different type of haphazard manner, and you do not have the metadata, will be of no use. Right? So you need to talk same language. What we will do, I request this session, I would say, was very unsatisfactory because of we were not equipped together with the right infrastructure and all of you are not well prepared. But this could be at least a good beginning, right? There is never too late. Until I see towards the latter part we all become more serious. So what I request Professor Manju that if you can make a WhatsApp group of all the participants who will not take this as a step A, will not take much time. We will again give you one more online session and give you data. You can do at your comfort with your home and your hostel and analyze the data file which will randomize and give you some new files. Two of you will not get same file. Even our courses when we teach, we give to everybody individual files, and then you provide and submit a report. No report is wrong, no approach is wrong. Alright, you just have to have some rationale. When I am saying I am using missing value imputation 70% or 30%, I have some rationale. If I can justify it, I am fine. Otherwise, somebody can say, no, 80% is also not correct, and 100% is only correct, if I remove that. So you need to understand those values and use that with the proper ways of your justification. So we will consider this as an incomplete workshop, only preempting you to think about big data, which is not very easy. But what is very easy, which we showed you live, instrumentation part. We have showed you multiple mass spec, live run. Data acquisition is not very difficult, which you thought you can do it. So that we have shown you 4 different mass spec live parallel runs happening, and data can be acquired easily. But then data, when we provide to you back, can you make sense of the data meaningfully and make some active interpretation? We'll give you some small files, not very big file, but we would like to do the exercise so that you at the end can reach out to some conclusion, and then we'll be happy to conclude this workshop and say that you have done something, and then I will ask Mr. Banju to release the certificate which you have prepared. But I think till then, if you get certificate, that will be not any value to you, and that will reduce our quality as well, that we have got certificate just because somebody was sitting in the room, right? So, and those who are really serious, who want to learn more, my TAs are here, they will sit with you and they can walk you through much more detailed manner. For others, I think we can stop the workshop. If you have questions, please raise hand and I can take it in. And please, as I said, next time onwards, all of you are the best hopes for the country, whether they are engineers or the medical students. Your time is very precious. When you come for any event, any activities, come prepared. Try to get best use of it. Okay? Otherwise, you spend your time also and you don't get anything out of it. Just one thing, like one report or one paper can make you so different, so much employable and so much in demand, you have no idea. Right away from the millions, you will be in just a few hundred. Right? And this is the first step toward that, to think like a researcher. And sooner you learn that, you have more time to easily absorb that. I think, in many institutes, even for medical students, they started implementing some of the research projects in the beginning, in the first year, second year, so that they start thinking, things slightly differently. Otherwise, you'll just look at, in a very objective manner. Going forward, your physiology knowledge and anatomy knowledge may not be so huge. ChatGPT and Gemini and Cloud, they will tell you the things much faster. Right? But your ability of comprehending the data, that's what will make you different. Can we still supersede the AI? Only way is if we can think differently and if we can think intellectually. And experimental design AI cannot tell you. You only can tell. For this disease, these are the right control. What should be by N? What should be sample size? How I should store my sample? How I should perform analysis? Only human brain can tell. Still AI cannot tell. And that's where I think your understanding and your concept are very important. Okay? Any question? Please raise hands.
Sir, how much this test cost to us?
It could be as low as 3,000 to 5,000 rupees, as low as this. All right?
Per sample?
Of course, per sample.
And we can ask for our sample to...
Any sample, I'm just saying, just think about it, how much we have brought things down. And we are generously funded by government, industry, all of these places, and we do provide that support to everybody else. It's not very difficult to do these things, guys, okay? And if you can do good project, you have no idea what the value it will add to you. And knowledge which you obtain will be, of course, immense value for your patient eventually. So just for your idea, I showed you multiple mass specs, right, in the facilities. One is our mass fit B facility, mass spectrometry facility at IIT Bombay. You go to that website, you will have the rate chart for every single analysis, which we run as external services. Most of the time, we prefer that you send your samples to us, and we only do the mass spec data acquisition, the instrument part. But sometime, let's say Professor Banju says that, no, can you help out in this part of, some part of the sample prep or digestion or something else, on that one-to-one collaborative basis, we can help that as well, right? But that we cannot do like for general, all country and many other international labs. But this could be done as well. But eventually we always, what I prefer, I have tried to at least build lot of proteomics lab around country. Many places we have stepped up Just because the PIs were not trained earlier and they had no idea how to do it, I conducted many faculty development program, which are funded by DST, University of Science Technology, 2 weeks, 3 weeks long training program. 25 faculty selected, they used to come to our lab and they get trained. We force them to do everything because PIs at the end, like, we don't want to do many things, right? But then if you do the things, you break the barrier, you remove the hesitation, and then they were able to take better decision and start the proteomics in their lab. So now many labs have started using it, but they don't have the facilities, which send the samples to us, right? So, of course, and we help you, and I really force you to do the things here. I can do once for you, but for how many times I should do it? And if you can do it, it is as simple as you have taken your plasma sample for your normal measurements, right? Just took 100 microliter of it, add 400 microliter of ethanol, and just mix it, keep overnight in the freezer, and just like this, send to us. Come on, you got all metabolite. How difficult it was? So you just need to know the right protocol. We will help you to get the right protocol and prepare you and step you up rather than we do the work for you. Okay? But at the end, data acquisition is very simple. There is some charges involved because we are also following the DBT and IIT Bombay and all the guidelines. But we go as probably the lowest in the country or anywhere in the world, for that matter. Even we have a startup company, Proteomica. In that, we are offering a lower charges than even what we have in our IIT Bombay facility. And that is one of the best instruments in the country in terms of full genomics. So what I'm saying, like, it's not very costly. Even your normal genomics measurement will be much more costly and chargeable as compared to what we are charging for proteomics and metabolomics. So do not worry about data acquisition part. Good question, good experimental design, right type of sample preparation, all that is your intellectual ability, your homework, which you do it here. Next part will help you out. We'll again handhold you for doing some data analysis, but then you should make the sense of data. We can't help you to make the sense of data. Because you know your disease better than us. Why we have to learn every disease? And then you will say, okay, is that really making sense or not? Okay, all right? So I request that, let's make a WhatsApp group where we'll give you guys a week time. We'll give you some tutorials to watch and then give you some data analysis file. Try it out. As I said, there's nothing wrong. Just take those screenshots and put it all together. Submit as a report. Okay? Sometime we give you some questions of quiz as well, not just you will just copy your friend's screenshot or somebody else and you will get it done. So based on that, then I'm hoping everybody will pass and all of you will be then so happy that you have achieved something. Okay? And you have lived up to what your level is and we have lived up to what we came for. Otherwise, this workshop was of no use. I could have given my 2 hours lecture here and could have gone. I did not give my lecture, I have prepared a long lecture. Right? But I thought lecture is not so useful as compared to for this kind of workshop when you say it's more like discussion, interaction, something you do on the hands-on and you break the barrier of doing something. That's what is more workshop about. Okay, otherwise supposing we heard a lot of good lectures in the morning, right? Okay, so I think I'll stop it here, right? And yes.
एलिमेंट्स एनालिसिस या कुछ कैरेक्टराइजेशन करवाना हो क्योंकि आई एम ए केमिस्ट्री बैकग्राउंड्स तो कैन यू प्रोवाइड इन योर लेबोरेटरी
So at IIT Bombay, again guys, try to know the resources. Many of the institutes are very well funded by the government with the mandate to provide support to the whole country. There are many DST funded facilities which are known as SAF facilities, Sophisticated Analytical Instrument Facility. Look around, something may be around in IIT Indore, maybe something might be around you here. I'm just saying, of course at IIT Bombay we also have it. Okay, so you can look at the SAF, SAIEF facilities, which is in around country, government puts crores and crores of money to upgrade those facilities so that population nearby can all make use of those instruments. Everybody can't buy it and can't maintain it and then don't use it all the time. But elemental analysis is very much possible. Okay, so feel free to check IIT Bombay's CRNTS and SAF facility website and you will find too many tests and everything will be very, very low cost. Any other observation or comments or questions? Okay, so at the end, those who have been here, I hope you got something useful. And those who also want beyond useful, more certificates, you have to do some extra work and do some assignment for us, which will do also some extra work to look at your assignment. And then we can conclude that we have done the workshop with IIT Bombay. And some of you who are actually researcher already doing something and you want to interact with my team, and want to do actual analysis, they will be here to provide you help. All right, thank you very much, everyone.
Thank you very much, Dr. Srivastav and his team, Dr. Anurag and all his team for such an extensive and interactive work session. And with this, we conclude our day, which was really full of. Dr. Mahadik is around and he wanted to say something before I conclude.
So one thing I must say, he should give you motivation to all of you. Like, he is your inspiration, right? I saw he is probably most sincere student sitting in the crowd, who has been listening and observing everything so much, with the value, and that's what makes him visionary, who can see the things better.
Sanjeev, I was with the team, and there in the biochemistry lab.
Okay.
The person from KI, from the Swedish Institute of Technology.
Oh, okay. Lochem, right?
Yeah. So he suggested that we do whatever the analyzers have, is sampling of say 1,000, 10,000, whatever number of people, and do the biobanking and do the test also. So I said, well, we can do it. So, what is your opinion?
No, I think as I've been keep talking, having the well-annotated sample on which we have good metadata, we have good observation, as Dr. Manju, it looks like you already thought about it and you already started those longitudinal samples and storage properly. Also, please keep in mind, as I was just mentioning, the storage condition becomes very critical for beyond genome. Like for just looking at DNA, anything is stable. When you go beyond DNA, RNA, protein, metabolite, they are very labile. So, if the goal is to look at many things, then one should do the storage in a very, very precise and controlled condition, very uniformly. So, we should come up with the SOPs, the standard operating protocol. Anybody who is storing samples should follow that. Otherwise, we will see a lot of variation, including at TMS repository or anywhere biorepository. A lot of governments have given biobanking. But the problem is they are not reliable. And why we see that? Too much heterogeneity. And because it was not thought of, anything left over, they just dump it. But if we think about as a good research resource, we should make a good SOP. You just need one person to be actually looking after the facility, who will have SOP that, okay, if you are taking the fresh frozen tissue, within 30 minutes of this, it has been, the frozen in the liquid nitrogen and came to the freezers and stored. And likewise, in your plasma sample, how we store. Even the kind of tube which we use, even that makes difference. In which kind of tube you store. Because when you see a lot of peaks in the mass spec, many times those are actually contaminant. They are coming from the different, things which you don't want to see them, right? So, all of these are very critical.
If we store them in protein cell card or DNA or RNA cell card.
So they are pretty good for definitely the DNA, right? They work very well. But they are not so good for many other like a dynamic molecule. When you go beyond DNA to RNA to protein to metabolite, they may not be so functional and so good. Although some limited way one could still get something out of them, but not very robust way. But if we can just give little thought to begin with, all the efforts could be like diamond, if we can really store them well. But if we are just storing it, then there is so much chunk of the junk tissue available everywhere, which will not yield anything. So problem is people don't have the confidence or respect for the biorepositories or biobanks in country that can you get something confident.
We are following the London Biobank.
No, thank you. I see you are always very progressive, thought process, and only my request will be, if we can, or some technical people can also be involved into deciding the SOPs, in which way we should be sampling the things, in what kind of conditions should be there. As I said, even sometime when I do project with multiple hospitals, we send all the reagents from our lab. We send the same tube to all the 5 centers to avoid any tube variability. In the COVID time, my students and the staff they went to the KEM and worked in the BSL-3 labs, trained their technician so that the storage conditions remain uniform. Because otherwise things will be very different. So, but it's just little effort required, and I'm sure you're already doing it. But
what is the sample size you think is a good one?
So there is, never end to this thing, right? And that's where different. I have seen something in Korea was very beautifully made, good biobanking with very well annotated sample. Maybe several 100,000 kind of numbers they had, but very well annotated. And a small country, but it is a very well organized.
Now for this biochemistry thing.
So again, if you can put some sort of disease focus, then it is much easier, because then you say, okay, I'm looking at only disease X, Y, Z. For that, maybe even 50,000 is a good number. If you say I'm looking at all India problem, then even 500,000 is not a good number. So one could actually start with, I would suggest look at more Malwa region specific problems, more specific. I don't know, is there anything very high incidence of cancer here? I had gone to AIIMS Bhopal and we see the gallbladder cancer was very high. 30-40% patients were all gallbladder. So is there something which we see very high incidence of some diseases happening around here? Those could be good biobank to begin with. And one could then build further from it. But this is actually an excellent idea.
You know that we have a Palwapli lab. So a population of 90,000. And there we can always do longitudinal study.
That's right.
So what is your suggestion for that population?
So do we see some sort of the genetic issues coming across that, and we always see that observation, or is it very heterogeneous?
No.
Or is it like the general population? Right. Yeah. So this is something, I would say the kind of effort what you're talking is actually very big. Something like that is happening in one of my projects known as the Human Phenome Project, which is also in the different countries, Singapore and Imperial London and all that. What they do, they look at just seemingly healthy individual, normal individual, but you have like 90,000 individual, and look at all their samples at maybe now 6 months later and 1 year later, and then look at their different profile. Eventually follow them up and say, okay, some of them have developed diabetes, some of them have developed cancer, some of them have developed this disease. Then you go back to the previous time point and see if the switch at which this disease occurred. But that's, as I said, very heavy investment of your resource and the overall system, because you are collecting those 90,000 individuals, all their samples over the next 5-10 years time, and keep storing and keep analyzing all that. Only eventually you will have the answer, maybe after 10 years, 15 years, that some of them were actually normal, but what was the switch point between normal to diabetes or normal to hypertension, normal to cancer, and those triggers which have led to that. I would say to begin with, we should show good proof of concept on focusing one or two good disease where your hospital has more specialization and focus, or this region has more problem, and that could set up a role model standard, which could be pitching to government to set up the much bigger things for the larger population. But this is, I think, something which is what one displays from progress.
The next thing you were asking the students.
Correct.
So I was out. So what was that?
I was saying that at IIT Bombay, all the students undergrad first year, we teach them bio. We should not teach them bio. They are all engineers going to be, right? But why we teach them bio? Because IIT Bombay feels right investment of giving the cross-pollination, this idea of fertilizing, that why bio can be important for them. And many of them actually have turned out to be very good biologists and commercial biologists, which biologists cannot do. Many of them do projects with us, many of them do PhD. One of my students, like an undergrad level, has become faculty back to IIT Bombay. So this is something which we inculcate in beginning. At this level of medical students, we should bring some more technical things to them, rather than just giving them the medical information. More some of the R&D things, so that they should look around much beyond their bookish knowledge. And that broader perspective is what will grow them much better. And again, something, just small credits which can help them do seriously will be, I think, a good way in the first, second year, so that they're not very busy that time. But they're thinking slightly more. And if they can do some small project, independent project, we'll actually start them think, how to design the experiment, how to conclude the experiment, what is the short back.
The stories we declared, say, internal grants for UG students 50,000, for PG students 1 lakh, and for faculty 2 lakhs. small projects. But the irony of the thing is that only till it is 2 years and only 7 have applied.
But I suggest you, truly speaking, it will be zero cost project, zero cost. You don't have to give anybody any money, and everybody will be able to do good project, just computational based projects. And we have to give them independent problem, and you will see they will go and search the data, and for every disease, lot of data sets have been made available. It is now mandatory when we publish any data, it's mandatory now to put that data in open access repositories. So one could give them, let's say team projects. Depending on their interest, one could make 10 teams in a class, and each team has given a specific problem. Now their investigation is read literature, go find out right data set, download data set, analyze data set, make interpretation and make report and present that. Zero investment, you don't have to give any money. And those among the 10 teams, maybe those 3 teams which have come now with a substantial hypothesis, those 3 teams can be given money to now test their hypothesis. And now take some samples and go test hypothesis. And 50,000 is decent money for that matter. You can analyze 20-30 samples and prove their this thing. As I said, they can publish some paper. There have been students from KM Hospital and all who have gone to US, some of them become faculty at Harvard. I have been so happy. This one paper has made the change in somebody's career. And medical professional, because you are not supposed to do research. If you can do some research, it will make you very different. So I think it's a good idea to inculcate that in the beginning, and that when we were talking, we thought at least when we are coming here, we should try to preempt them. Of course, we can train them, but we can preempt. But I'm very happy to help us for whatever way we can do from our side.
Thank you so much.
Thank you.
Thank you, sir. Thank you all the faculties who are really making this symposium grateful, and all the students and future doctors, future pathologists, future clinicians. Thank you very much for all the day. Interesting. And now you all are invited. Now you all are invited for a high tea, and you all are invited to have discussion with Dr. Srivastava if you want.
And as I said, I would request your team to provide a link to all of them to join a WhatsApp group, and all of them will be given some assignment. And after successful submission assignment, then I think we should teach visual language.
So please give your numbers to Dipanshu.
Or you can simply generate one link and all.
Yeah, that is there. Yeah, that is there.
I'm sure make it.
Thank you very much.
All right, well, thank you everyone. I teach little hard way, but I hope at least that force some of you to realize the potential. I'm sure if you can do this more, you will be much more prepared, and you will then enjoy that what you have been talking. You can do very well. All right. Thank you very much.